we're going to start with, if you would all rise, if you are able, and place your hand on your heart, and we will recite the Pledge of Allegiance. Thank you. Scott, would you lead us, please? To the flag of the United States of America and to the republic for which it stands, one nation, under God, indivisible, with liberty and justice for all. Scott Tosher, would you please take the roll? Thank you.
Iyad Al-Masri, Kim Barrett, Kim.
I am here.
The record reflects Kim is here. Dan Bernal, George Blumenthal,
Here.
Maria Bonneville,
Here.
John Carruthers,
Here.
Marguerite Casillas,
Here.
Judy Chow,
Here.
Leandra Clark Harvey.
Here.
I think I heard a here, but the volume is a little low.
Here.
Yeah, the volume is very quiet. Shannon Dahl,
Here.
Monica Carson for Deborah Dees.
Here.
Thank you. Anne Marie Deliege.
Here.
Isabel Durone.
Here.
Mark Fisher Colbury.
Here.
Thank you. Elena Flowers.
Present.
Judy Gasson.
Here.
Vito Imbasiani. Rich Lahara. Sorry, Rich, I didn't catch you. Rich's mic seems to be muted. I'll come back. But the record
I'm here. Can you hear me now?
Great. We can hear you great. Thank you, Rich. Pat Levitt.
Here.
Hala Madnat.
Here.
Linda Malkus.
Here.
Shlomo Melmed. Carolyn Meltzer.
Present.
Chris Miaskowski.
Here.
Adriana Padilla.
Here.
Joe Panetta. Joyce Saki.
Here.
Marv Southern.
Here.
Shauna Stark.
Here.
Carol Watson. Yael White.
Here.
Kevin Zhu.
Here.
And Keith Yamamoto. Great. Thank you, Mr. Chair. We have a quorum.
Thank you. I'd like to apprise the board of just some recent activities. A few weeks ago, your president and CEO, Jonathan Thomas, and I had the pleasure of traveling down to the Lundquist Institute, which is located on the grounds of Harbor UCLA Hospital and Medical Center in Torrance, California, south of downtown LA, for the very happy occasion of their ribbon cutting to open and rededicate the Shared Research Lab. In attendance was the benefactress, Melanie Lundquist, PIs and researchers at the Institute, postdocs, students, and a good smattering of the architects who built this beautiful facility. After greetings, we had a tour of their refurbished and up-to-date lab spaces, presentations by the main investigators there, and JT and I spoke to laud them and their accomplishments to date and the future promises of the Lundquist Institute. Secondly, as part of my own personal goal this year to expand outreach and spread the message of CIRM to the citizens of this great state that support us with their tax dollars, I gave the keynote address to the annual convention of the Veterans of Foreign Wars in Sacramento a week ago. There were 450 members of the VFW in the audience, quite a large audience for me to do this speech. They represented every county, city, and town in the state. And it was a good event, and one of the major consequences of the speech is invitations by many of the members there to reprise the speech at their own local post in all the various corners of the state. I'm going to repeat this effort in 2 days and Saturday to a similar audience in Visalia, which is the annual convention of the American Legion. You see a common theme there. I'm going to keep the chair's report short today because We have a very full agenda. I want to keep us on time. Thank you. Going to be followed by the Vice Chair, Maria Bonneville.
Thank you, Vito. The last 3 months have been packed with meetings and outreach opportunities, including Rotary clubs, conferences, workshops, and congressional visits. I'll highlight just a few of the meetings. In April, I was in DC and visited with several congressional offices, both in and out of California. These visits provide an opportunity to tell CIRM's story and emphasize areas of research funding important to both CIRM and the members with whom we meet. They continue to be very positive meetings. At the ASGCT annual meeting, I moderated a panel exploring the many challenges to access and affordability of cell and gene therapies. The panel brought together a diverse set of stakeholders to explore issues from different vantage points, including the commercial payer landscape, highlighting how different coverage standards and financing structures create acute challenges for small self-insured employers ill-equipped to absorb the cost associated with rare disease gene therapies. Medicare and Medicaid, tracing how FDA oversight and clinical trial data requirements shape reimbursement pathways, and understanding how lengthy Medicaid coverage analysis delays patient access. The administrative burden hospitals face in offering cell and gene therapies, from navigating single case agreements to facilitating cross-state Medicaid care within a center of excellence model, and the broader cultural shift required as therapies like CAR-T expand into community hospitals and cancer centers. And the CMMI CGT access model and the evolving policy landscape as approvals multiply and CGTs begin to extend beyond rare disease indications. Next, I participated on a panel organized by our former colleague, Abla Creasy at LA Best. The panel convened a mix of perspectives, examining what it will take to move the field towards sustainable, equitable patient access. The panel advised on 3 core strategies: designing patient access into drug development from the start, building scalable, repeatable delivery systems versus one-off customized interventions, and evaluating these therapies based on their long-term lifetime value rather than upfront cost. Underlying all 3 is a call for shared accountability across industry, government, payers, and patient groups. California has been proposed as a model to demonstrate how these ideas can be integrated into a coherent system. And last, Sham and I attended a meeting in DC which convened stakeholders and was focused more on reimbursement and insurance challenges. This conference arrived at many of the same conclusions, but delved further into finance models and risk pooling. All of these activities expand our network of stakeholders, including healthcare providers, payers, patient advocacy groups, and industry partners with whom we can collaborate to develop meaningful solutions to the complicated CGT access and affordability landscape. Thank you.
Thank you. I'm going to be followed now by our President and CEO, Jonathan Thomas. If you come to the podium for this tripartite presentation, and you'll introduce the following speakers.
Mr. Chair, Madam Vice Chair, esteemed members of the board, great members of our team, and members of the public, it's a pleasure to present President's report today for this fine June day. We're going to have 3 different speakers that are going to come up to give you some updates on some very important matters. And in order, this will be Dr. Rosa Canet Aviles speaking about our long-term budget forecast, Dr. Joe Gold on a number of updates of companies and where things are happening with some of our awardees. And last but not least, Sham Patel, Dr. Patel, coming up to talk about a most productive trip that a number of us took to visit the CCCEs who are kicking off their respective programs under our CCCE overall program. So I am going to, without further ado, note that you'll recall back in September, I presented to the board a long-term forecast which contemplated spending down remaining Prop 14 funds over a 6-year period. We have taken a very detailed look at this to determine if that's the optimal stretch to be considering, and the team and through consultation with members of the board, have determined that rather than a 6-year forecast, we foresee an 8-year forecast. Now, mind you, this was in September, so it's actually now 5 versus 7 years. But towards that end, I'd like Dr. Canet Aviles to come up here and present to you a short presentation which details our thinking on the subject. Rosa?
Thank you, JP. Mr. Chairman, Madam Vice Chair, distinguished members of the board, it's a pleasure to come here today to present this updated forecast. So here's the clicker. And I want to thank my colleague, Ms. Jennifer Lewis, for her partnership in putting this together as well. So as JT was saying, we are aligning our long-term funding strategy with the impact to patients. And these two messages are going to come in the next slide. So when the strategic allocation framework was approved about 3 years ago now, we developed a plan that could allow us to move funding into the field in a quick way. Over the past year and a half, as you very well know, because you've been approving all these programs, we've been deploying programs, and we've been also at the same time evaluating the portfolio and where the opportunities will be likely to emerge. And some of these portfolio analysis is actually coming in September, as you will see. What became clear as we were doing all these analysis is that to maximize the patient impact to Californians, it's not all about the pace of spending, it's about having also a flexibility to invest when the opportunities are ready and when the programs are mature and where the potential impact will be bigger. So this 8-year forecast shifts us from a more compressed model to a more readiness and opportunity picking a base model. It gives us more flexibility to respond to emerging opportunities, keep the continuity in the pipeline, and adapt to new technologies as the field is evolving. And there's also an aspect of rebalancing the funding where the costs are higher. This next slide shows us what JT was mentioning just a minute ago, the original forecast that was approved as part of the SAF from now till fiscal year 30 to 31, as we originally forecasted. The plan concentrated the funding into a relatively short time frame, which is shown now for the actual from now till the end. This approach assumed a more compressed timeline, assumed more equal numbers for opportunity development that we believe is not the optimal way to do it as of today's analysis. So this is the current investment revised funding revised forecast. It extends the deployment horizon by approximately 2 years, up to fiscal year 32-33, but maintaining the same overall level of funding. The main difference here is not the total amount invested, but the timing and the distribution of the fundings. This revised plan gives us more flexibility to align the funding with scientific readiness, as well as the needs that come from the portfolio as it's advancing. And very importantly, I think, it gives us the ability to support future opportunities that we can anticipate well today, especially as technologies are evolving. So before moving into the details of the specific changes that we've made, I want to make sure that when you see the left that says pillars, I just want to clarify that does not correspond to the program. So as an example, PDEP, do not confuse PDEP with the PDEP program. The PDEP has 3 programs. It's the preclinical development pillar that contains the PDEP program, the RAPID program, and the technology development program that we haven't yet presented to the board and is coming in December. So just so clarify. So for the clinical pillar, we have increased the overall funding relative to the previous forecast and consolidated clinical fundings into the single pillar, and also moved some funding into later years, as this will be the clinical trials is what will be capturing up to the end of our funding. So we've added some funding at later years to capture opportunities within clinical. That's what's closer to patients till the end of our funding. And this gives us more flexibility as opportunities mature. It will also allow the September late stage portfolio discussion to help inform future funding decisions. For the preclinical, we have adjusted funding levels and timing to reflect the contribution of platform-based programs for genetic therapies through RAPID. So the relative between PDEP program and RAPID is now different because the RAPID is going to have the focus on genetic platforms, and usually that's for ultra-rare and rare diseases, right? So we are capturing that there. And then we have rebalanced the focus to support at the earlier stages. There is more money to support earlier programs. The earlier programs means that we go from development candidate up to IND. So it's the whole path. So at the beginning, earlier, we will have more earlier stage programs. So there's more money, while preserving the capacity for more advanced programs later that could then, hopefully, some of them would go into the clinical. That's why we have money also at the end for clinical. So for discovery, we have reduced the overall funding relative to the previous forecast. And this doesn't mean, this is very important, that we consider discovery as less important. We have basically rebalanced the fundings towards clinical, where the cost of advancing the therapies is a lot higher at those later stages. So as you will see, there is more for clinical, the preclinical stayed the same, and discovery has less. Now, this has been done through staging differently than this for program. We believe that we can achieve greater impact from discovery by reducing the frequency of the DIS4 awards and focusing on maximizing the value of the funded networks and the data generation and the downstream translation activities that are happening as we are funding this every year. And by comparison, and some of that is going to be presented by our colleague, Dr. Janie Byram today, who's going to present the Infrastructure 9 program about the data tools and software that we will generate to do that type of collaboration and data sharing and leveraging the resources. So the approach creates opportunities to partner also with other organizations and aligning with complementary larger scale discovery fundings, which is something that some of the members of our board have been asking us to do as well. So as we are generating this data, we have time also to talk to other partners and leverage. There's the Allen Institute, there's other organizations as well, Michael J. Fox and others, that we can partner with to leverage those fundings. And similar programs that we have modeled, the DIS4-2, operate on a multi-year rather than annual funding cycle. So I was involved, for example, in the Accelerated Medicines Partnership to which we modeled this. That was one every 5 years. It wasn't one every year. So that's the rationale for the discovery and how we have strategically distributed the funding here. For education, we have adjusted the timing of the updated scholars program, basically, and this is to better align with workforce development needs over time, and that's been part of some discussions with the board, public discussions. So as you will see in September, the scholars program is coming for presentation to the board for approval now this year. So we've accelerated that. So you will see this reflected in my colleague Ms. Lewis, today's budget. And lastly, for infrastructure, we have added funding for the Alpha clinics to maintain the clinical trial and patient access infrastructure that we've built across California, and that's led by my colleague, Dr. Shyam Patel. So overall, these changes are really about balancing the fundings across the pipeline, maintaining flexibility, and making sure we are positioned to support the strongest opportunities as they emerge. Thank you for listening. Dr. Blumenthal. Sorry.
Thank you. Quick question. This is really great. And I really do support this general framework. But I had a question about education. Mm-hmm. Because on the one hand, given the decline in federal funding for education, this is a time perhaps to accelerate education spending as suggested in that table. On the other hand, I am concerned about the fact that there could be a drop off at the end, so that there isn't really continuous support for students near the end of that 8-year period. Do you want to comment a little bit more about that?
Yes, thank you, Dr. Blumenthal. We have right now invested, so the $200 million on fiscal year 26-27 are the renewal of the, I'm trying to think which one that corresponds to. That's actually the hybrid program that we are just launching now. And that's going to be the renewal of the Compass and the Bridges. That's going to go for 5 years. The next one will be the Scholars. You do have a point, but I think it's up to the board to decide whether I think some of the workforce needs are going to be integrated into the pipeline of the pillars. So for example, for clinical, PDEP, and DISC, there are fellows and researchers that will be funded through that as well throughout the years. So I do think that it's a very important question you're asking. I do not have an answer for it. And I think it's probably a tough choice that the board will have to make, whether we go with the development of the therapies or we refund something at a later year, for example, when things finish at the 31st. There was never, sorry, I'm being long, but there was never sustainability for workforce development. So this is hard. Yeah, and thank you. Yes, Judy. Oh, sorry, Kim.
Now it's working. Great. So thank you very much for this thoughtful presentation. And I definitely understand and support the rationale to really prioritize the clinical development. But I think this needs to be carefully messaged in an environment where people are really struggling for research funding, and particularly for early stage discovery type projects.
Mm-hmm. And thank you, Dr. Barrett. I agree with you. And I think that I was trying to be very careful in the way that we are justifying the discovery. As you can see, like the total amount of in every one of the pillars is now more relative and more balanced to the cost of what that stage usually, and that was something we didn't take into account because I think we just didn't think about that carefully, and we are updating that here. But you're absolutely right, and I think a great partnership now that we have with our communications colleagues. We have Amy Adams and Scott here helping us, and the team of communications should probably partner with us. We will partner with them to communicate this. Thanks. Yes. Dr. Miaskowski.
Thank you. I have a question about the alpha clinics.
Yes.
And this is maybe a curiosity on my part. What plans are in place to understand the sustainability of these clinics when CIRM funding ends? Are there discussions happening about that?
There are. And as I was interim leading the patient access, we started very thoroughly thinking about this and understanding what the outcomes have been from the current Alpha Clinics investment that has been, I think, 15 years now. I would like to defer to my colleague, Dr. Patel, who's now leading this, to provide an answer. And perhaps when he presents the CCC, or if you want to come now, Sham, you can talk about that, because that's definitely that's a key question, and we have taken. And when I said Alpha Clinics funding, I did not say refunding. So I was very careful to say that. Please, Dr. Patel.
The Alpha Clinics network, one of the things that we're looking at is the increase in the trial capacity across all of those sites, and how that is sustained going forward. One of the things that the Alpha Clinics really ended up doing, our funding ended up doing, was providing some stability on the staff to support both the engagement with sponsors as well as the launch of those trials and support of them. And so we want to see, as we go forward here, what is required to keep that going, and also what additional functionality we can add to the network that would help patients in California access both clinical and commercial therapies. So over the next 6 months, we're going to be digging deep into that and then coming back to the board with a plan.
Good question.
Hmm.
Okay.
Hmm.
Thank you. Oh, Isabel, Ms. Duron.
That's okay. I'm actually projecting. And so I'm putting it on the table because given both cuts at the federal level that are going to impact state, given state reconsiderations with revenues, et cetera, and where they put them. I think, and this goes somewhat to Amy as well, but I think we as a board need to think about this in terms of communication outward. I mean, you said 2032. I thought we would be wrapping up sooner than that, but that's lovely. But I really do think we need to start thinking in terms of preparing our public for some wonderful discovery as we go, but also for the possibility, to your point, and to the fact that some care might actually be, the spigot might actually be turned off somewhat abruptly if we cannot come to new resolution and new funding. I just think that in order for the public not to be totally gobsmacked by a reality that happens to them later on the road, that perhaps Amy needs to think of a communications plan, and when we start to talk about limitations.
Yeah.
And about us trying to figure out the best things to do with what we have while we have it.
That's excellent. Yes, that's a very good point, Ms. Duron. I think that something that will complete a little bit the picture, and I wish it had been now in June, but it will come in September, is going to be showing what the wins of, or where are we going in terms of the later stage portfolio that we have. How are we going to achieve also these SAF goals and what to expect? And I think that will also help with messaging that we don't have an infinite faucet, right? And that's something that we will also partner with Ms. Adams in communicating, as you very well said. Thank you.
Thank you, Rosa. TT, you want to continue with?
Thank you, Rosa, and I want to double down on thanking Jen for all her hard work on this forecast. So thank you very much, Jen. At our last meeting, I was going to initiate as a sort of companion piece to the Closer for Cures segment, a reporting out of how a number of our awardees are doing to update key developments to the board so that you get an ongoing feel for where things stand. We're just about ready to deliver that when the rocket news hit, as you recall, at the end of the last meeting, which was the very important news of the day. And so this is going to be the inaugural effort. Towards that end, I've asked Dr. Gold to come to speak to you about 5 of our awardees and an update on where things stand. Before, Joe, you come up, I want to make one last comment on Rosa's presentation for newer members of the board. When she talks about we're accelerating funding for the Scholars Program, a lot of you probably don't know what that is, and that's something that's been around for a while, and it targets young faculty, which are the very faculty we're concerned about with the federal funding cuts. And so we're, as Jen will describe, allocating a considerable amount in this coming year immediately, directly to address that issue. So, Joe, please.
Thank you, JT, and good morning. JT asked me to specifically summarize some of the CIRM grantee activities at the American Society for Gene and Cell Therapy meeting in Boston last month. And of course, ASGCT is the premier showcase for developers of these type of treatments. And as you can imagine, CIRM grantees are very active at this meeting. And I don't have time to describe all the CIRM activities there because our grantees are a prolific bunch. And in fact, you're going to hear from some of our grantees later on in this meeting, which will be very exciting. But I did want to focus on a few of the clinical discussions, because I think they're quite impactful, and I think they mesh really nicely with two of our framework goals, namely that of advancing 4 to 7 rare disease therapies to the biological license application, the final stage for approval, and also for propelling 15 to 20 other disease therapies to late-stage clinical trials. So the first presentation I want to talk about came from our grantee, Tenaya Therapeutics. And Tenaya is targeting a rare cardiac disease called arrhythmogenic right ventricular cardiomyopathy. And this is a genetic condition where the structure of the heart actually changes and electrical signals don't promulgate very effectively, and it winds up leading to arrhythmias and heart failure and sudden cardiac death. And the majority of these cases are caused by mutations in a gene called PKP2. And when I say the majority, I mean something like over 40% of these patients have these mutations, and that means about 70,000 patients in the US. And what Tenaya is doing is they've developed a viral vector which is very effective at transferring the defective gene into the heart. And what they're finding in their early stage clinical trial results is that, first of all, the therapy is safe, which of course is the most important thing to discover first off, but also that the patients who are treated are showing a big decrease in both the number and severity of the arrhythmias. So this is quite exciting. And Tenaya is going to be meeting with the FDA later this year to talk about the design of their next trial, which should be their pivotal trial. In other words, that which is going to get them to full approval. So we'll keep an eye on that. Now, viral vectors are the workhorse of gene therapies, but there are some other approaches which are being used, and they can be advantageous for specific conditions. And one of our grantees is ImmuSoft Corporation. And ImmuSoft is targeting a rare disease called MPS 1. And this is a condition, a genetic condition in which cells lack an enzyme which allows them to break down specific types of sugars, meaning they build up in the cells, you get cell death, you get multi-organ system failure, you get intellectual disabilities, and in the worst cases, death. And there are 2 current therapies for this condition. The first is a bone marrow transplant, which it is curative, but we know about the caveats that come with bone marrow transplants. You have to find an appropriately matched donor. You have to typically treat the recipient with some pretty toxic drugs to precondition them to allow these transplants to engraft. You have to worry that the recipient is going to reject the transplant, so you have to keep them under the care with very powerful immunosuppressive drugs, potentially the rest of their life, which have some very severe side effects. And the worst case scenario is that the transplanted immune system attacks the host. And this is graft versus host disease, and it can be fatal. So the other treatment which is being used is less toxic, but also less effective. And this is weekly infusions of an enzyme going to the circulation. The problem with this treatment is that, aside from the fact that it's every week, is that the enzyme does not penetrate into the brain very effectively. So you're not really able to address the CNS aspects of the disease. Also, because you're giving 1 infusion per week, the levels of the enzyme rise and fall during that week. So it's very hard to control the level of this enzyme. So it's helpful, but it's not a cure. But ImmuSoft has taken an interesting approach. They start with the observation that our naturally occurring B cells are incredibly potent factories at churning out very large quantities of proteins and secreting them. And typically these are antibodies. But what ImmuSoft has done is they isolate B cells from the individual patients, and then they do a non-viral modification using something with the evocative name of the Sleeping Beauty transposon, which I could talk about later if anybody's interested. And they engineer these cells then to express very large quantities of this missing enzyme. And the benefits of this approach is that not only are these cells really potent at producing this enzyme, but B cells are long-lived and they're very good at penetrating into different tissues, including the brain. So the therapy gets access to all the tissues where you want it to be. And their initial clinical trial results show that, first of all, again, it's safe. Secondly, the cells are present. They've stayed engrafted. And they're showing decreases in the blood and cerebrospinal cord fluid levels of these sugars. And they're also seeing some impacts on some other symptoms. So again, very, very early results, but very exciting. Now, I mentioned graft versus host disease. Another of our grantees is TRIX1, and they have developed an off-the-shelf therapy to stop graft versus host disease. And in this case, they're starting with T cells from healthy donors, and they're virally engineering them to turn them into a type of immune cell called a TR1 cell that has the normal function of dampening down immune responses. So they're using these cells in a clinical trial with patients who've had to have bone marrow transplants due to cancer treatments. And as I said, it's an off-the-shelf therapy, so we can go right into these patients. And the results, again, safe, the cells are persisting, and they're getting some pretty good indications that tolerance may be being established early on. So again, that's exciting. And if this approach holds up, it potentially would have some applications well beyond GVHD. For example, you might imagine these cells might be useful in maintaining acceptance of other types of transplants, and they might play a role in dampening the effects of autoimmune diseases too. So again, something to keep an eye on. And okay, autoimmune disease. So another of our grantees is Fate Therapeutics, and they are doing something interesting. They're taking a treatment which was originally designed as a cancer treatment and turning it against lupus nephritis, which is an autoimmune disease. And what has been found in the last couple of years, which is really interesting, is that the same CAR Ts which are used in certain types of blood cancers seem to be very effective at putting lupus into remission. And this is because these CAR T cells are designed to kill off B cells, because those are the types of cells which become malignant in these types of blood cancers. But these are also the types of cells that go rogue in lupus, some other autoimmune diseases. And really the most interesting part here is that they have found that if you give these kind of treatments to lupus patients, most of the B cells are killed off, as you would anticipate, but eventually they come back. But for reasons we do not understand, the immune system has reset itself. So the B cells that come back don't cause lupus anymore. We don't know why. It's really, really interesting. But patients get back a normal complement of B cells. They have normal B cell mediated immunity. So this is great because it means that therapy does not have to last forever. In fact, there's a good rationale why you wouldn't want it to last forever, so they get their immune system back. Now, the problem with this approach and other people who are trying this is that it has all the downsides of typical CAR T production, meaning it's a one-off for each individual patient. You can't scale it up. You can't have it ready ahead of time. So that's a problem. But Fate has taken a very different and interesting route. They start with pluripotent stem cells, which they can convert into T cells. And they take these T cells and they engineer them in two ways. First, they knock out the normal T cell receptor in these T cells so they can't attack the host, so no graft versus host disease. But secondly, they introduce a receptor specific to kill off B cells. And what they have found in their early clinical trial results is, again, safety. Second, the cells persist long enough and they're seeing a knockdown of the patient's B cells. So it all looks really, really good. And Fate has already met with FDA and they've agreed that the next clinical trial will be their pivotal trial. So again, very exciting. The last group presentation I'll talk about came from Elpida Therapeutics and separately their founder, Terry Pivrolakis. And Elpida announced that their CIRM-funded CMT4J trial for rare disease is open, and they hope to be treating their first patient very, very soon. And Terry gave one of the keynote addresses at ASGCT, and he talked about his odyssey from being the parent to a child with a devastating rare disease to someone who became just a fierce and tireless advocate to develop a therapy for his child, now the kid with this disease, to going on and founding a not-for-profit company that is tackling 5 different rare diseases for kids. It was powerful, it was moving, and it just one of these jolts you need sometimes to remember that there are people out there who are really all in fighting the good fight, and that CIRM has an important role to play in supporting these efforts, both the rare diseases and more prevalent diseases. So just quickly in summary, I mentioned that we've got Tenaya, who is going to be going to FDA to talk about their next trials, a pivotal trial for a rare cardiac condition. We've got Fate, which has already got concurrence from FDA on the design of their trial for a common disease for lupus. We've got exciting clinical results from Immusoft and from TRIX1 that's going to propel them down the pathway towards later stage clinical trials. We've got Elpida opening up their trial too. And what I'm talking about here is just the things which are happening at ASGCT. There's a lot of other things going on behind the scenes in other formats. And what this really means is that we're building up a lot of momentum towards these strategic goals I was talking about. And the rest of this year and next year should be a really exciting time to see what's about to happen. So I would just tell everybody to stay tuned. Thank you.
Thanks very much, Joe. That was a great summary. By the way, looking quite dapper today, if I do say. The last speaker I would like to introduce, we've seen a minute ago, Dr. Patel is going to describe the trips made to the 3 CCCE sites and how those went and where they are in the rollout of their respective programs. Sham.
Thank you, JT, and thank you to the ICOC. I'm excited to be presenting to you today in my new capacity as the Patient Access Team Lead. That's my 4th role at CIRM. So I have a few slides to walk through site visits that we did with the Community Care Centers of Excellence. So as you know, these just launched this year. Can we go to the slides, please? Oh, it's on me. Thank you. It won't advance. So while those are advancing, there we go. So just as a reminder, the Community Care Centers of Excellence are mandated by Prop 14, and the intention was to expand the clinical network in California and to make both commercial and clinical stage cell and gene therapies accessible to patient populations that are underserved by the current infrastructure that we have in the state of California. And so they were designed to fund the delivery as well as all the training involved with launching cell and gene therapy trials and delivering these in a commercial capacity as well. So since we just launched the awards, the patient access team took a site visit to each of those 3 new facilities. And we also brought along many of our team members. And so I'll go through some of that, some of our observations, and really speak to the power of what is being built at each of these facilities. So these are the 3 facilities. I know it's a little bit hard to see, so I'm going to walk through that. So we funded 3 Community Care Centers of Excellence in Central and Southern California. One is the Community Health System in Fresno, Clovis, that serves the Central Valley. The second is the Lundquist Institute. You heard just a little bit about that from JT earlier. This is in the South Bay in Torrance, but it serves the South Los Angeles, East Los Angeles, and South Bay communities. And lastly is Loma Linda University. This is in the Inland Empire, and it serves both the Riverside and San Bernardino counties. These are massive counties in the state of California by area, stretching all the way from the outskirts of LA to Las Vegas. And so I'm going to go through in a little bit of detail about each of these facilities. But one thing I want to highlight is that in each of these 3 cases, it took the unyielding vision of one person to have the foresight that cell and gene therapies are going to be important for their patient communities and really building up all of the support they needed within the institution, leadership, the staff, as well as external stakeholders. And that's going to come across in the slides where they have built this partnership. Everybody's really excited about what they're doing, and they really want to bring these therapies to their patient community. So this is us at Loma Linda. Some of us dressed up in bunny suits. I think they made us do this on purpose just to see what it would be like. But we got to tour the surgical suites, and I'll describe that in a little bit. But we were very fortunate to be joined by many of our internal stakeholders, including JT, the grants management team, the communications team, the programs team, Dr. Joe Gold, who was very enamored with certain carpet designs in one of the facilities, as well as the patient access team. And so for us, site visits are important because we get to meet the teams, we get to see all the work in person, and we really just get to build relationships that we can then expand upon as we go forward. And so I'm going to start off with the community health system in Clovis, Fresno, and then go through the other 2. So here, Dr. Abdullah had a vision, and primary vision was to bring CAR T cell therapy treatments to local community there instead of having to travel 200 miles to go to the Bay Area or 200 miles to go to LA. And you can see that in that tiny map right there, that little blue dot in the middle, that's the authorized treatment center at Clovis, Fresno for the KITE T cell therapies. That allows those patients to get treated there instead of having to go to the Bay Area, which is that top blue spot, or to the LA or San Diego region, which is the bottom blue spot. And that is a big deal for these patients to be able to get treated there and have access to the cell and gene therapies. And so right now they are onboarding all major commercial cell and gene CAR T products while they build up their clinical trial infrastructure. We got to tour an old office building they're turning into a manufacturing facility that will allow them to manufacture a cell and gene therapy drug product for clinical trials. In collaboration with UC Davis. And they're working with several community partners, including Blood Cancer United. You may know them as Leukemia and Lymphoma Society for patient navigation and community engagement, as well as the Fresno Center, which is tied into local communities there in terms of training community health workers, as well as engagement with the community there, informing them about cell and gene therapy trials. I'm going to move on to the Loma Linda University. So as I mentioned, this serves two of the biggest counties by area in California, a huge catchment area. And Loma Linda is well on its way to being able to support both commercial and clinical cell and gene therapies. It's going to use CIRM funding to expand all of that operation. So first and foremost, they're an authorized treatment center for multiple commercial cell and gene therapies. This includes sickle cell gene therapies as well as CAR Ts. And as Nick told us during the presentation, there's a significant sickle cell population in their community. And so being able to provide that access to that cell and gene therapy there is important. They are also a treatment site for a number of neurological disorder clinical trials in cell and gene therapies. So in that picture, you see Dr. Bowling demonstrating to us image-guided neurosurgery. So Dr. Bowling has been instrumental in leading this institution to be able to support the delivery of cell and gene therapies for neurological disorders. This includes CIRM-funded therapies. We AskBio's gene therapy for Parkinson's disease, as well as Neurona cell therapy for epilepsy. These are complicated procedures that require MRI-guided surgery, as well as training in novel delivery devices and many, many robotic systems. And all of that has been enabled by the vision of Dr. Bowling. They're also developing a centralized clinical research administrative unit with a singular focus of cutting trial activation time down by half. And similarly to Clovis, they're building a manufacturing facility to be able to support cell and gene therapy clinical trials locally. Finally, they have several community partners that they're working with to train community health workers for their communities, and also to leverage data about engagement and to better inform future engagement. And lastly is the Lundquist Institute in my hometown of Torrance, California. This is a unique facility. So here the vision was to be able to provide cell and gene therapies to patients in an ambulatory outpatient setting, but that's their main focus. They're only going to support trials as well as commercial therapies that are delivered in an outpatient setting, and that's unique to our entire network. They're renovating an old conference room into a advanced pharmacy suite to be able to support cell and gene therapies. We got to see the room. It's going to look wonderful when it's a pharmacy. And they're also accelerating their timeline, which we love. So they're moving faster to be able to generate, to be able to support these types of therapies. And they're already started discussing with trial sponsors. And lastly, they're collaborating with several community partners in their community, including RFK Institute, the South Central Prevention Coalition, and Access Advocacy, to build multiple disease advocacy groups and boards to have real clear engagement around diseases that are important to their community. And they're also very passionate about working with us to engage our earlier stage preclinical programs to help really bring the patient voice into the trial design and trial execution. So we're really excited to be working with them going forward as well. So I'm going to end with a special shout out to Dr. Jeff Lomax, who many of you know. Jeff has been working at CIRM and has served CIRM for over 20 years. And sadly, he's retiring at the end of July. We will miss him. We want to recognize all the contributions that he's made to CIRM over the last 20 plus years, not only on the policy side, but also on the infrastructure side. So I mentioned the CCCE program. So Jeff led the scoping, the development, and the launch of this program. And so that's going to be his lasting legacy as he leaves CIRM here, is to see this program sort of grow up over the next few years. So I want to thank Jeff for all of his service, and we are very delighted to have worked with him over the years and wish him all the best as he moves on with his retirement. Thank you.
Shyam, don't run away too soon.
I thought I could escape.
First, congratulations, very exciting news. Thinking particularly about the community outreach, and I recognize some of those folks that you have, that you're working with. I think it would be really lovely if you add to the slide the demographic reach and catchment area of the patients in those particular areas, just so we know when we talk about these underserved populations, we really know who's in that catchment area and whether or not we can measure that, in fact, they're being caught.
Yeah, definitely. Yes.
Second of all, I think that it's really critical to have this kind of information point outward back through your community partners to those communities so they can see this is something they do need,
Yeah.
right? That it does impact them. And I think that using your community partners who are training what you call promotoras, AKA community health workers who are becoming the bridge across the country to reach vulnerable populations particularly, but to add to a shrinking labor force that, or one that needs to be built or rebuilt. So I think it would be critical. Finally, I hope that you're actually bringing them to these centers so they can see it as well
Yes.
and start to dream and believe.
Yes, definitely. I think there's going to be a component of that. And so a couple of points that I just want to make there. So one of the things that really heartened us, especially at the Lundquist Institute, where they had every single one of their external and internal stakeholders at the table. And so all those community partners, UCLA, everybody was there to have a really rich discussion. And one part of that that really came up there was because the clinicians were there, the clinicians were talking about how when they want to refer their patients, even for standard of care, to facilities like UCLA, City of Hope, and others in that region, they run into all kinds of access barriers around insurance coverage, particularly for the adult population. And sort of that led to a workflow for us as well, was to figure out what's happening in our trials. And then on the same side, as you mentioned with the community partners and the training with the community health workers, there was a discussion around, well, how do we all work with each other to use the appropriate training materials, right? And as well as what communities are we engaging and how do we measure all that? And so coming out of that is also what is our metrics for success on the community engagement side as well. So I think all this is going to be building as we go forward here and really to operationalize this network and to put all the learnings together and really take action going forward. So thank you so much for your feedback.
Dr. Patel, sorry. By the way, this is phenomenal. So I'm thrilled with this development network. I was very interested in the Central Valley outpatient one. What is the exact model? Is it an expert goes there for a period of time? I'm trying to think how the model works, or is it someone who's there all the time that can help deliver these when it's fully up and running? I'm asking that because I'm just intrigued if if we can learn from that and replicate that in other areas of relative rural settings, because a lot of these rurals don't necessarily have larger hospitals or hospital systems. They have, critical access hospitals, and this outpatient thing sounds just phenomenal. So I would love, once it's up and running, how it's replicated. So I was wondering, what's the care model? I can't imagine a high-end medical oncologist being there 100% of the time, but maybe there is. I don't know. I'm just asking.
Yeah, and so this is particular to the Lundquist Institute as well. I'll speak to that because I think that's the most relevant of your point here. So for the Lundquist Institute in Torrance, where they're going to be supporting us in the outpatient setting, they first have to start by training all of their staff on how to deliver these therapies. And so that's where they're working really tightly with the UCLA team on that training. So there's going to be some cross-training, having the Lundquist staff actually go to UCLA, get trained firsthand, and using a lot of the previous Alpha Clinic resources to be able to do that. And I think, as you mentioned, there's probably going to be some level of expert, coming there. And I think there's joint appointments between UCLA and Lundquist that does help with that. And then we're going to see how that plays out in that setting. But you're right, as we go further and further out, where there isn't a direct tie to a major medical institution, how does that play out? And I think this is going to inform us quite a bit on how we then scale out to other institutions.
I don't see any. Oh, yes, yeah.
I just wanted to say I love this. It's great. It's lovely to see it in action, and I love the thoughtfulness that's come of the conversations of being in person, and how this is going to grow and benefit. So thank you for sharing.
Thank you. And one of the things that really stood out for us was as we were doing the tour, I think it was at Loma Linda, and they spoke to a nurse, and She was just so much in awe of having delivered the first gene therapy at that institution. It was sickle cell gene therapy. And as we were walking away, they were mentioning that, the nursing staff here recognizes the impact of these therapies, but it is hard. These are really complicated therapies that require a lot of training and then a lot of monitoring. So it's incredibly hard to deliver these therapies, but at the same time, they recognize the impact.
Ditto, this is just amazing. This is incredible. So congratulations to you,
To the microphone.
congratulations to you and your team for this incredible launch. It's very inspiring. I guess my question has to do with how CIRM is encouraging these centers to think about a sustainability plan that goes beyond the CIRM funding.
Yes, great point. So one of the things that's instrumental in all three of these facilities is that there's full leadership buy-in to the CEOs of all three institutions who were present at these meetings. And they also had committed funding to build out the infrastructure. I think going forward, it's going to be a combination of the ability to deliver the commercial therapy and how that plays out for their economics. And on the clinical side, it's going to be a steady flow of trials coming their way. But you're right, the sustainability is really going to be critical. And especially one of the things that we really want to be careful of is what Isabel mentioned in terms of the training programs and that engagement and what happens to that, because that's not a revenue making system right there, right? And how do we sustain that going forward?
Dean?
I think this is really fantastic. And I remember reading these grant applications and being really, really excited. So I'm thinking about the broader medical and nursing community and lessons learned, right? The example you gave of the nurse. I guess, what is our obligation to disseminate what we're learning in publications to the broader community? Because I can imagine academic medical centers saying, we can only do this at a UCSF. I'm using my institution as an example. And we're proving them wrong.
Yeah.
And I really think there needs to be articles about the principles that we've applied, what we're learning. I was thinking as Maria gave her presentation about access and affordability, those are really important concepts that we should claim as ours and put them in medical journals, nursing journals to say, this is possible. Communities can learn this. Primary care docs can refer. Nurses can be engaged in this. So I don't know if there's plans for that, but I would really like to encourage us to do that because I think it's going to be a really important service to the nation.
Thank you for your encouragement. I think that's an important part of disseminating learnings going forward. So as we were talking earlier to John's question about those facilities, one of the things that came out of there was that every single manufacturer for those CAR T therapies has their own requirements, and they had to sort of manage all of that in different ways and build all that learning and infrastructure in place. And that's sorts of learning that could be going out as well, in addition to the training of the nursing staff and the clinical staff. And I think it's important for us to be able to gather all that and disseminate it in the appropriate platforms. Thank you for raising that. It's definitely something we'd love to work with you going forward on.
To build on that, I also wanted to see that we have articles written about our outreach to underserved populations, because I think if we are able to show that our programs, particularly in the Central Valley, are reaching underserved populations that are usually ignored, that would make a very powerful impact for an article.
Thank you. And as you've learned over the last few months, Amy's always a step ahead. So she and her team had independent conversations with all three CCCEs when we were down there to figure out a communication strategy.
So the word I would use is template. You're building templates. And I think that could be a very, very powerful thing, not just for California, but for the nation as a whole. So I'd start, if you want to use a word, I would start saying we're building the template for delivering this kind of care into a new setting, really. So that's just my recommendation.
Thank you, Dr. Malkus. I'll be using that word endlessly going forward.
Sham, I see a lot of writing in your future.
Thank you.
Any other?
I'm going to add
Elle?
one word to Linda. Toolkits. Develop the toolkits that can then be disseminated, particularly in the community level. Those would be excellent.
Yes,
And I'll be glad to help disseminate them.
of course. Thank you so much.
Okay. If there are no other questions, I don't see any on the line. So JT, you want to?
So I'm just going to wrap up here. We're going to, in the interest of time, I had some comments on AI. We're going to defer that till our next meeting. And I want to thank all three of our speakers and all members of the team who were instrumental in getting all the work done that was discussed today. And I just want to close in my own shout out to Jeff. I know you're watching. You've been a stellar member of the team for many, many years. Thank you for all your hard work and dedication, and we'll definitely miss you very much. Thank you, Mr. President. Mr. Chairman.
Thank you, JT. That was a series of wonderful presentations. Thank you, everyone. We can move now to the part of the agenda called the consent agenda. Today we have 5 items, and that would be the minutes from the last meeting, the consideration of 5 new appointments and 2 reappointments to the scientific members of our grants working groups, amendments to the preclinical development concept plan and the RAPID program for rare disease, revisions to the award management policy, and the usual request to attend remotely. The way the consent agenda is, I would like a motion to accept all of these without further discussion, unless a member of the board wants to abstract an item or more.
Move approval.
We have a motion second. Is there any discussion on that? Any further abstractions? No? None. Any member of the public have a comment on this? If not, Scott, you can proceed to a vote.
Okay, I'll do a voice vote in the room, but I must poll individual members who are participating via Zoom. So all those in the room in favor say aye.
Aye.
Those opposed, nay. Any abstentions? And on the phone, Leandra Clark Harvey.
Aye.
Thank you. Monica Carson.
Aye.
Mark Fisher Colbury.
Aye.
Rich Lahara.
Aye.
Carolyn Meltzer.
Aye.
Shauna Stark.
Aye.
Kevin Zhu.
Aye.
Great. Thank you, Mr. Chair. Motion carries.
We can progress now to agenda item number 11. We're now going to consider the applications submitted in response to our preclinical development program, the PDEV program. This is the work of the Applications Review Subcommittee, and Dr. Haley Lim will approach the podium and give the presentation. Thank you, Haley.
Good morning. It's my pleasure today to present to the board the Grants Working Group recommendations for the PDEV preclinical program. So I'll be presenting a brief overview of the program and then walking you through the process by which we arrived here today with the recommendations for your approval. As you know, we have our 6 impact goals here that have allowed us to develop and launch several programs up to now, and many planned as well. These were established in 2024. The PDEV program aligns with, oh, I went back instead of forward. Here we go. The PDEV program aligns with Goal 4, propelling 15 to 20 therapies targeting diseases affecting Californians. And the PDEV program itself is aimed at accelerating preclinical development and prioritization of innovative therapies for Californians. And so as such, the objective of the program is to accelerate completion of preclinical development, FDA IND clearance, and clinical trial startup for stem cell-based and genetic therapies. In broad, oh, I keep pressing the wrong direction. Okay. Here we go. The overall structure of the program for this current fiscal year is 2 iterations of this program. This is the second for the closing fiscal year. And the program is allows a maximum duration of 5 years and up to 13 million in award amounts and varying co-funding amounts depending on the applicant type. We projected with a 160 million total initial allocation, approximately somewhere between 12 to 21 total awards across both rounds. I'll speak to a little bit more of this later, but we have already, as you may recall, in December, approved 12 applications against this 160 million budget already. And so what you're seeing today is against the remaining 42 million or so for the 160 out of the 160 initial allocation. So a little bit about the award structure. So to accelerate the preclinical development, the program is designed to allow flexibility in entry points. So we allow 2 entry points to this program, but a singular outcome, which is that expected FDA IND clearance at the end of the award. So depending on what stage that the applicant comes in at, they can come in at what we call the early stage. These applicants have early proof of concept with their candidate in the indication that they're aiming for, or the applicants may be further down in their development pathway and come in at what we term the late stage PDEV awards. And these folks have already had a pre-IND meeting with the FDA, gotten feedback, and are working on their sort of IND enabling studies that will allow them to get to that IND clearance at the end of the award. And as you can see, for each of these stages, they have budget limits and durations for each of those stages. But again, I want to emphasize, regardless of where these applicants come in, the end goal for all PDEV awards is to achieve that IND clearance. And I also want to take a moment here to emphasize that, these are 5-year awards for up to 13 million. And so these applicants that are coming in at that early stage, the CIRM staff and team are really dedicated to working with these awardees to develop their projects. And for those that come in again at that early stage, there is that important inflection point between the early and late stages where the CIRM team will be working with the awardees on their pre-IND meeting strategy, sitting in on those pre-IND meetings with the awardees, and then based on the feedback from the FDA, working with the awardees to adjust the milestones for that award, as well as award disbursements for the remainder of that project. Transitioning to the review process by which we bring these applications to you. The PDEV program for the second round had pre-submissions by which we applied our program preferences. The top 30 or so applications advance to full applications, come into CIRM. We conduct our typical eligibility screening. The applications then go to merit review, the scientific review, and then the applications out of there come to here for recommendation and final funding decisions. And so just to give a couple of numbers here for this current round under proposal here, we had 126 pre-submissions, 23 were invited to submit full applications. Ultimately, 22 of those went to scientific merit review, and 8 of those were recommended for funding by the grants working group. And how did they arrive at those 8 projects? The panel is composed of essentially subject matter experts across many different areas, as well as our board members that are patient advocate and nurse members of our board. The expertise spans scientific area expertise in specific disease areas, regulatory science, manufacturing, as well as product development. And of course, our board members provide the patient perspective on the projects. In terms of the scientific scoring of these applications, we use for all of our programs is a scale of 1 to 100 with a median score of 85 and above, a recommendation for funding, and a score of 1 to 84, not a recommendation for funding. I will emphasize here that the scoring is meant to be holistic. It encompasses all of the review criteria and all facets of the review. And we encourage our reviewers to score across the entire range to signal sort of their enthusiasm for the projects. And the review criteria against which they evaluated the applications are listed here. Broadly speaking, we have 5 review criteria: the value proposition, the rationale, the project plan and design, the project team and resources, and of course, the population impact. From these, we come to the recommendations for the application review subcommittee. So as I mentioned before, for this current round, 8 of the applications out of the 22 that went to scientific review were recommended by the grants working group. As you can see here, the total applicant requests for those 8 applications exceeds the amount of funding that's available for this program. I will also mention here that under Prop 14, we report on any application that is not recommended for funding, but which had 35% or more of the reviewers score to fund the application. And if you looked in your materials, there is one application in this round that received a minority report. It is included within the review summary for the application, which is numbered PDEV 19728, which is listed here, and it received a median score of 84. I will point out also that the CIRM team supports the majority position in this particular application to not fund 19728. Now, as I mentioned previously, we had 8 applications that were recommended by the grants working group. The CIRM and less than the 89 million available for funding for this program. And so the CIRM team recommendations are presented here to you. Essentially, to summarize, the CIRM team recommends that the application review subcommittee fund the top 4 ranked applications that are highlighted in green here, and that recommends to not fund the rank 4 through 8 applications, as you can see here. And the rationale is laid out in the memo that was presented to you, but in brief summary, the CIRM team looked at the grants working group summaries, as well as the competitive landscape for each of these projects, and also considered the CIRM portfolio for the preclinical and our preclinical and clinical portfolio in examining each one of the 8 applications that were recommended. And ultimately, the team didn't find any compelling reason to advance any of the ranked 4 through 8 applications above the top 4. So I think with that, I will go back to Chair Imbasciani.
Thank you. I was going to interrupt you anyway at this point, Haley. I just want to remind the board members, this morning before you came in, we put in front of you at each of your places a handout that compiles in one place many of the slides that show in color coding the recommendations of the grants working group and of the CIRM team among the total applications. The PDEV applications are on pages 2 and 3. And it might be handier for you to use the handout rather than the slide. Okay, I would like, thank you, Haley, but don't go away.
Yes, and can we switch to the Excel?
Yeah.
Please.
Could you switch to the slide that's got all of the applications, including those not recommended by the grants working group, that one? Yeah, you might need to toggle periodically up and down. Thank you. I would like to do this in 3 parts. I would like, first of all, to entertain a motion to remove from further consideration for funding by the ICOC all the applications, the 12 of them, only the bottom 12, that scored at the grants working group 84 and lower. To remove the 12 applications at the bottom that scored 84 or lower.
So moved.
Thank you. That's right. By the way, board members, you've been apprised of potential conflicts. If you're conflict, can't make a motion either. I don't think that inclusion. That was Mark.
Yeah.
Thank you. We have a motion
I'll second.
and we have a,
Yeah.
Marguerite Casillas.
okay. First of all, I'll open the comment section now to the board members first, followed by the public.
Mr. Chair, could I just give a little guidance?
Thank you.
At this, because this program is what we call oversubscribed. So there are currently pending at this moment more applications for funding than can be provided by the program budget. Those members who have a conflict with respect to any application in this group cannot participate in this discussion. That said, when we call the roll call, when your name is called, you can indicate your vote, yes or no, except for those applications with which you have a conflict. So for the discussion purposes, we'll have to have you sit tight.
In the room.
Mr. Chair.
Yes,
No,
Isabel?
I'm not commenting on anything except I want to comment about COI.
Oh,
conflict
like
of
a point
interest.
of order, if you will. Okay.
Excuse me. I oftentimes, when I look at the list, I oftentimes see how many times I'm booted out of the voting arena. And so I think of the public as saying, well, she has a lot of conflict. I think that it's, if you could describe to the public why, who can't vote, what conflict of interest means, because it means that I have about 1,000-mile relationship with a Stanford or a UCSF. I'm not engaged in any of these research projects, but I get it. So I don't know if you could explain, just because there's some of us who constantly are on the outside looking in.
Sure. So there's nothing really easy to explain about conflicts of interest. What I would say, I think initially, though, to the appearance issue you've raised is our own COI policies that apply to board members that the board has adopted to apply to its members preclude any member from being an awardee or receive funds under an award. So as a threshold matter, anytime a member has a conflict of interest, it's due to other financial interests that they have that may be connected to an individual, a subcontractor awardee on an application, or they have a financial relationship with the institution, such as they're a faculty member, or they have a contract to provide services to an organization. But under no circumstances is it because the board member stands to receive any sort of funds or anything like that. So it's the standard rules that apply to any public official at a city council or any other state agency.
Any questions on the motion from board members? If not, is there any member of the public, either in the room or online, that would like to address the motion? You may approach. I'm going to let Claudette, would you guide? We have somebody in the room who wants to address. I think you need to approach a microphone, ma'am.
Okay, so we're going to open the floor for public comment. If you're joining us in person, you can approach the microphone here on the right side of me. And you may state your name for the record, and please, we will be setting a timer for 3 minutes. Also, for any participants that are on the phone, if you would press *9 to raise your hand, and when you're called on, you're going to press *6 to unmute yourself. But okay, go ahead and state your name.
Alexis Lorea. Hi there, my name is Alexis. I am here today. I'm an attorney and advocate, and most importantly, I'm the mother to a little boy who was born with adrenoleukodystrophy and was identified on newborn screening. And I remember the first phone call that completely changed my lives. I was looking at a totally normal, healthy baby boy, and now was faced with a completely devastating and neurological condition that came my way that I had never heard of. Thankfully, newborn screening gave us something that previous generations of ALD families have never had, and that's time. Today, my son is healthy, but every MRI that we go to gives us something that many ALDs see without warning, and that is the onset of cerebral ALD. While current treatments halt the progression of this disease, they don't eliminate the challenges that we're faced with. We have bone marrow transplants, which, while effective, depend on finding a suitable donor match and undergoing chemotherapy. And my husband and I, unfortunately, come from different ethnic backgrounds, so finding a donor is incredibly challenging if we can find one at all. Families facing ALD should not have to consider this option when time is absolutely critical and face this challenge. And SkySona at one point created a tremendous hope for our community. However, there's concerns again about chemotherapy, risks, eligibility, side effects, access, and long-term outcomes. And recently in August, Bluebird Bio put out a memo noting that there's a black box warning and shared that 15% of their clinical trial patients have developed leukemia or some other sort of blood cancer. That returned many ALD families back to a single, safer treatment pathway, the bone marrow transplant, thus leaving us with our only option at this point in time among many of our transplant centers. There is a clear, unmet need for additional safe options for children with cerebral ALD. Rare disease families like mine spend much of our lives waiting for someone to decide whether our children are worth investing in. You all are different. CIRM is different. You have the ability to support therapies that otherwise may not receive funding that these children deserve. And respectfully, this is not simply a question of whether this research should be funded, it is when. Every month matters. Disease progression doesn't pause while families search for matches. It doesn't pause while researchers seek funding for our families. And it doesn't pause for another grant cycle application. Children diagnosed today may not be in the same clinical position 6 months from now. And families race against a disease that is not waiting. I'm part of Florida's Genetics Newborn Screening and Advisory Committee, and we had an ethicist come and talk to us.
All right, thank you for your comments. I appreciate you coming. Thank you, ma'am. Your 3 minutes are up. Thank you. Next public comment, would you please come up and you may state your name? You will have 3 minutes. Thank you.
Hi, everybody. My name is Andres Berliel. I'm the co-founder of Aspen Neuroscience and the Senior Vice President of Research. I first came to CIRM about 10 years ago with Parkinson's patients, advocating for a grant for Parkinson's disease. And we were fortunate enough to get a DISC 2 award in 2016. It took us 7 years from 2016 to get our clinical trial started, but since then we've dosed 14 Parkinson's disease patients with their own autologous iPSC-derived neurons. Really, we're the only group in the world that can say that, the most advanced autologous iPSC-derived cell therapy for Parkinson's disease. And in March of this year, we're really excited to present exciting 12-month data from our first 8 patients, showing that not only was it safe, but these patients were getting better. Something that's really exciting for something like Parkinson's disease, where you only get worse. Unfortunately, some of the patients that helped advocate for this program in 2016 died before the clinical trial started. And others screen failed in the trial because they had degenerated to the point where they were no longer eligible for cell therapy. So it's because of that experience that I have with the patients and understanding how urgent these cell therapies are to get to people that I'm coming to advocate for our grant today. Our grant received a score of 84. It's 19751. And I know that there's not funding for these grants, and there's even grants that were awarded a score of 85 that are not recommended for funding, and that the CIRM staff is not recommending our grant for funding. But it's really because of my experience with the patients and my belief in our potential curative therapy for cerebral adrenoleukodystrophy that I feel compelled to come and ask you to consider our grant anyway. I think that it fits CIRM's priorities of cell therapy for the CNS. It's an unmet rare disorder using a pluripotent-based stem cell therapy. And it's because of our connection with patients like Alexis and her family that really want us to keep moving this forward and do everything that we can. So we've been very successful raising money for Parkinson's disease, and we've been a partner with CIRM and very grateful and thankful for that. It's been much, much harder to raise money for a rare disease. I think from the investor community, it's difficult when you say, you know, we're going to maybe be able to treat 100 patients per year. But with rare diseases, I think the person on the GWG that was most in the best position to review our grant was also one of our collaborators, so he was recused. And I think that's one of the cautionary tales of working on rare disease. There's just not a lot of experts. But I think, that's why I think it's important to hear from the patient community about that met need. And Dr. Lund submitted a letter in support of our grant.
Okay, thank you very much for your public comment. Is there anybody else in the room? All right, anybody on the phone? As a reminder, this is for PDEV 19751 and below. I don't see any more public comment.
Any other final comments from board members in the room? If not, we can close discussion and proceed to a vote.
And as a reminder to members, Bonneville, Dahl, Derone, Flowers, and Miaskowski, when I call your names, please indicate that it's not with respect to those with which you have a conflict. Maria Bonneville,
Yes, except for those with which I have a conflict.
Marguerite Casillas,
Yes.
Judy Chow,
Yes.
Leandra Clark Harvey,
Yes. Yes.
thank you. Shannon Dahl,
Yes, except for those with which I have a conflict.
thank you. Anne Marie DeLiege.
Yes.
Isabel Derone,
Yes, except for those with which I have a conflict.
Mark Fisher Colbury,
Yes.
Elena Flowers,
Yes, except those with which I have a conflict.
Vito Imbassiani,
Yes.
Rich Lahara,
Yes.
Chris Miaskowski,
Yes, except for those with which I have a conflict.
Adriana Padilla,
Yes.
Marv Southard,
Yes.
Yael White,
Yes.
Kevin Zhu.
Yes.
Thank you.
Thank you very much. Could I ask the AV team, could please substitute this slide now for the one with just the top 8? Is there a problem?
Gil is controlling the slide right now.
Sorry. I would like the short, perfect. Thank you, Gil. Okay. I think the best way to facilitate the further discussion now would be to entertain a motion that, well, let me precede that. This slide shows the 8 applications recommended by the Grants Working Group. We just heard a presentation from sorry, yeah, Haley, that the CIRM team has recommended we fund the top 4 only. Why? To fund all 8 would cost $89 million. We only have 42 million. If we were to accept the top 4, we would spend about 41 of that 42 million, leaving 1.9. Okay, so I think the best motion would be to accept the recommendation of the CIRM team to fund the top 4.
So
And not-
moved. So
and
moved.
not the others. Thank you.
So moved.
We have a motion.
I'll second.
The second came from? Thank you. Okay. Scott, yes.
Yes, and so same rules apply because there is still, the pending applications exceed the program budget. So therefore, the rules that applied a moment ago apply here as well. So if you have a conflict to one, you cannot participate in the discussion of this.
Okay, we're going to start with comments from board members first and then open it to the members of the public, as we usually do. Anne Marie, yes, Julia.
A comment and a question. A comment is first, I would like to thank the 2 presenters from the public who came, and we recognize how courageous it is and probably understand some of what you're going through. We don't pretend we understood all. We have also several considerations that come in our mind, but wanted to thank you for the effort that you made today. And that applies to all these that may come later during this session today. A question for you. Many of the reasons are why you've eliminated some of the funding, and we understand you had to make a proposal for making sure that a presentation that comes in the budget. But the reason was that you didn't find the CIRM team, didn't find any compelling reasons to move it ahead. Can you explain a little bit what could be, give us an example of what a compelling reason might be, for instance?
So one might be, for example, if the project was completely unrepresented in our portfolio, for example, or I don't know if the CIRM team wants to chime in here with anything in particular, but I think it would have to be something really outside of, what we typically would consider a part of like the entirety of our scientific review in terms of if there was some significant flaw with one of the recommended applications that were ranked higher, that would impact the way that we would administer that application or anything. And I think I would also just maybe point out that in terms of, looking at the 8 applications that were recommended, the top 4, not only just purely on the score, I guess, in terms of the scientific review, I would say is that, the grants working group did have some concerns with some of those projects as well. But I think the concerns that were brought up for the top 4 were concerns that could be easily addressable with award management and milestoning and that sort of award management approach. Whereas some of the concerns that were brought up, I would say on the 4 through 8 ranked applications had a mix of those types of concerns as well as other concerns that could be addressed in a resubmission in terms of, let's say, their approach to the project or the value proposition and other aspects of the project.
Rosa?
Dr. Dulesh, if we had the money, we could fund them, right? The one that has come up is actually under this 4 that we see here, 85. So if we were to fund that one, that would have to be a very strong rationale based on the guiding principles that overpasses those other 4. So it's really about, if we had the money, we could fund, and then we could go one by one. And we based our rationale on the guiding principles, really. The transformative for clinical impact, the patient access, and the disease representation. Those were the 3 factors.
Board members? Any board members that are not? Sorry.
So having reviewed some of the 4 that passed the mark but won't be funded, they were really outstanding proposals. But if we don't have the money, we don't have the money.
Thank you, Marvin. Any board member not in the room want to make a comment? Do you see any?
There's no hands raised.
Okay, we'd love to hear from members of the public now. The same rules apply. Please identify yourself for the public record. And you have 3 minutes. Thank you.
Good morning. My name is Jiwu Wang. I am the founder and CEO of Leo Biotech. We are the lead applicant of PDAV 19729. So I also I'm the founder and scientific director and a professor at the Sintelon Institute. I founded in San Diego as well. So our project, as proposed, is to produce iPSC-derived beta cells and encapsulate them in a device developed by our partner company, Insulin, to provide treatment to T1D type 1 diabetes. And our compelling reason I'm trying to present here to be funded, to be selected in this round, not next round, is we can bring this product to patients much quicker than any other program that's being considered or being funded for treating T1D. The reason being, although our main scheme of encapsulation has not been successful in the field for many years, but the new method we developed through our collaboration with Insulin, my colleague, Dr. Grace Wei, will speak a little bit more about that after me, has been tested in humans, is ongoing and expanding trials in Canada to show great effects. And we are ready from the cell therapy part. Our technology, very powerful messenger RNA-directed iPSC generation and differentiation has been adapted, licensed by leading players in the iPSC field, like the major Japanese companies been using our technology to develop therapies. And we have shown we could produce beta cells to the purity of 96% and above, which is twice that achieved by other methods in the field. So our encapsulation is ready, is in human. Our cell production is ready and cheap. This is in reference to another program my company is doing for the upper age funded liver manufacture by printing a whole liver, our cost producing each cell type is about 10 to 20% of any other conventional methods, such as using cytokines, because we use messenger RNA. Through COVID vaccine, we already know the power of messenger RNA in expression and changing things in biology. The last point I want to make is there are some newer technologies, alternative, that's worth considering and funding.
Thank you, sir. Thank you for your comments.
Right.
All right. Do we have any other public comment in the room? All right, thank you. State your name for the record, and you have 3 minutes.
Try again? Okay. I'm Grace Way, co-founder of Enslin, based in the Bay Area. I'm speaking in support of 1972-9 PDEV. Enslin, I'm here to give an update on our activities since the time of application. Our trial NCT 06408311 concluded a few months ago. We have ongoing data coming from that, which was primary islets, which is the current use in transplant. And those results are ongoing, and we see islets coming out of those devices that have been taken out to look at. And we have just signed a contract for funding for a new trial in Canada, again with islets. And we would love to bring a trial back to California, which is home to the company. We were a spin out from UCSF, which was also funded through the work of Tejal Desai on developing these membranes. So the encapsulation process, as Drew mentioned, hasn't succeeded till we have, right? And the key differentiator is it protects and carries the cells in your body, as opposed to right now, you're either going to have to take a lot of immunosuppression drugs, or if you don't do that and your cells are trying to evade, you don't have a safety valve necessarily. Some of the things that we've learned in working in our trial in Canada, Canada is 25 times the size of California. It has about the same number of people. It has very remote geographies. Our therapy encapsulated could be packaged in a box, sent on a bush plane on flights 5 miles north, and you can have a local nerve block. You can have a local procedure done in 10 minutes. That's how long it took in our last trial to implant, and you would be done. So in terms of your presentation on the community care therapies clinics, I think this is something that could be deployed without a lot of extra specialized training or personnel, and it could obviously ship within California very easily. So I think that would be our argument to have it considered as a candidate for therapy. Thank you.
Thank you.
All right, is there anybody else in person? I don't see anybody else. I do see a hand raised on the phone. If anybody else on the phone line wants to make a comment, please press *9 to raise your hand. But number that ends in 3750, if you'd press *6 and state your name, and you'll have 3 minutes to make a comment.
Hi, my name's Paul Grand. I'm the CEO of MedTech Innovator, and I'm speaking on behalf also of PDEV 19729, as Grace just did. So just for those of you who don't know, MedTech Innovator is the largest healthcare accelerator in the world. We work with companies from early stages all the way through commercialization. We've supported 838 companies who've graduated our accelerator and have gone on to bring 500 FDA-approved and cleared products to the market, which is pretty incredible and impactful. And about 25% of those are based in California, which is fantastic. So we're huge fans of California and California innovation. I'm also a type 1 diabetic myself. So this technology is very relevant for me personally. We see about 2,500 companies applying to get into our accelerator every year. Insulin, the company behind this therapy, is one of those graduates of MedTech Innovator, and has nothing to do with the fact that I'm a type 1 diabetic. They did that before I was even diagnosed at age 49, late in life. So just very quickly, I just wanted to say that the technology that we're talking about here has gone through a huge vetting process at MedTech Innovator just to get accepted. Out of thousands of applications, we only accept about 3 to 4% of the applicants every year. And this technology would dramatically improve access for patients, both in a timely manner, meaning that they'll get this therapy much faster than some of the other technologies being developed for type 1. The treatments that require injection into the portal veins are just, not going to be nearly as compelling as an implant as this technology is, and it also doesn't need the kind of engineering of the cells, which is, something that I think is, while very exciting, this is, I believe, a better approach. And by the way, there's nothing wrong with having multiple horses in the race. So even if you're funding some of these other approaches, I think this one is worth funding for sure. And then lastly, I think from an access perspective beyond timing, because this will get to market faster than some of the other therapies. Also, there's this incredible ability to improve access, as Grace was saying, to patients in rural areas who today just do not have access to primary care at the advanced kind of levels we would have at academic medical centers in places like San Francisco and LA, where I live. So just to give you the thumbs up from my perspective, this one's incredibly exciting, worth funding and supporting. And it's difficult to raise venture capital for these kinds of.
All right, thank you for your public comment. And back to you, Chair. There's no other hands raised.
Okay, thank you, Mr. Grant. Healy, I have a question for you before we close conversation. It might be helpful, board members, if the community knew the next opportunity to apply for this program or reapply.
Yes, so we're anticipating that applications are scheduled to open end of July, early August.
Of
with
this year.
Of this year, right, just in a month, essentially.
Oh.
And that the applications will be due in October. So application open end of July and due in October. Thank you.
Right, thank you, Haley. Okay, further comment from the board? If not, the motion once again is to accept the recommendations of the team, which is to fund the top 4 and not to fund the bottom 4.
And again, those members with a conflict are members Bonneville, Dahl, DeLiege, Derone, Flowers, Miaskowski. Maria Bonneville,
Yes, except for those with which I have a conflict.
Marguerite Casillas.
Yes.
Judy Chow. Sorry. Leandra Clark Harvey.
Yes.
Shannon Dahl.
Yes, except for those with which I have a conflict.
Anne Marie DeLiege.
Yes, except for those with which I have a conflict.
Isabel Derone.
Yes, except for those with which I have a conflict.
Mark Fisher Colbury.
Yes.
Elena Flowers.
Yes, except those with which I have a conflict.
Vito Imbastiani.
Yes.
Rich Lahara.
Yes.
Chris Miaskowski.
Yes, except for those with which I have a conflict.
Adriana Padilla.
Yes.
Marv Southard.
Yes.
Carol Watson. Yael White.
Yes.
And Kevin Zhu.
Yes.
Thank you. The motion carries.
Carries, thank you. If now you would turn to the first page of that handout, we're going to move to agenda item 12, consideration of those applications submitted in response to our CLIN2 program, our clinical program. And Dr. Sembrano is going to lead this presentation. Thank you, Gil.
Okay, good morning, everyone. Thank you very much. I will try to expedite a little bit because I know we're running a little late. Can we switch the slides to the next presentation, please? Thank you. All right. Okay, so I'm going to present to you the recommendations from the Grants Working Group as it relates to the CLIN2 program. I'm going to go over what the program is about, the review process, and then conclude with the recommendations from the working group itself. So as mentioned by Haley and others, we have strategic impact goals that we are trying to achieve through these funding opportunities. For the CLIN2 program in particular, we want to propel 15 to 20 therapies that target diseases affecting Californians to late-stage trials. And we do have an updated CLIN2 in that the cycle represents, that includes support for emerging novel clinical trial designs. It incentivizes stage-appropriate market access strategy, development and pre-commercialization activities, and incorporates prioritization of innovative therapies for diseases that affect Californians. So the objective of the CLIN2 program is to support the completion of an interventional phase 1, 2, or 3 trial for an innovative stem cell-based or genetic therapy that's addressing a serious unmet need with potential for transformative benefits to patients, families, and the healthcare system. And so we designed the program with that in mind. This is just an overview of the structure itself. For the CLIN2, we support 4 cycles of this program per year. So it's on a quarterly basis. There are co-funding and maximum award requirements that differ depending on the stage of the clinical trial, as well as whether the applicant is a for-profit or nonprofit. And then for non-California organizations which are eligible to apply, there are restrictions in terms of where the funding can be utilized. Our estimate for this fiscal year is to make 9 to 16 awards because they vary in amounts that can range from under 5 million to 15 million. That is an estimate from the total allocation we had this year of 135 million. All right, so let me go over the review process. So the review process that we use for this particular cycle utilized the qualification process that uses preferences in order to make objective assessments of applications that come in and narrow the pool. This will be the last cycle that utilizes this method. But under this method, the top 7 applications advance and then go into an eligibility assessment, merit review, and ultimately the funding decision, which comes to the application review subcommittee. And in terms of the numbers that we started with 14 applications that were submitted that went through that qualification process. 7 advanced, 7 were eligible, and 3 were recommended by the grants working group. The panel composition is similar to that which Haley described for PDEV, but in this case, we also have the addition of access and affordability experts that help us assess the access plans that are provided by the applicants to better estimate their ability to advance a therapy that's ultimately going to be accessible, and that they have plans in place that will help them do that. The scale for scoring is the same as with PDEV. It's range from 1 to 100, with 85 and above representing exceptional merit and warranting funding. Below 85, those applications are not recommended for funding. And similarly, the scoring here is holistic based on all of the review criteria that we will show you here. The value proposition, meaning what is the overall value of this therapy, and whether it offers a transformative and compelling value proposition, the rationale, the plan and design, the project team and resources, and the population impact. All right, so let's get to then the recommendations from the grants working group. So we did have 7 applications. There were 3 that were recommended for funding. The not recommended shows 2 because 2 withdrew. So there's really 4 that were not recommended, but there's 2 that remain under consideration. The 3 that are recommended have a total applicant request of about 21 million. The funds available are 61, so there are adequate funds to fund all 3. In terms of minority report, we have 1 application that qualified for a minority report. I won't go through what a minority report is since we just heard that from Haley. So this is the application that qualified for the minority report. It is CLIN 2-20117. It is a phase 1/2A dose escalation study for the treatment of focal articular cartilage defects in the knee. And this received a score of 80. CIRM's position on this is that we support the majority position to not fund this application at this time. Here is a summary then of the CIRM team as well as the grants working group recommendations, which is to fund these 3 applications that scored 85 or higher, and for the total amount shown there. I'm going to go through each of these just so that we can understand a little bit more about each of the recommended ones, as well as the one that received a minority report. So we created a slide, and there's also a memo that describes some of the background that the team looked at in each of these projects to assess what the team recommendation would be. So for the first one, which is CLIN 2-19848, this is entitled the phase 1/2A first-in-human dose escalation study in subjects with progressive multiple sclerosis. This is a therapeutic that is composed of CAR T regulatory cells. And what they do is they basically reduce the population of B cells and also increase the number of regulatory T cells in the immune system to bring that aspect of the immune system back, which is not working properly in multiple sclerosis patients. So with this therapy, the CIRM team recommendation is both based on the grants working group scoring as well as the patient access considerations of the project and the novelty of this indication together with the approach that they are using. So the CIRM portfolio shows that the PDAB portfolio and active CLIN 2 portfolio do not currently contain any awards that are addressing multiple sclerosis. So this would bring a project in this area into the portfolio. The allogeneic CAR T regs are a particularly distinct modality in that portfolio. And when looking more globally at external projects outside of CIRM, as we know, there are no currently approved treatments. And this even among existing trials, late-stage trials, of which we count 11, this still represents a novel modality compared to those programs. Okay, so let me go to the next application. Did that switch? No, there, oh. There we go. All right, so the next application is a phase 1/2 study of an AAV9 gene therapy in patients with FOXG1 syndrome. And so FOXG1 syndrome is a neurodevelopmental disorder that is an ultra-rare disease. And so in this application, they proposed to address this through a gene therapy approach, which would basically provide the FOXG1 gene in patients in order to then recover that otherwise failed ability in patients. And so from CIRM's assessment, this approach was something that was described by reviewers as addressing a clear unmet need with a high potential to result in positive, meaningful clinical outcomes. The PDEV and CLIN2 portfolio do not currently contain any awards that address FOXG1 syndrome. And its assessment of the competitive landscape, there are no approved U.S. treatments that are addressing this, nor clinical trials that were noted. Okay, next application is CLIN2 19526. So this is a proposal for an AAV immune gene therapy for high-grade gliomas. And the high-grade gliomas includes glioblastoma as well as other severe brain tumors. And the CIRM team recommendation on this is aligned with the GWG scoring. The CIRM portfolio diversification through a novel therapeutic approach and differentiation compared to the external landscape are key reasons for this. So the approach, as mentioned, is agnostic to the tumor subtype. So it includes glioblastoma as well as other high-grade gliomas, where there is no established standard of care. The reviewers did describe this as a clear unmet need and one that could result in a positive, meaningful clinical outcome. The PDEV and CLIN portfolio do contain some awards here. The PDEV contains one award that addresses glioma, and the CLIN2 portfolio contains 5 awards that address glioma. This particular project has a somewhat unique and distinct therapeutic approach compared to those existing awards. So although it adds to the same indication, it does bring a new approach in terms of the therapeutic. And in terms of the external landscape, there are about 49 late-stage clinical trials that were noted, but of course, no approved treatments at this time. And even compared to those landscape programs, this does represent a novel modality in comparison. All right, so those are the 3 that are recommended by the grants working group and the team. I just do want to make a mention of the other application that received a minority report, just for your awareness. So this is CLIN2 20117. This is a phase 1/2A dose escalation study for the treatment of focal articular cartilage defects in the knee. And so this is one that proposes using allogeneic human embryonic stem cell derived chondrocytes as a cell therapy to treat articular cartilage defects. And you know, the CIRM team recommendation is based both on the GWG scoring as well as some noted concerns by the grants working group that we share. The proposal itself, as noted multiple times in the summary, seems to conflate osteoarthritis with focal cartilage lesions, meaning there is a target population that includes about 27 million for osteoarthritis, but the actual impacted population with focal articular cartilage defects is more on the order of 30 to 70,000. And so I think it's, you know, that's one key element that was noted in overstating the overall clinical impact and addressable patient population that this could have. And the grants working group also did have questions as to whether this represented an unmet need, given that there are existing treatments for focal articular cartilage defects. In terms of the portfolio, we do have one award that addresses focal cartilage defects using human embryonic derived chondrocytes. And the CLIN2 portfolio contains one gene therapy award that addresses osteoarthritis. In the external landscape, there are 3 late-stage clinical programs addressing focal articular cartilage defects or cartilage degeneration, and one approved U.S. cell gene therapy treatment addressing focal articular cartilage defects in the knee. And this one would represent a novel modality compared to those external landscape programs. All right, so that is a review of each of these projects. And so, Mr. Chairman, we can go back and discuss as.
Great. Please stay at the podium. Gil, thank you for the presentation on each of these applications. I have a question before, in order to sculpt the best opening motion, are there one or two applications that are not recommended by either?
So there are two applications under consideration that are not recommended by the grants working group or the team. And it includes the one with a minority.
Exactly. Thank you for the clarification. That's precisely what I wanted to hear. So the chair would entertain a motion first, as we did with the PDEB, to remove from further consideration from funding those two applications that were not recommended by either the team or the grants working group.
I move. Yeah.
Emery moved.
I can second.
Marguerite seconded. Good. Okay. Any discussion on this motion from members of the board? I don't see any online. No. Okay. Any member of the public who would like to address this motion?
All right, if there's any member of the public in person, would you please make a line or come up to the microphone here on my right-hand side? And for any member that is on the phone, press *9 to raise your hand. Okay, we have two people on the phone.
Okay.
The number ending in 1046. Please state your name for the record and let us know what application you're calling about, and you have 3 minutes. Phone number that ends in 1046. Would you please unmute yourself by pressing *6?
Sorry. My name is Aaron Huvera. I am here to speak on behalf of the Batten CLN 6 motion. I am an executive chef and the proud father of Charlie Pearl Huvera, who is 5 years old. Charlie is an amazing young girl who lights up every room that she walks into and is friends with everybody. She's incredibly vibrant and full of life. But we are fighting CLN 6 Batten disease. I want to speak directly to the hesitation that you might feel looking at the scientific panel's original score. I completely understand and respect the necessity of rigid scientific review. When this application was submitted months ago, the reviewers flagged legitimate concerns regarding manufacturing readiness, potency, assays, and regulatory alignment. But I'm calling today to tell you that the program you're voting on this afternoon is completely different from the snapshot reviewed months ago. Every single technical gap identified by the GWG has been bridged. The analytical testing is complete. The cell-based potency assays required by regulators are fully validated. The manufacturing path has been completely stabilized to support dozens of children. And our recent Type C meeting with the FDA provided a clear, safe, and collaborative path forward. The scientific and operational risks that triggered a Tier 2 score simply no longer exist. The vector is bottled. The expert clinical teams are standing by. We're not asking you to ignore the reviewers' original concerns. We are telling you that the work has been done, the barriers have been overcome, and the only thing standing between our children and a life-saving trial is funding. Please grant a conditional approval so your staff can verify this completed data package and let us save our kids. Thank you.
Thank you so much. Now I have the number ending in 4710. Would you please unmute yourself, state your name and the application you're calling for? Would you please press *6 to unmute yourself, number ending in 4710.
Hi, this is Kristen Gray. I am the founder of the Charlotte and Gwyneth Gray Foundation to Cure Batten Disease, and I'm calling to support the application for the Batten CLN6 gene therapy trial. Can everyone hear me?
Yes.
Okay. I just want to share briefly my story. I had two children, two girls that were born typical and met all their milestones. In 2015, when my daughter tripped over a few packages and had an arm tremor, our worlds changed. And 3 weeks later, she was diagnosed with Batten CLN6. They told us it was rare, it was incurable, and she would likely not live past the age of 6. They also said your other daughter, Gwen, has a 25% chance of having that same diagnosis. She too then was diagnosed with Batten CLN6. So rather than accept that reality, my husband and I were in a race against time to create a foundation, carry the burden of funding our own research that ultimately resulted in a year from diagnosis in a clinical trial at Nationwide Children's Hospital. In 2015, the diagnosis. In 2016, patient 1 was treated. And over the course of those few years, we treated 15 patients. We knew that the treatment was safe. We knew that the treatment was somewhat efficacious because I stood and personally witnessed a child that was more symptomatic that was dosed versus a child that was non-symptomatic, asymptomatic that was dosed. And she's sitting here with me today, still eating by mouth, still bearing weight, still going to school every day, still having verbal skills. But because that cohort wasn't separated with the data, all of the data was lumped in and proven low efficacy. That technology, as you might have known, was taken over by a biotech company, Amicus. And after review and, in my opinion, answering to Wall Street, they deemed that the small, ultra-rare patient population would not profit. And so ultimately, I was excited about this being taken on, and then it sat on a shelf for 4 years. All of that hard work, and after seeing my daughter lose her life after 13 and a half years of fighting, I didn't want that to happen again. Over the grief that I experienced through my daughter's death and loss, I was continuing to get calls with new diagnosed patients with CLN6. And so I knew that I could not let this just sit on a shelf. We have patients. The reason we are here and why we have moved so aggressively in our timeline is because the patients are here. In the middle of burying her, I am answering calls. We have 17 to 20 patients within the enrollment criteria right now, and I will not turn my back on this community. For all the children that have lost their life to this insufferable disease, I will not stop until there is a cure. Please help fund the cure. With the excess of available funds, they would be put to great use with this indication, and we would.
Thank you so very much for your public comment. Are there any other public comments? I don't see any more hands raised.
Hello?
No further comment from anyone on the board? In that case, Scott, you.
Just one quick question.
Sure, please.
Can you please clarify when when will be the next timeline for resubmission?
That's July 30th.
Hello, can you hear me?
All right, the motion is to not fund the 2 applications that were not recommended by the Grants Working Group and by the team. The following members have a conflict with at least one of these applications, so please append that sentence that I've given in the past. Those members are Derone, Fisher-Colbury, Flowers, and Miaskowski. Maria Bonneville,
Yes.
Marguerite Casillas,
Yes.
Judy Chow, Leandra Clark-Harvey,
Yes.
Shannon Dahl,
Yes.
Anne-Marie Deliege.
Yes.
Isabel Derone,
Yes, except for those with which I have a conflict of interest.
Mark Fisher-Colbury,
Yes, except for those with which I have a conflict of interest.
Elena Flowers,
Yes, except those with which I have a conflict.
Vito Imbasciani,
Yes.
Rich Lahara,
Yes.
Chris Miaskowski,
Yes, except for those with which I have a conflict.
Adriana Padilla,
Yes.
Marv Southard,
Yes.
Yael White,
Yes.
and Kevin Zhu.
Yes.
Thank you. The motion carries.
The motion carried. Okay. Now we're left with the three that were recommended, both by the GWG and by the CIRM team. As has been our tradition, that we take the clinical applications one at a time. So why don't we start in order with the one addressing multiple sclerosis? I'd like a motion to fund 19848.
So moved.
Second.
Gil went through the portfolio review and the patient impact. Are there any other questions? And any member of the public? We do have one?
Okay, I do see, there are some hands raised on the Zoom. Are
Oh.
there anybody in person? Again, this is for application 19848. The phone number ending in 8509, would you unmute yourself and state your name? You press *6 to unmute yourself, the number 8509. Okay, there's also phone number go ahead, 8509.
Hello. Hi. Good afternoon, board members. My name is Hamida Al-Ghazwi. I'm calling from Alexandria, Virginia. I'm a mother of Maryam, who is 7 years old. Maryam can still see, eat by mouth, use a gait trainer to move. And most importantly, she still enjoys spending time with us and her own.
All right, thank you, ma'am. This is only for application 19848.
It's the multiple sclerosis application.
Number 9394. If you are, oh, that hand went down. Okay, phone number 4710. If you're calling about application 19848, please state your name and.
Hi, you guys. This is Kristen Gray. We had several people trying to raise their hands, and you guys shut it down before they were able to speak. I'm on a text chain with everyone, and 3 people I could hear saying hello, and then the public comment was shut down.
Technical advice?
We are currently
There were
on-
3 people in the, I understand.
Right, thank you. We are currently on application 19848. We've already moved past the other ones. I don't see any hands raised for this application.
All right. I think this is a parliamentarian issue. I don't think we've taken a vote already on that.
So the motion is to fund application CLIN 2 19848. The following members have a conflict, so you will not be called. Isabel Derone, Elena Flowers, and Chris Miaskowski. Maria Bonneville,
Yes.
Marguerite Casillas,
Yes.
Judy Chow, Leandra Clark Harvey,
Yes.
Shannon Dahl,
Yes.
and Marie DeLiege.
Yes.
Mark Fisher Colbury. Vito Bastiani,
Yes.
Rich Lahara,
Yes.
Adriana Padilla,
Yes.
Marv Southard,
Yes.
Yael White,
Yes.
Kevin Zhu.
Yes.
Thank you. That motion carries. Thank you. The next motion should be to fund 19928. This is FOXG1 syndrome, the neurodevelopmental disease. Chair would entertain a motion to fund.
Moved.
Thank you, Margaret.
Second.
And Marvin seconded. Thank you. Any discussion on this application from board members? or any member of the public? You do not see any hands. Just take a second look. Okay. Okay, good. Thank you. No further comment. I think we can proceed.
Marguerite Casillas,
Yes.
Leandra Clark Harvey,
Yes.
Shannon Dahl,
Yes.
Anne Marie Deliege.
Yes.
Thank you. Mark Fisher Colbury,
Yes.
Vito Imbasciani,
Yes.
Rich Lahara,
Yes.
Adriana Padilla,
Yes.
Marv Southard,
Yes.
Yael White,
Yes.
and Kevin Jew.
Yes.
Great, that motion carried as well.
Good, thank you. Then let's go to the final one. I would like a motion to fund 19526. This is the immunogene therapy for high-grade glioblastomas and other members of that family.
So moved.
Second.
We have motion and Shannon seconded. Thank you. Discussion on this last application from members of the board in the room or online? You do not see any. I don't see hands. Any member of the public want to address this? Hearing none. Scott, you can proceed to the final vote.
2 seconds. 2 seconds. Great. Maria Bonneville,
Yes.
Marguerite Casillas,
Yes.
Leandra Clark Harvey,
Yes.
Shannon Dahl,
Yes.
Anne Marie Deliege.
Yes.
Mark Fisher Colbury.
Yes.
Thank you. Vito Imbasciani,
Yes.
Rich Lahara,
Yes.
Rihanna Padilla,
Yes.
Marv Southard,
Yes.
Yael White,
Yes.
and Kevin Jew.
Yes.
Great, thank you. That motion carried as well.
Thank you, Scott. That closes that agenda item. Where are we?
May I ask a question?
Absolutely.
Just as a
Dr.
point
Barrett?
of information, it appeared that there were members of the public online who felt that they were not able to make their public comment on a previous motion. I don't know if we have a mechanism for their comments to be entered into the record, even though the motion has already been voted upon. But I feel very strongly that as a public body, we should have an opportunity for these people to speak.
Thanks. Thanks, Kim. I'm very sympathetic to that. I was caught up in the moment. I didn't have any parliamentarian answer to that. But I think if we have captured those phone numbers and can contact those people, I think one solution would be to include their intended comments in the record. Would that satisfy everyone? Does that seem, I'll ask counsel, does that seem untoward or?
No, from a compliance standpoint of Pat V. Keene, we offer public comment commensurate with the motion that is made in the discussion. I'm told that there have been occasions where someone has raised their hand after the voting has already proceeded on an item. So in those circumstances, we fulfilled our obligation to make public comment available. Obviously, I can't speak to any other particular circumstances unless I know otherwise. That said, there is a public comment item at the end of the agenda that people could speak to at that point as well.
That's a very good point. If the members of the public who wanted to comment on that now passed application, there is a point at the very end of the agenda of today's meeting where you can make comment on anything that we've talked about today and anything that, in fact, that we didn't raise today for a future agenda item. So that would be the way to go then. That's a more elegant solution.
There are two board members with their hands raised, Carolyn Meltzer and then Leandra.
Let's do that in order then.
Yes, thank you so much. For those of us who are on Zoom, we could hear someone saying hello and trying to put in a comment, and I don't know that they were heard by those in the room. So I don't think it was an issue of being past comment period. They weren't heard in the room is what I'm conjecturing.
Yeah, but those of us online could hear multiple people trying to weigh in with comments.
This is Leandra. That was my comment as well. There were people we could hear, but you couldn't hear.
It seems that that's the case.
I may not be the right person to talk about technology. My understanding, at least, is we're not just listening, we're looking at a visual representation of those folks who have called in and have raised their hands. So that's why you will hear the team ask those and identify them by telephone number. Don't know how that may or may not address a particular circumstance for someone. But if they haven't raised their hand, we would not necessarily know that they wish to speak to that item. Because there are members of the public who dial in, who hold back waiting for their item. So if we don't see a raised hand, we won't ask that number to unmute itself.
Okay. Well, then let's keep with the.
I had one comment because I don't think you're seeing our raised hands. I have my hand raised, and the three individuals did have their hands raised during the public comment period.
This is Leandra. I saw that as well.
Yes, I saw that as well. It's Carolyn.
So considering it was the CIRM's, it's a technical thing. It happens. It's technology. The question is, how do we move forward to ensure that those that did have their hands raised and tried to get the attention of the board have an opportunity to share their public comment?
Yeah. I think if the chair wishes to take their public comment now, I was told that the hands raised once I had already started the roll call vote. So if that was an oversight, I'm not sure, but at least going forward, you can have their comment now.
I'm happy to
The
do
hands
that
were raised before the public roll was, I just would like to clarify, the hands were clearly raised beforehand and they had been in things. So they were not raised after the roll was called.
This is correct.
Well, we have two options. One is to do it now. Another would be to put it at the end where there is an agenda item to accommodate this. The problem is I'm not sure if the first option is licit or legal. Joyce.
My recommendation is that we should lean towards accommodating the public comments, given the fact that it sounds like this was a technical failure.
I would agree.
Agree.
You want to do it now? Let's do it now. Good. Okay. Members of the public who would like to address, let me pull this out again now. Sorry.
Okay, so I have.
Clin 29114, am I right?
It's throughout the CLIN application.
Yeah.
So I have a phone number ending in 3925. If you would unmute your phone by pressing *6, you'll have 3 minutes.
Hello, my name is Maigualida Sarceño. I'm the mother of Suaiki Sierra Sofia. We live in Fremont, California. Both of my kids are impacted with Batten disease. Sierra is 6 years old. She loves to play outside, explore nature. She still has speech, vision, and the ability to walk, but those abilities are quickly slipping away. Dwight is 4 years old and the most affectionate little boy. He runs and still plays just like any other kid his age, but very soon he will start to experience the first symptoms of this terrible disease, just like his older sister. Right now, he is the youngest known with Batten disease, CLM6, and stands to benefit greatly from treatment, and he is minimally impacted by the disease at this point of the time. I'm here today to give you the context of time that spreadsheet cannot show, the GWG report suggests that our needs can be addressed in a future funding cycle from administrative standpoint. A delay until 2026 sounds neutral. For my children, it is a progressive, irreversible loss of life. CON6 pattern disease is an uniformly fatal neurodegenerative disorder. It does not pause to fiscal quarter of grand cycles. Children with this mutation rapidly lose their language, their vision, their ability to swallow, and eventually their life. A 6-month delay is not a pause in development. It is a permanent loss of cognitive and motor skills that can never be recovered. My child does not have until 2026. If you defer this program, you are not delaying a trial. You are declining how much of our child's brain are left to save. We urge you to look at this program with the life or death urgency it demands today. My daughter Ciara is 6. She still can qualify for this gene therapy. She's losing her abilities very quick, and this is her only chance. So please take us into consideration. Thank you for your time.
Thank you so very much for your public comment. The phone number ending in 9394, if you'd press *6 and state your name. You'll have 3 minutes.
Hello. Good afternoon. Can you hear me?
Yes, we can.
Can you hear me? Okay, great. Good afternoon, board members. My name is Sierra, and I'm calling from Haines City, Florida, and I'm the proud mother of Azalea, who is 4 years old. Azalea is full of laughter, joy, and smiles. She is the most determined, on-the-go child I know. She is defying all odds, but we are fighting CLN6 patent disease. I want to directly address the reviewer's concern regarding our trial eligibility and recruitment feasibility. The GWG noted a contradiction in targeting independently ambulatory patients while setting an eligibility age of 4 to 16 months, questioning if these patients actually existed. I am calling you to tell you we exist. My child is within this exact age bracket, and she is ambulatory right now. But because of how fast this disease strips away motor skills, that therapeutic window is heartbreakingly narrow. 6 months down the line, it will be too late. The reviewers worried we wouldn't find 10 patients. The reality is that our children are retaining the exact skills this investigational therapy is designed to preserve today. If you force us to wait, Azalea and the rest of these children will cross the line, become non-ambulatory, and lose their eligibility forever. We are not a theoretical population. We are identified, we are ready, we are here, and our children are running out of time. We do not have 6 months to wait. We may not even have a month. Please do not shut down the window on them. Thank you.
Thank you so much for your public comment. The number ending in 8207, if you'd press *6 to unmute, you have 3 minutes.
Yes, my name is Jill Weimer. I'm the Senior Director of Therapeutic Development at Stanford Research and the former Chief Science Officer at Amicus Therapeutics. I have been involved with the Gray Foundation in the development of this program since its inception in 2015, and I've spent the last 25 years working in the Batten disease research space. I have worked tirelessly alongside a number of families, and I have seen the true value of this program. And as Kristen mentioned, the initial program enrolled 13 children. Those children, many of them were late in their onset of the disease progression when they were treated, but there were a number of kids, her daughter included, that were very early in their disease presentation and have shown very obvious improvement following treatment. The review panel raised a number of concerns about modifications to our manufacturing and CMC processes, and I wanted to just speak to those in this public comment forum. Many of the choices that we've made during this process have really been built upon learnings over the last decade of this program to really improve the manufacturing processes and to modernize them as the analytical assays and the manufacturing work streams for gene therapy manufacturing and AAV manufacturing have improved over that time. So we're not planning to make major changes to the actual TPP or to the actual final product, but really just modernizing processes from pivoting from adherent triple transfection to suspension. We have been able to go from manufacturing processes at nationwide.
All right, thank you so much for your public comment. The number ending in 8509, if you would press *6 to unmute yourself, you'll have 3 minutes.
Good afternoon. Can you hear me?
Yes, we can hear you. Thank you.
My name is Hamida Al-Ghazwi. I'm calling from Alexandria, Virginia. I have a 7-year-old daughter. Her name is Miriam, who still can see, eat by mouth, still can walk assisted, and she still can enjoy her time with us. But she's still fighting CLN6 pattern disease, and that's slowly taking a toll on her little body. Since she got diagnosed, our family experienced like financial since like she needed to be watched 24 hours. For years, family like ours had carried the burden of funding the research, and that's what the foundation has done for so many years. Family like mine had held fundraising after fundraising, asking community for help. Funding this clinical trial will be a great help for family like mine. We are willing to travel from where we are to California to treat our daughter. Thank you.
Thank you very much for your public comment. Is there anybody else in the room or on the phone that would like to make a public comment? On the phone, please press *9 to raise your hand. I don't see any hands raised.
Are any of our members who are participating by phone able to hear or see anything that we here in the room do not?
This is Leandra Clark Harvey. I do not see any hands raised, nor do I hear anyone vocalizing the need to speak at this time.
Great, thanks, Leandra.
Thank you, Leandra, for that help. Okay, I'm going to ask, where are we on our agenda as far as a break?
We are well past that. Yeah. So if we could maybe take 5 minutes, which I know means 10, then we can get back. Our lunch is somewhat constrained at the back end. So as soon as we can get back and get on with the.
Here we go. We're on recess for 5 minutes.
Yeah, 11:40, hopefully.
Yeah. Oh, yeah. That one's far. Yeah. Just
warning, everybody. About 2 more minutes left in the break. 2-minute warning. If everybody could take their seats.
Okay. I hear noise.
Yeah, we would like to start momentarily, so if everybody could start taking their seats, much appreciated.
Welcome back, everyone, from our little recess. We're now at agenda item number 14. Our Director of Communications is at the podium. Amy Adams is going to introduce us to our recurrent, felicitously recurrent a section of the agenda, Closer to Cures. Amy?
Thank you. I'm pleased to introduce our Closer to Cures speakers for this meeting. As always, we're going to be hearing from 2 of our awardees, and these 2 will be talking about outcomes from clinical trials that are being funded by CIRM. I'm going to introduce both speakers now so I don't interrupt the flow later. First, we're going to hear from Thomas Chalberg, and he is the founder and CEO of Genesance. He'll present results of a phase 1b clinical trial testing a genetic therapy for knee osteoarthritis. And you can start forming a line right here to be in the clinical trial. This work is supported by a CIRM CLIN2 award. And after that, we're going to hear from Stephen Deeks. He's a professor of medicine in residence at UCSF and a faculty member in the Division of HIV, Infectious Disease, and Global Medicine at Zuckerberg San Francisco General Hospital. That was the hardest part of my day. He will discuss results of a clinical trial testing a new approach for treating HIV infection. You may have read about his results, which he presented at ASGCT, and they were highlighted in a story in the New York Times, among many other publications. And his work is also supported by a CIRM CLIN2 award. And without further ado, Thomas Chalberg.
Thank you so much. My name is Tom Chalberg. I'm a co-founder at Genesance and the CEO, and it's a pleasure to be here as a scientist and a lifelong California resident. I'm extremely proud of the work you all are doing at CIRM. So thank you for all of your support. I want to talk about knee osteoarthritis, which is a highly prevalent, disabling, degenerative disease, perhaps the most prevalent degenerative disease that affects millions of people around the world and millions of people right here in California. Knee osteoarthritis, patients present with painful knees, painful and swollen joints, and often receive oral pain medications like NSAIDs, Tylenol, or opioids initially, and then eventually graduate to needing something more, and that's steroids, which do work, but only for a period of a few weeks, and unfortunately can't be redosed, and other therapies like hyaluronic acid and platelet-rich plasma that really have low evidence or no evidence of efficacy. And so patients are left in this zone of no effective treatments with compromised pain and function, nothing to do to slow down or stop the disease, and finally succumb to joint failure and a really horrible surgery, the total knee replacement, which many patients are either ineligible for or cannot make a successful recovery and still have chronic pain. That leaves us with a cause of osteoarthritis of the knee being significantly morbidity and mortality, the #1 cause of disability, and a top cause of chronic pain, a top cause of opioid use and abuse. And so I want to next tell you about our proposed solution, which is called GNSC001. It's a transformative disease-modifying therapy in knee osteoarthritis. And I'll start by describing the role of interleukin-1, which is a major bad actor in the disease. It plays a central role in inflammation, in pain, and disease progression. So IL-1 itself is a nociceptor, meaning that it detects pain signaling in the nervous system. It causes rampant inflammation. In fact, is the most potent inflammatory molecule in human biology, and it directly destroys cartilage. The original name of IL-1 was catabolin because of its ability to catabolize through joint tissue. And so all of the hallmarks of OA, a painful swollen joint that leads to cartilage degeneration and joint failure, find their roots here in the activities of interleukin-1. There have been a number of genetic studies as well that have linked molecules in the interleukin-1 pathway directly to osteoarthritis risk as well as severity. And so that being said, previous attempts to target IL-1 have been mixed because drug delivery to joints has been very, very technically challenging. When we inject a drug directly into the joint, like a small molecule or a protein, it quickly leaves. And that's in a matter of just a few hours. And so the ability to treat a chronic disease with an injection to the joint has been impossible. At the same time, systemic delivery essentially results in large systemic side effects with very, very little of the drug actually reaching the joint. And so what this really has needed is new technology to approach the way that we even get molecules into the joint to treat the disease. So the GNSC001 is our solution, which is really a localized biofactory that produces a therapeutic protein right there directly in the joint where it's needed. And so patients are treated in the office. It's a simple injection into the joint, the same way that steroids or other therapies are given today. But once it's inside the joint, this gene therapy vector encodes something called the IL-1 receptor antagonist. And this directly interferes with that interleukin-1 signaling that we just talked about in the joint. It really works by through two cell types. The vector goes inside of synoviocytes, which line the inside of the joint, as well as chondrocytes, which are that important cell type that's right there in the cartilage. And once it's inside those cells, because these cells don't divide, they really just stay, and it continues to churn out fresh therapeutic IL-1RA or fresh therapeutic protein over years following a single simple in-office treatment. And so to talk a bit about our program and the clinical development program and how the CLIN2 award has played directly into our ability to advance, I'd like to share a couple of really important insights here. So we began our clinical development journey with a small phase 1 study. It was an investigator-sponsored single ascending dose first in human study at Mayo Clinic with only 9 subjects, and this was really just to look at safety. That's when we applied for the CLIN2 award funding and really enabled us to move forward to do Donatello, which is a phase 1b study with 67 patients. This was the first multi-center, double-blind, placebo-controlled study where we were able to look at safety, dose ranging, with and without immune conditioning. And the conclusions, as I'll show you on the next few slides, is that the therapy was well tolerated and resulted in robust IL-1RA expression. So that therapeutic protein was there and lasted the entire duration of the follow-up period. From here, what we hope to do is move into the late stage development. So this would be the first pivotal trial. We're calling it a phase 2/3 study called Raphael. Our goal with that would be to enroll 240 participants. And that's really aimed at showing our patients getting better. Is their pain improving? Is their function improving? And so we do think there's a potential opportunity for additional future CIRM support here. We'd like to initiate that study in 2027. So Donatello, which was the main subject of the previous CLIN2 award, as I said, was a phase 1b study design. And really the objectives were to determine the safety and the correct dose to move forward for further development, and also to investigate the potential role for immune conditioning. Because this vector is a foreign protein, there are known immune responses. And so is there a role for providing that immune conditioning at the time of treatment? And so patients were randomized into one of 5 groups. They were either placebo control or a lower dose, 1 times 10 to the 12th vector genomes with and without oral immune conditioning, or the higher dose, 1 times 10 to the 13 vector genomes with or without that immune conditioning. And then we actually added a 6th group. So we expanded the scope of this work beyond what we had described in the original grant to enable us to treat, even more patients with a modified and sort of less invasive immune conditioning regimen, to ask whether that gave us the same results as that as the immune conditioning in the first 5 groups. The primary endpoint was safety at month 12, and we also took small samples of the fluid from the joints at every visit from patients so that we could biologically assay that to see whether we were producing the therapeutic protein. Very importantly, we also used the CLIN2 grant to support improvements in manufacturing. So whereas we started with a fairly, non-scalable early stage manufacturing process, we've been able to adapt this to a large-scale commercially viable manufacturing process. And what that means is that the cost of goods to manufacture this will be commercially viable, and it will really improve access once this therapy gets to market at prices that are, reasonable and covered by, most people's insurance. And furthermore, we reached agreement with FDA on this new manufacturing process for the GMP drug to be able to be used in the next clinical trial. So now I'd like to go on to talk a bit about the results that we saw in Donatello, where really the study met the study objectives and provided encouraging early results. We enrolled and treated a total of 67 participants at 9 clinical centers, both here in California and across the United States. And you can see the consort diagram here. These were, equally spread among the groups. And importantly, we really strove to enroll participants that were reflective of the diversity of the California OA population, and we achieved that goal both in terms of age, gender, and race and ethnicity. In terms of the primary endpoint, there was a very favorable safety and tolerability profile. We didn't see any toxicity related to the gene therapy. We didn't see any signs of systemic IL-1 suppression. What we do see is the same thing you see whenever you inject a biological agent into the joint, which is mild to moderate swelling of the joint in about 10% of patients. And so in other words, this was very, very well tolerated and as expected at all dose levels, and in accordance with what we wanted to see. Secondarily, we looked at this important measure of how much protein we were producing in the knee joints of the patients that got the therapy. And what you can see in the placebo group is that basically patients started with very low levels. This is a naturally occurring protein that you can measure at tiny levels in the joints, and it basically stayed throughout the 12 months at that same low levels. But in all of the groups that received GNSC 001, we saw an immediate increase in the levels. And what that dotted line represents is the target levels that we believe are therapeutic given the IL-1 levels in order to block this IL-1 signaling that causes disease advancement. And what we saw in a majority of patients in the majority of the groups is that we did reach that therapeutic levels, and those levels were maintained all the way out to 12 months following a single in-office injection. So very encouraging that we were able to see this on-target pharmacodynamic effect. And then finally, we did measure, as an exploratory endpoint, patient-reported pain. So we asked patients, how is that knee pain doing on a scale from 0 to 10? And what we saw was a really encouraging trend. And although this study was not designed or powered specifically to measure pain outcomes in the same way that the next trial will be, we did see a number of encouraging responses observed. And you can see in the placebo arm that there was, a slight improvement in the placebo group. And in the 2 low-dose cohorts, we also saw a slight improvement, maybe slightly larger than the placebo. Whereas in the 3 groups that received the high dose of the therapy, we saw more meaningful improvements and a continued improvement in trend between month 6 and month 12. And so when we looked at a pre-specified pooled analysis, we really saw a trend favoring the high dose at 6 months, and that became statistically significant by month 12. So again, although this wasn't sort of a pivotal study that was designed to measure pain, it's very consistent with what we hope to see in terms of these encouraging responses. I next want to play a short video of one of our investigators, Dr. Susan Bukata, who's the chair of orthopedics at UCSD, which is also, this trial was conducted in collaboration with the CIRM Alpha Center at UCSD, who helped support it. And so she's going to describe her experience with the therapy and the 2 patients that she treated.
The first patient was actually really interesting, incredibly active, big traveler. It's San Diego, right? Tons of hiking, gardening, working in the yard, things that she loves. She's recently retired, and her knee was a little unstable, very swollen, starting to really interfere with her ability to do those things. She wanted to have something to help calm her knee down, so that she could do all of these things that she aspired to do. So, I think what was most remarkable within the first 6 months, dramatic improvements in function, right? Motion of the knee, the ability to get back into more activities, getting on a bike. I actually had to work carefully around her trip to Antarctica for the 6-month visit. Because she felt comfortable enough to get on a boat where she had to deal with balance. Also, if you notice with the knees, in the first couple of months, the swelling starts to go down significantly. I got to tell you, by the 2-year mark, that knee is functioning incredibly well. She's running all over the world. She's hiking, no intention of ever coming back to have something surgically done. It really brought her back to the lifestyle that she wanted to have, to be so active, to have her affected knee match the function of the rest of her body. And that's really what she wanted. So our second patient, she would classify herself an older mom. So even though she was pushing her 50s, she's a 5-year-old and a 7-year-old, and really wanted to be running around and doing things with her kids, but had bilateral knee swelling and pain and functional difficulties. She couldn't get off the floor if she was down on the floor with her kids. She couldn't go out for these long walking activities with them. We obviously had to pick one of the knees, pick the more effective knee, and gosh, the change was remarkable over the course of the 2 years. Even within the first 6 months, her function was much improved. The swelling was starting to go down. Even though she was still having some pain, she could do more things with her kids. Go to Disneyland and make it through the whole day. Be able to keep up with them with activities. And then by the time the 2nd year follow-up came around, I could not believe my eyes. The knee looked normal. The swelling was gone. The fluid we got out of it was totally normal. Her motion was back to almost normal. And in fact, her main comment to us was, great, now how can I enroll to get my other knee treated? The dramatic responses we saw in the swelling, in the effusions, that is dramatic, right? And we know that even the structural changes that happen in arthritis, in cartilage, really happen when people are having these inflammatory flares. So by cutting back on these inflammatory flares, certainly the patients that I had could really move forward with their lives in a lot of ways. And to be able to offer this to patients, particularly younger patients, highly active patients, that's really what they're looking for. And this is an important tool that can help us to control that inflammation potentially and prevent those progressive moments.
So it's really, moving to hear these personal stories. And I think, hearing from an investigator who kind of lived day in, day out, or month in, month out with these patients for 2 years and saw the improvement, I think speaks volumes about, what this really means to patients. And so what we're doing at Genesance now looking forward is thinking about how do we bring this to patients in need. And I want to just quickly describe sort of where we are in that journey and creating a path to registration in terms of next steps. So while we have been, really operationalizing, this CLIN2 award as Donatello with the first patient in and the enrollment and the follow-up period. We've been also working very closely with FDA. We received fast track designation in 2024. We conducted a very important type C meeting on manufacturing, as well as a type B meeting on clinical. And then we received most recently RMAT designation within the last year. Since then, we've submitted and received clearance for our phase 2/3. That's a pivotal protocol that now FDA has evaluated and we reached full alignment on. And so really all of these program milestones show continued momentum, both in clinical and regulatory, and give us a clear path forward to registration. This phase 2/3 Raphael study is really focused on clinical and structural outcomes. So this will take patients with painful knee OA. They'll be randomized to receive either the therapeutic or placebo. And the primary endpoint is going to be that pain and function, that, are patients improving in terms of pain and function versus placebo all the way out to a year. And then we'll continue to follow them both in terms of symptoms and in structure, MRI readouts, out to 24 months, or a total of 2 years. And I want to leave you with, one intriguing just piece of, scientific information that we think shows the power of what this therapy can do. So what we've seen recently is anecdotal evidence from very, very large clinical studies with IL-1 inhibitors that suggest that they can actually delay and avoid the need for joint failure and the need for joint replacement. And so on the left-hand side, this was a large study in cardiovascular patients, but more than 1,000 patients entered this study with knee osteoarthritis. And the ones who got an IL-1 inhibitor, their probability of advancing to a joint replacement was cut in half, from about 12% to 6% over 4 years. And that's extremely intriguing, but then even more so was replicated in a subsequent study, also looking at another protein in this same IL-1 pathway, showing that there was a 30% reduction in the rate of knee replacements. And so this is really what we're aiming to do, is not just to alleviate symptoms, but actually slow the course of the disease. And thinking about what that could mean to patients right here in California, we're seeing about 100,000 knee replacements annually in California that cost more than $5 billion of economic burden. A 50% reduction would be a $2.5 billion annual savings just in California alone from the reduction in joint replacements. And I think that really illustrates the power of what blocking interleukin-1 and the power of what Genasan 001 can offer to patients, both in terms of, symptomatic health active benefits, but also in terms of healthcare costs for all of us. So I'd like to leave it there and just say that Genasan 001 we feel is a very promising treatment for knee OA. There are no FDA-approved therapies beyond just pain management, simple pain management. Genasan 001 has disease-modifying potential to alleviate suffering and save billions in California healthcare costs. So I'd like to give a special thank you to all the Donatello participants and investigators, to our study team, and of course to CIRM and all of our investors as well. Thank you. I'm waiting for questions. Yes, thank you.
May I ask a question? Okay, thank you. You talked about you had a broad swath of California residents, both racial and ethnic. Did you break those down to see if the impacts were similar? And then did you compare them against each other to see whether or not there might be other inhibiting issues where some would do better than others?
We did. So we looked at a number. So first of all, we looked at the demographics that came into the study versus sort of California demographics with knee OA, and they were largely very reflective of the California, demographic with knee OA. And I think that really showed that our outreach efforts to sort of include a diverse patient population were, successful. Also in terms of who stayed in the study, that retention is really important. And that was also reflective, if not a little better than we had, more proportional for traditionally underserved populations. In terms of outcomes, there were no differences across groups. Again, it wasn't really powered to look at many subgroups because the numbers get pretty small as you look at subgroups of subgroups, but we did not notice any differences. And that, we think that's very consistent with OA biology, that this therapy should be, broadly applicable to, all types of patients that are suffering from OA.
So one more question. So you say it will save healthcare a lot of money, but how much is it going to cost the patient?
So we don't know yet is the simplest answer. I think our efforts to improve the manufacturing, I think when people think of gene therapy, and I hear this all the time from the community, they think of $1 million, treatments or $4 million treatments. And this is certainly what we've seen in sort of rare diseases. Our goal is to make this broadly accessible and available for the cost of less than a joint replacement today, right? When we look at the manufacturing and where we came from, that was impossible because the cost of the therapy just to make was quite expensive. And so the really value of being able to use the award to advance that manufacturing technology to lower the cost now gives us that possibility. I think in terms of what will the price of the therapy be, we don't know that until, of course, we get the results from the phase 3 and we have discussions with physicians and payers and so on. So while we've not done a lot of that work yet, we do really aim to make this broadly available to any patient that has that's suffering from the OA.
I think I had my hand up for a while. We'll go ahead and ask the question. Susan Bacata presented two people who had, swollen knees. What is the possible target of stage of disease for this injection? Because you mentioned about knee replacements, and knee replacements are usually the last step. So are you talking about someone with a little slight injury to someone who reaches a certain stage of osteoarthritis? That's question number one. I'll let you answer that first before I ask my second.
Sure. So very briefly, once a patient is a candidate for joint replacement, their cartilage is essentially absent. There's nothing, like a lot of degenerative diseases, there's unfortunately not a lot left to save. And so we're looking at patients earlier in the disease process where they clearly have symptomatic disease and they're clearly progressing, but there's still cartilage there left to save. So these are patients that are probably a few years away from being candidates to knee replacement and really slowing down and stopping the diseases in its tracks there.
So still having some reserve. My second question, this is against IL-1 beta. You said you collected synovial fluid to follow the pathway for the inhibition of IL-1 beta. Did you look at other pro-inflammatory cytokines? Is it solely just IL-1 beta? Because there's several pro-inflammatory cytokines that might be driving the disease, but this is obviously one of the key ones that might.
No, that's a great point. So interleukin-1 receptor antagonist directly blocks both IL-1 alpha and IL-1 beta. It's thought that IL-1 beta is the more important, but it does block both. In addition, IL-1 is very far upstream. So if you block IL-1 beta signaling, there's IL-1, IL-6, IL-10, and other downstream cytokines that also get downregulated as part of that inflammatory cascade. So while we know that IL-1 may not be the only inflammatory cytokine that's at play in osteoarthritis, number one, blocking it does block other inflammatory cytokines as well. And number two, it probably is the most important player, so much so that if you just block IL-1 and its downstream effects, you're going to have a major, major impact on patients, both in terms of symptoms and in terms of disease progression.
Kim, followed by Joyce.
Yeah, thank you very much for this exciting presentation. And as somebody who has painful knees myself, I'm personally very excited about this, especially with a family history of OA. But I wondered what your plans are to extend this beyond knee OA. My husband had both his hips replaced in the last couple of years, and it would have been nice if he'd had some other options.
Yeah, that's a great question. So biologically, interleukin-1 is implicated in many other types of osteoarthritis: hip OA, spinal OA, foot and ankle, hand OA. And so there's certainly an opportunity to expand. I think the reason we're starting in knee OA is, number one, that it's the most prevalent and also, the most troubling in some ways. Knee pain is so debilitating and affects locomotion and cardiovascular health and many other aspects of life. We also know the most about it by far in terms of clinical science, those endpoints that we can really measure. And so our plan is to, advance this in knee OA until we have, a very clear evidence that in a late stage study, this improves symptoms, and then expand to other joints, including hip, foot and ankle, spinal, et cetera.
Thank you for the presentation. It's really exciting given the prevalence of osteoarthritis. I guess my question has to do with something you said earlier, which is your vision is to make sure that this is easily, and I'm sorry, I can't look at you because now I have to be speaking into the mic. I can't do both. Whether you talked about the vision of making sure this is broadly accessible, and I noticed that the physician that you spotlighted there was an orthopedic surgeon. Is there any reason why we could not train primary care physicians, family medicine docs to administer this injection, given the fact that there are many parts of this country where access to an orthopedic surgery may actually be more limited than to access to primary care physicians?
Yeah, that's a great question. So I think osteoarthritis patients are seen sort of by a number of specialties, by rheumatologists, by orthopedic surgeons, by sports medicine doctors, kind of non-operating sports medicine doctors, as well as even pain specialists in some cases.
As well as primary care physicians. I saw many of them when I was practicing in
Yes.
Boston.
So in terms of the, procedure itself, it's an intra-articular injection. And I think any physician that can give an intra-articular injection and does so today with drugs like steroids or viscosupplementation would be easily able to administer, GNSC 001. It's a simple procedure. In terms of, whether a particular person has the expertise to do that, I can't necessarily speak to, but we do think that, by making it an in-office procedure, we've made it, as broadly available, as simple to administer, as possible.
So in the interest of full disclosure, I'm somebody who studies pain, and I've had 2 knee replacements.
Expert.
Yeah. So I'm curious about your outcomes for the next clinical trial. And I couldn't tell from your graphic whether or not the decreases in pain intensity were clinically meaningful. So I'm curious about that. And I'm wondering, have you considered using the improvement in function as the primary outcome rather than pain? Because pain is a challenging one to measure. And there are some people that don't have pain with OA, but they have terrible decrements in function. So I'm curious about that.
Yeah, that's a great question. So generally, our pain measurement, the one that I showed, was NRS pain on a 0 to 10 scale. And generally, the literature says that a difference between arms of 1 unit on a 10-point scale would be considered the minimum clinical import difference. And you can argue, is it 0.8 or 0.5 or maybe a little above 1? When we discussed with FDA, they agreed that 1 is clinically significant. The difference that we saw in this early study was about 2.5 on a 10-point scale between placebo and the high dose. And so we would consider that very significant. Thank you. And we're hoping to replicate that in this larger study, right? Of course, that's really, designed to measure pain. In terms of pain versus function, so what FDA has said is that, pain is the primary complaint, and we want to see patients improve in pain. But actually, they have asked us to do co-primary endpoints of pain and function. And so for us, that will be NRS pain, and it will be function on the WOMAC scale. And those measure, really the activities of daily living for patients. So we feel both are clinically very appropriate and clinically meaningful.
Good. Thank you. Shannon?
I'm scientifically curious about the timeline to regeneration and then the durability of that. How are you thinking about, how long the IL-1RA is expressed and how long that elevation takes to facilitate regeneration, and then how durable that regeneration will be over time with or without ongoing IL-1RA expression?
Sure. So, okay. So in terms of durability, we're transducing really what are non-dividing cell types. The synoviocytes divide only very slowly over years, and the chondrocytes really are completely postmitotic, do not divide. When we look at, our experience in preclinical studies, we've seen out to 18 months. We're now getting back the 2-year data from our clinical study in Donatello that we hope to publish later this year. When we look at other indications where AAV has been used in non-dividing cells, like in the nervous system, in liver, in the retina. There's been expression out to 7, 10 years following a single injection. And so that's, certainly what we would expect to see. There's no scientific reason why in another non-dividing cell type we wouldn't see the same thing. The second part of your question was the timeline about degeneration and joint failure and joint replacement, I think. So, what we see, depending on the particular population you're treating in osteoarthritis, is that over a, 3-year period, about up to 20% of patients will progress to joint replacement. And so while it's not the same like short-term endpoint that pain is, where you kind of see an immediate impact, it does take longer to develop. But these are timelines, 2 to 3 years that clinical developers can kind of fathom to follow patients and say, oh my gosh, look, we see a difference in joint replacements at 2 years, a difference at 3 years, it gets bigger at 4 years. So we do plan to follow out these patients for long term and hope to see, that difference continue to grow and that benefit continue to accrue to patients.
Thank you very much. JT, and then we're going to have a second presentation before the lunch break.
Thank you.
And also just would like to remind the members that we've invited both teams to join you at lunch today. So this won't be your last opportunity to interrogate further.
Thanks very much, Tom. Super interesting. Question, NAV 1.7 has been implicated in OA. Does your treatment have anything to do with that?
Yeah, so NAV 1.7 is a really interesting molecule. Obviously has to do with pain, sensing. And there's, in fact, there's some, efforts by companies, large and small to kind of interrogate that pathway, including in OA. I would say that that's mainly driven at the pain response pathway, and ours is separate. It's not looking at the, NAV or the pain sensitization itself. We're really looking at the inflammatory aspects and the cartilage degeneration aspects, which are related to pain for sure, but sort of form a separate pathway.
Thank you very, very much.
Thanks so much.
Amy, are you?
She's not introduced.
Okay.
Hello, everybody. Soon my slides will show up. I'm Stephen Deeks. I'm from the University of California, San Francisco. And when the slides are coming up, I just want to say up front, we really appreciated the support we got from CIRM. The clinical trial that we're about to talk about was very complicated, first in human, very intensive, and more or less got started during what I call deep COVID. Not an ideal time to start a clinical trial. Like this. The University of California, San Francisco eventually said they could not support it because of all the COVID stuff. And we ended up transferring it to UC Davis. And, it took time to get this up and going. And I just want to apologize to my the program team and everyone else about those delays, but I think it paid off. All right. So, HIV. We have great drugs for HIV. If you take an antiretroviral drug once a day, the virus is suppressed, goes below the level of detection, we can't find it, people live a normal lifespan. And these drugs are getting safer and safer. And now we have injectables every 2 months. So the question is, why do we need a cure for HIV? Because the fundamental problem with treatment is that it requires a person to be engaged in healthcare, to stay engaged for their entire life. If you're born with HIV, that means you have to have access to healthcare, for decades and decades and decades. And that just is not possible for many people. What are the challenges? On an individual level, some people just have difficult with adherence issues. They confront mental health issues, substance abuse, domestic violence, poverty, stigma, all sorts of issues prevent people from accessing care continuously. And even those who go on long-term treatment, again, most do well, but not everyone. We're concerned about obesity, concerned about polypharmacy, long-term drug effects, results in some people not being able to stay on therapy. And some people develop drug resistance, although that's less common. But this is not the big issue. The big issue is more at the public health level. all right? Treatment is expensive, and it requires someone to have access to support. People need to have healthcare insurance, has to be sustained, has to cover the drugs. Not a problem in San Francisco, but trust me, around the United States, particularly areas of the Deep South, this is a big, big problem. No one's actually covering the treatment anymore. And then, of course, we have the global stuff, where I think things get very challenging. Every few days now, we're reading in the newspapers about massive cutbacks in terms of the capacity, the amount of programs that are going on to support treatment. And so globally, we are witnessing now thousands and thousands, maybe many more people who've been on treatment, going off treatment, virus rebounding and progressing. So there is a great unmet need with treatment, and that great unmet need is the capacity to find someone with HIV, give them a single injection, and let them go on with their life, okay? A so-called one-shot cure, all right, which we thought was aspirational. But as I'm going to show here, I don't think it's no longer aspirational. I think it's feasible. And I think that this clinical trial is one of the first clinical trials to provide some excitement in this area. It's a small study, not definitive, but provides a rationale. Now, how does this work? When you look at these curves, right, ART, A-R-T, that's antiretroviral drugs. You see where it stops. The blue line is the virus load, how much virus is in the blood. 2 weeks after stopping therapy, 90-some% of people, the virus just explodes. Goes from nothing up to 1 million copies, destroys the immune system, people get sick. The goal of therapy, the goal of a cure here, is to encourage the immune system to block that from happening, so that rather than having that massive explosion of the virus, the virus goes up a little bit, but the immune system controls it. And we think this is possible because we see this naturally. 1 in 100 people who acquire HIV control the virus on their own, and they do so because they have CD8 positive T cells that target very specific areas of the virus and have certain phenotypic characteristics that we think we can genetically engineer. Now, Boro Dropulik, who's in the room back here, and his team at Caring Cross, several years ago, using their experience with CAR T cells in cancer, developed a potential one-shot cure, CAR T cell approach. I won't go into any detail here, but essentially, it's basically, using CARs that identify on the HIV envelope protein that parts of the virus that binds to the cell and causes the virus to infect the cell. And so basically these CARs, one on the CD4 domain and one on the CCR5 binding domain, these CARs basically bind to attack these targets on the HIV and kill the virus, or more specifically, kill the cell that's expressing the virus. So typical CAR T cell approach that we see in cancer. And we know that these CARs, that the product, which is called LV-GP120 dual CAR, the dual CAR product in mice does things that we want it to do. So we conducted, Mehrdad Abedi, again, the study was done at UC Davis, not UCSF. Mehrdad Abedi, who's well known to the CIRM family, has had a lot of support in the past, CAR T-cell expert, conducted a 3+3 typical dose escalation safety study. First cohort, no conditioning, low dose of cells. Did not expect to see anything. If it was safe, you go to the second cohort. Here we use low doses of chemotherapy designed to just reduce the lymphocyte count. This is not the kind of chemotherapy doses used in cancer. It's low doses, provide space for the CAR T cells when they get infused. So we give people the conditioning, low dose of cells. See what happens. If it's safe, go to the last cohort, which again is the low dose conditioning and high dose cells. Okay? We ended up enrolling 9 participants, but 2 who were in cohort 1 were allowed to actually re-enroll later on in cohort 2 or 3. At the end of the day, we ended up with 11 separate infusions. It's a little bit complicated, not really important for the story I'm about to tell. Basically, 9 people got the infusions and were interrupted therapy. Now, Here's how this works. I know very few people here deal with HIV, so I just want to show what happens. On the left, you've got a person on ART, antiretroviral drugs. The viral loads on the axis on the left, time and days on the y-axis, x-axis. People who are on treatment come in, they undergo what's called an analytic treatment interruption, ATI. We stop antiretroviral drugs to see what happens. And if bad things happen, if the virus takes off, we put them back on treatment. We can do this pretty safely. We do this all the time. Goal here is that on the day you stop treatment, we infuse the CAR T cells. If they get the conditioning, we do the conditioning 3 days earlier. But you infuse the cells on the day we stop treatment with the idea that the cells will be active once the virus begins to rebound. And so you have a low amount of virus that's now starting to rebound, and we time it such that the T cells basically block that from happening, prevent the explosion, and allow the immune system to get a hold of the virus and maintain control. That was the theory. Okay? What happened? First of all, it's very important. I'm going to show you the data pretty quickly, but you need to know we have done thousands and thousands of treatment interruptions in people with HIV. We know what happens. You stop drugs, 2 weeks later, the virus begins to rebound. It takes off, and it goes up exponentially. It happens in people who start therapy early, who generally do better, or people who start therapy late. Every now and then, maybe 5% of the time, someone will control the virus because their immune system works. All right, the study, we basically enrolled people who were between the ages of 18 and 70 on long-term treatment, didn't have had a pretty good immune system, didn't have any significant morbidity, and so forth. Now, I know I'm going to get asked about this, and I apologize up front. We enrolled only men, which is very classic in these types of cure studies. These are very intensive cure studies and require a lot of people to basically have a lot of time to do these studies and have been on long-term treatment. In the urban centers in the United States, that generally turns out to be older men. Women have lots of HIV, tend to be younger, tend not to have been on treatment longer, and there are a lot of other significant barriers to getting women into these types of studies. We did have a fair amount of race and ethnicity diversity, but all cisgender men. Okay. Cohort 1, no conditioning, low dose of cells, did not expect to see anything, and we did not see anything. Okay. These are 3 people. They all started therapy late. You can see where the virus, they stopped therapy at time 0 when they got the infusion. About 2 to 3 weeks later, virus takes off, gets to a high dose, put them on treatment. Okay. 1, 2, 3, not much going on. And not too much of a surprise. They didn't get any of the bone marrow conditioning. They didn't get the lymphodepletion, the space for the cells to go, and the dose was low, and we did not see very much of the CAR T cells in the blood. Cohort 2 now is where we expected to see something. Okay, 3 people. First person, again, nothing. Standard rebound. Chronically infected person, started therapy very late. No rebound for 10 days, virus takes off, goes on treatment. The next person is different. This person started therapy during early infection, within a few weeks of the infection, so which is generally a good thing. He stopped therapy, and we went back and we measured. He had no virus, no treatment in his blood, and he's controlled for almost 2 years. Now, as I said before, people do control every now and then, but they typically don't control at this very low level for this very long time. So this was very unusual, but not impossible. This could have happened without CAR T cells. I guess maybe 1 or 2% of the time this would happen at this level for this long. So that was surprising, but again, not definitive. But then it happened again. Same exact story, more or less. This person now is out closer to, he just came back yesterday from a long trip. His virus is still undetectable. It's been now over a year. Same story. And now that is totally unexpected to have two people with such a rare event. But not totally impossible, but most of my colleagues who've seen this found this intriguing, that there must be something going on. And the amount of CAR T cells that was in the blood was a bit higher than in the previous cohort. Cohort 3, the low dose ablation and a standard dose of cells. First person, chronic infection. Again, not much happened. Second person started therapy early. Again, control, not tight control, but the type of control that we typically see. Virus did not explode and go up to 1 million. It went up to about 1,000, which is a very low number, stayed there for a few months, slowly rebounded. So clearly a biologic effect, not quite as impressive, but 3 out of 3 people who started therapy early have now shown some evidence of control, and 2 have shown very tight evidence of control. The last person is a bit more interesting in terms of the fact that he actually had chronic infection, which again is he started therapy late in disease. His T cells got down to 200. Usually people's immune system doesn't really ever get rebuilt, but he went back on treatment. He went on the study, and he surprisingly is controlled for over 3 months, which is actually quite very, very unusual. So 4 of the 9 people in this study, a very, very high percentage, showed some evidence of control. But the issue is safety. Okay? People with HIV are not people with cancer, right? They have options. And so the threshold for this type of intervention is quite higher. This is going to have to be scalable, affordable, tolerable, safe, and so forth. And we have pathways to get there. But the big thing that drove most of our concern early on was the concept that these CAR T cells would cause the classic CAR T cell side effects, cytokine release, the neurologic stuff, ICANS. We did not see any of that. And in fact, this has not been seen in other studies of infectious disease and even rare autoimmunity. So we don't think it's going to be a major problem. We can talk about why. One person, unexpected, probably had some pre-existing, we're pretty sure had some pre-existing bone marrow reserve problems that we did not know about. We're trying to figure this out. But he actually got the low dose cyclophosphamide, which traditionally just causes a slight dip in his numbers, actually got profound pancytopenia. That resolved in terms of neutropenia. His lymphopenia is slowly resolving, but this was a serious adverse event, unexpected, in the study. And we had no problems with treatment interruption. In my conclusions, we can say that the product in this study was safe. We saw none of the toxicity that concerned us. The conditioning regimen did cause an issue. We're thinking about ways to avoid conditioning in the future. CAR levels did persist, but lower than we typically see in cancer. We think that might be because the amount of antigen around to induce the CARs is low. Post-treatment control, 3 out of 3 people started early did have substantial control, really unprecedented to see those kind of numbers, but the numbers are small. But most of my colleagues who've seen this are intrigued, think something's going on, and the question is what to do next. And we do believe that these data do support moving forward. We don't have specific plans on how to do this, and there's going to be a fair amount of discussion within the field on how best to go about doing this in terms of whether it's ready for a randomized study or we need to do more dose escalation. Extended team, I mentioned the great team at UC Davis. I mentioned Borrow and his team at Karen Cross. And once again, I do want to basically show my appreciation for CIRM. And they showed a lot of bravery and tolerance and patience in getting the study done. Thank you. Any questions?
Floor will be open. Yes, John.
One quick question. You mentioned about the number of circulating CAR T cells. Did you do a bone biopsy or bone marrow biopsy to assess engraftment based on the lighter neuroablative versus the heavier?
We have that capacity. We do bone marrows for research, but we did not do it in this study. We did do some gut biopsies, and we found some evidence of persistence. HIV in this setting, at low amount of HIV, it accumulates in lymph nodes. That's where it is. And so, that's where we think the T cells are. And so if we get these gentlemen to come back, they're at Davis, so it's not easy. We plan to do lymph node biopsies to see well, that's where we think the cells are.
Thank you for your presentation. I think you anticipated correctly. I'm going to ask you a question about having this study be limited to all men. I mean, 36 years after NIH established the Office of Women Research, Research on Women's Health, I think I need more than what you said to explain why you excluded women, especially since you are projecting that this result of this study may have implications not only nationally, but globally.
I cannot defend what we did. I can just explain that it is, we tried. We always try. And we do cure studies all the time, and they're not always 100% men, but they're usually 90%. And we did one study where we actually enrolled 50% women, right? But it required us to have a separate $250,000 grant to do the outreach, right? To do the education. There's a lot of skepticism among the populations that we run into, particularly about gene therapy, which is fine. We can overcome that. But you need to provide transportation, childcare, and we did it, but it was expensive and it required an intense effort. But this is definitely a problem. We have studies in Africa too, where they have the opposite problem. There it's all women. The men are very hard to engage. And so I do think at one point in time that there's already discussions about studying this in an African setting where it'll likely be all women. I'm sure that was not sufficient. And I've been answering that question for 30 years. You think I would get better at it?
Any other questions from board members?
Just,
I'm sorry, I can't see you. Yeah.
Yeah, Anne-Marie, how long would you expect the effect to last?
Well, that's a great question. We actually did back in the 90s, some of the first CAR T cell studies ever done. NIH funded. There's a company down the road here called Cell Genesis. Many may remember that. It was the first CAR T cell company, and it was HIV. Didn't really work. First generation. Bora was involved. Then everyone left Cell Genesis and HIV and did great things in cancer. They're all coming back now. But we went back and we looked at those guys that got CAR T cells in 97, 98, 10, 15 years later. These cells were there, easy to find. So it's actually the longest study ever done in terms of persistence. Having said that, we did not see that so much here. We're not entirely sure why. The amount of cells that we saw are lower than expected, and they really basically were only detectable for a few months. But 3 things. Number one, these people probably, because they had such low virus to begin with, all that virus is probably in lymph nodes. So we think their T cells are there. Number 2, there's a theory that we believe that if you have the right kind of cells at the time that virus explodes, 2 to 4 weeks, and you blunt that, the immune system can get its act together. So the CAR T cells are doing all the work while the B cells and NK cells and everything else gets their act together and does what it's supposed to do, which is control the virus. So we think the CAR T cells may actually be allowing the immune system time to take over. And that's in fact what we're seeing. Number 3, if you look at the amount of disease, right, in cancer, it's like a basketball. In autoimmunity, where CAR T cells are having this great effect, it's like a golf ball. In HIV, it's it's less than a BB. We have very little, the number of cells that have HIV are tiny. It's not cancer. So the number of cells that you need is going to be tiny. And with a little bit of antigen around, you're not seeing much expansion. So we think that the amount of, even though we have trouble finding the cells, if they're in the tissues, we don't need very many of them because there's not much virus. But the question that you ask is the focus of a lot of the mechanistic work we're doing.
Thank you very much for your presentation. I'm from UC Davis, so thank you for highlighting
UC
my
Davis.
colleague, Dr. Abedi.
Yes.
I wondered, and this also speaks to the sustainability of the effect, is there any possibility for synergism with ART such that you might be able to maintain people on a lower dose of ART and maybe address some of the compliance issues?
That's another great idea. The idea being that, you know, ART is fine as long as you take it. People can't take it.
Yeah.
They go on and off. I mean, if there are 1,000 people online who have adherence problems, there'll be 1,000 reasons for it. Everyone's got an issue. And yeah, so you're right. So if you give a CAR T cell and they just provide the buffer, the background. So if they go on and off, that is absolutely something that there's there's some discussion about that.
Thank you. Any other questions?
Thank
If
you.
not, we're going to thank you for this absolutely wonderful presentation.
Thank you.
Thank you. And we are now going to adjourn for lunch, and we are due back in this room at?
We are due back at 1:30. That's when they'll shut down lunch. Members of the public, there are resources in the mezzanine for the rest of the team and board. If we could ask that the board members get first crack at the line so that we can make sure that we can resume at 1:30, and then the team, that would be great. Thank you. We'll resume at 1:30.
Our meeting in 2 minutes.
To have a little sound for the remote audience.
Well above the
I understand. Some of these already have it open because you were looking for
If everybody would take their seat, we're going to start the meeting here in about 15 seconds. Thank you.
We need a lot more people. We're getting ready to start. Okay, everyone. All right, ladies and gentlemen, members of the board, before we proceed with the next agenda item, I'd just like to backtrack a second. After some reconsideration of what happened earlier, I wanted to say something about the vote we took on the CLIN 2 application, the one especially that did not have the opportunity to hear all remarks from the public. And I think I'd like to have entered in this discussion into the record. The best way to do that would be to ask, is there any member here, member of the Application Review Subcommittee, the ARS, the people who can vote on these applications, who would like to make a motion to reconsider our vote? I'm not necessarily asking for you to do that, but if there's anyone that would like to do that. Is there any member of the ARS not in the room but online? And there are no hands. Okay, there is no motion on the table. I'm going to proceed with the agenda then, Scott. Pardon? I'm going to proceed with the agenda. Okay. Thank you. So next, pardon me, I want to introduce a resolution to honor the many years of service to CIRM given by our dedicated and talented transcriptionist, Beth Drain. Beth is our doyen of digital dexterity. You see where this is going. It would take a thesaurus to name all the nouns that describe her. Stenographer does not begin to do her justice. She is our escritoire par excellence, the amanuensis of our words and deeds. Titles like copyist, penman, or calligrapher would have once been appropriate, but long ago are the days of quill pens, inkwells, and sheepskin. Beth instead uses all the modern audio and visual modalities to be our truthful registrar of words. Her shorthand keyboard chronicles our history. Her computer screen is our modern palimpsest on which she scribes the arguments for and against and notarizes our votes for posterity. She is our scrivener, our transcriber, our typist recorder, and our clerk of record. And she does this in person, even though that means since 2012, she has to commute to our meetings from the great state of Idaho. And like some spiritual doula, Beth was there at the birth of CIRM, attending the earliest meetings of this board starting in 2005. She has transcribed 228 meetings of the ICOC, many of them held conjointly with the Application Review Subcommittee. But she also covered all the other subcommittees of this board from their beginnings: governance, finance, standards, intellectual property, and presidential search committees. She expanded her domain earlier in this decade with the advent of the Accessibility and Affordability Working Group and the Communications Subcommittee, not to mention the Task Force on Neuroscience and Medicine. In total, Beth has transcribed 52,039 pages. That's 11,771,189 words of open session hearings. Clearly for Beth, no tabula is ever left rasa for very long. Beth has been eyewitness to CIRM's genesis and evolution, documentarian of its history. She is in a real sense a cornerstone of this institute. On occasion, she would have only an audio recording to work from without benefit of video, but she had come to know all our voices, our verbal tics, our syntactic idiosyncrasies, and she would gloss over our hems and haws, squelch the ers, ahs, and ums, and produced works of flawless accuracy. On paper, it might seem to a reader that we all think and speak in polished paragraphs. And she did this all with profound kindness and warmth. Her routine professional interactions have grown into lasting personal connections that have left an indelible mark of care and community upon CIRM. So therefore, be it resolved that the governing board of the California Institute for Regenerative Medicine, on behalf of the people of the state of California, wishes to express its deepest gratitude to Beth C. Drain for her service to CIRM and for her dedication to the advancement of stem cell research and to our mission. Thank you, Beth.
Motion to accept the resolution.
Seconded by Christina. Thank you. So let's start the discussion. I think Vice Chair Maria would like to say a few words.
Beth is simply someone you cannot imagine CIRM without. Since 2005, she's shown up meeting after meeting, year after year. The story of CIRM has been captured all these years by Beth. She is our historian. But what makes Beth truly special isn't just the numbers, it's who she is. Her professionalism has always been matched by a genuine warmth. We don't just appreciate Beth, we love having her in the room. She brings a steadiness and a kindness to CIRM that leaves an impression on everyone around her. And she leaves behind not just a legacy of impeccable work, but a community of people who are deeply grateful to have her. Thank you.
Mr. President, Jonathan Thomas.
So Beth, it's only fitting that in a meeting that we are paying tribute to Jeff Lomax, who's been here seemingly forever, that you well predate him and predate everybody in this room, including Gil and Scott and Tricia, if you're listening, everybody. You've been here since inception. Your skill set, I've always felt, is way beyond my competency level. I don't know how you do what you do. It's a real treat that somebody can handle that task and be the scribe and the historian who will forever be etched in print and accessible to all of those who go back as CIRM proceeds through its process, however long that lasts, and will have the record of everything in real times that happened, which is critical to not just here, but anywhere else that you have worked with as well. So on behalf of the team, we want to thank you for everything you've done for all these many years. It's been a real pleasure to have you as a colleague in all of our meetings, and we'll miss you very much. So thank you.
I think I'll be brief. Not known for that, but just because everything everyone else has said absolutely captures what I would want to say. Couple things. I had some free time. All those words, that's 14 copies of War and Peace that you did. 67 24-hour days of nonstop speech. So, does it feel like it? I just wanted to thank you, Beth, because ever since that winter in '05 at City of Hope, which was my first meeting, you've always been here on my left side. It would be very strange to go forward without you here, and I will miss you very much.
You too.
Thank you.
Thank you very much.
Beth, we have this token of our appreciation for you and all your years of service. So thank you. This is for you.
Thank you very much.
Yeah.
Thank you very much.
Mhm.
Thank you. Thank you. It's a bittersweet moment for me. I've been a reporter for over 40 years now, and it just really dawned on me in the last few months that I'm 78 years old and I shouldn't have to be working. So even though I won't be here in person, I'll still think of you all on a regular basis, even though your faces change over time. And I hope that I'll be able to still contribute in maybe editing your AI transcripts. We'll see if we'll work that out. Then I won't have to travel. Since my husband is older than I am and not well, I'm very uncomfortable leaving him at home alone. So even though I'll miss you, thank you for having given me the opportunity all those years ago when I first met Mr. Klein and Melissa King, that then led to the very beginnings of CIRM when you did your first, I can't even remember the name of the group now. What was that best? I can't remember the name of it now. Yeah. Can't remember. That's how long it is. So thank you very much. And let's get back to work so we can have that glass of wine at the end. Thank you.
Right. Well, we can't do it without you, so. If there are any other comments. Okay.
I just wanted to say that you were seated next to me a few times when we were here, and I was impressed by your digital prowess, but also by your ability to withstand the distractions that I posed. So thank you.
Okay.
Mr. Chair,
Sure.
we actually need to take a vote.
I was just thinking, oh, wait, Mr. Jensen?
Okay.
He's at the microphone. Public comment before the vote.
Yeah, my name is David Jensen. I've been writing about the California Stem Cell Agency for more than 20 years.
Get closer to the microphone.
No, you can't hear me?
Now I can.
I don't. Okay. And one of the things I wanted to say, how much I appreciate all of the kudos that Beth has received are well-deserved. And I think the CIRM itself should be congratulated itself on the openness that it has demonstrated by producing these transcripts. Few other governing boards of any agency in state government produce transcripts on a regular basis. And it's been a real asset for the public to see. And I think it's CIRM does a great, should be saluted for the public service it has performed in providing those transcripts. And thank you, Beth.
Oh, that's sweet. Thank you. Any other member of the public like to make a comment? If not, we're going to take a vote, Scott.
All right, it'll be a voice vote in the room, and I'll pull the members on the Zoom individually. All those in the room in favor say aye.
Aye.
Those opposed or abstain? And on the phone, Leandra Clark Harvey,
Aye.
Monica Carson,
Aye.
Rich Lahara, Carolyn Meltzer,
Aye.
Shauna Stark,
Aye.
Kevin Zhu.
Aye.
I don't think, Mark Fisher Colbury. Great. Thank you very much. Motion carried.
Thank you. And thank you again, Beth. We're going to move on to agenda item number 16. This is the Infrastructure 9 program, Data Science and Software Engineering Concept Plan. Janie Byram is going to lead the presentation. Thank you, Janie.
Thank you. Great, thanks. My name is Janie Byram, and it's my pleasure to present the Data Science and Software Engineering Awards Concept Plan to you on behalf of the CIRM team today. And our mission, we start with, our mission drives us, and CIRM's mission is to accelerate world-class science to deliver transformative regenerative medicine treatments to a diverse California and the world. Or in an equitable manner to a diverse California and the world. And so today we will share the concept plan that is designed to maximize the value of CIRM's initial investment and its research awards and accelerate world-class science through the development of new tools. All right. So for today's concept, we'll begin with some background information, and then I'll move into the elements of the concept. So I want to begin by emphasizing that life sciences, including regenerative medicine, has seen massive data growth in the last 20 years, and we need transformative tools to help researchers capitalize on that data. And this is due to several factors: technological advancement, including in the life sciences, and advances in computation, increased investment in research and development, and more data accessibility due to data sharing. So data generation is doubling every 2 to 3 years, and we need software tools to capitalize on the volume and variety of data. And so these data are rich, but they are underutilized, and they must be integrated to allow for comparisons across data sets, across models, and across disciplines. So we need tools to integrate data at a large scale and be able to integrate different types of data. So I'll elaborate on that in the next few slides. Right. This is a quote by Francis Collins, the former head of the NIH, addressing an HHS House subcommittee about the future of biomedical research. And he says, The challenge is how to store, retrieve, integrate, and analyze this mountain of complex data. And it underscores that we need to be able to leverage all of the data that we're generating so that we can reveal meaningful insights. And CIRM has a goal of maximizing the value of this data, and this is for two primary reasons. If the data remain siloed, then we're leaving insights on the table and sciences will be slower. And it's also inefficient. Being able to squeeze the juice out of individual data sets by sharing them and combining them with other data sets reduces the amount of data generation that needs to be funded in the first place. So recently, in 2023, CIRM began tracking research data sets generated by our awardees, and we see that CIRM researchers generate vast amounts of diverse data. So illustrated here is the variety of data sets from different techniques or approaches, and these data sets were tracked using CIRM data sharing and management plans. And you can learn more about these data sets at CIRM Data Explorer at datahub.cirm.ca.gov. And the goal of CIRM Data Explorer is to promote the findability of CIRM-funded research to other researchers so that they can incorporate these into their research programs. So some of these data sets are publicly available and some of these data sets are still in progress. And this is primarily data from discovery stage awards, but we have recently implemented our data sharing and management plans for our preclinical and clinical awards, and we'll be tracking those outputs as well. So I want to emphasize here the variety of data types and the volume. Each circle is proportional to the number of data sets generated using that technique. And you can see we have a lot of transcriptomics, for example. And additionally, within each technique category, there are sub-approaches. So each of the smaller circles, I'm not sure if it's showing very well on the TV, but each of the circles shown within the larger categories is a different flavor of a broader category, and their sizes are also proportional to the number of data sets. So these data are generated using a lot of different approaches, and it's a huge challenge to compare data across techniques, and that's limiting what we can learn from these data sets. So in addition to the diversity of data across techniques, data is also generated across scales and models. For example, animal models, cellular and molecular data, electronic health records, and patient data from trials. So being able to bring these data together will enable researchers to develop more accurate and more complex biological models, and they can ask more nuanced questions. And in general, more connections equals more potential to find targets or biomarkers because there's a fuller picture of what is interpretable by researchers. So how can we integrate and analyze the mountain of complex data? We can build software tools to integrate data, and this can accelerate research. So during the strategic allocation framework, CIRM defined an ambitious goal to catalyze the identification and validation of at least 4 novel targets and biomarkers, ensuring integration into preclinical or clinical research for diseases in California. And there was a recommendation at the time to enable data science collaborative efforts via dedicated grants. And so to increase the probability that CIRM realizes this goal, we can address research bottlenecks by supporting software projects that help the researchers make connections between different types of data, enabling more impactful data science. And I'll outline some of these research bottlenecks in the next slide. So multimodal data, including patient data and data across scales, disease areas, and biological systems, they need to be integrated to be leveraged for biological insight, and this requires significant investment. Software tools are needed to accelerate stem cell and genetic research, stem cell-based and genetic research. And additionally, dedicated funding is lacking for the development and maintenance of open source software that's crucial for the discovery of potential therapeutics. We surveyed our Remind awardees at the Remind annual meeting, and the biggest bottlenecks to employing cutting-edge data science in their labs was funding for a data scientist and also software tools that are user-friendly and don't require a lot of computational expertise. So those bottlenecks can be addressed through a program that develops and sustains open source software that drives regenerative medicine research and can catalyze data science research to integrate and expand the impact of both CIRM-funded data sets and also other data sets in the research community. So what is open source software? Open source software is software whose code is publicly available, allowing anyone to view, modify, enhance it, and redistribute it. And I wanted to give an example of open source software that's been essential in the life sciences. So this is scikit-learn, which is a machine learning library for a programming language called Python. And the name comes from Scientific Toolkit for Machine Learning. And this toolkit is the most popular machine learning framework. And in a survey of over 24,000 people, over 80% of respondents use this package. And on GitHub, which is a code repository, over 65,000 people have liked it or given it stars. And the original paper that introduced this package has been cited over 130,000 times. So it's very well used, but what does it do? It's a software toolkit for scientists that are not experts in coding, and it can be used to learn from a data set and help with predictions and classification and clustering. So based on this data set and the data sets that the model was trained on, what happens next? And also, if you have a number of cells, do they belong to an individual that has a disease or a healthy individual? That's classification. Or clustering. Can we put cells that have similar phenotypes together? So this has started as a very small software package built one summer by a PhD student in a Google Summer of Code. But since it was an open source project and because there was a need for it in the community, it's been developed over time into what it is today. And it has made data science accessible to researchers and is now an essential open source software for scientists. And so I just told you that computational software and infrastructure are integral to scientific research, and the research enterprise is increasingly relying on open source software. And today we're proposing a concept plan for data science and software engineering awards to support open source software that has a specific scope of solving data integration barriers in regenerative medicine. And so CIRM can support reusable, scalable software tools to link diverse data sets, and this will accelerate research in regenerative medicine and ultimately maximize the potential of CIRM-funded data. And so during the development of this concept, we've received feedback that we already support data science projects in our discovery awards. And here I want to highlight that the software tools coming out of Infrastructure 9 would be distinct from those that are resulting from our existing discovery programs. Tools supported by our discovery programs are generally ancillary to research projects, and they have no requirements for software engineering standards and best practices. And they're also highly specialized for a specific research question, often without a plan for deployment or community use and maintenance. And so the maintenance tends to end whenever the paper is published or the trainee leaves the lab. And so Infrastructure 9 projects would follow open source best practices, resulting in tools with enhanced scalability, reusability, and reproducibility. And through a dedicated grant mechanism with requirements that are tailored for software tools, we can have a collection of tools that are built for extension and community use in mind with a plan for maintenance. So now we'll move into the scope and structure of the concept for the Data Science and Software Engineering Awards, and I'll start by previewing the proposed guiding principles. So as a reminder, guiding principles help CIRM shape its portfolio, and the Infrastructure 9 portfolio will address barriers to integrating multimodal data in regenerative medicine and innovate through open and collaborative software development to accelerate research and to create broadly applicable and validated tools that maximize the value of CIRM-funded data. And these guiding principles will inform the selection and review criteria, as well as CIRM team recommendations post-grants working group. Okay, moving into the objective and approach. Okay, so the overarching objective of this program is to support the development, maintenance, and/or extension of innovative open source software for integration of disparate data types to accelerate research in regenerative medicine. And the expected outcome is the development and deployment of an open source software tool to solve a multimodal data integration bottleneck in stem cell-based and genetic therapy research that has wide applicability and scalable impact. And the approach is investigator pairs leading interdisciplinary software engineering projects.
And we propose that the scope should focus on software tools, that is computational tools, pipelines, or resources to address data integration bottlenecks to catalyze data science and regenerative medicine research. And this scope would bring about tools that accelerate the identification or validation of novel targets and biomarkers. And we focus this scope to supporting the stage of research where we know there's a need for this program, which is our discovery stage. And so examples of types of approaches that could be employed in this award program would be AI, artificial intelligence or machine learning AI or ML approaches for in silico screening, protein design, molecule generation, cell and gene engineering, et cetera. Or knowledge graphs for bridging structures, behavior assays, functional genomics data, spatial data, and pathway or network data. So a knowledge graph is a framework for connecting diverse data sources, and they're used for modeling complex networks to map relationships between genes, pathways, drug targets, treatments, et cetera. So how could these approaches be applied? Some specific examples of applications could include a cell analytics software to read out present and future cell states without having to destruct the cells. So read out behavior without having to destroy any cells, or a new omics analysis tool to integrate spatial transcriptomics data with multi-electrode array data to link spatial gene expression and electrical signaling to understand disease mechanisms and identify new targets. So this program would have an annual call with an award duration of up to 2 years, and the award amount is up to $500,000 total costs, funding around 15 to 20 awards per call with an annual budget of $10 million. In terms of eligibility, the applicant must meet CIRM's definition of a California organization. We'd have 2 California-based investigators, and the co-I must not be a part of the same lab as the PI. And there will be requirements for both regenerative medicine expertise and computational expertise on the core applicant team. And the effort for both investigators be a 5% minimum effort. For application and review processes, Infrastructure 9 will adopt a GWG selection process to allow the GWG to select applications that are most responsive to the funding opportunity and hold the greatest potential for impact, and also allow CIRM pre-planning for improved scientific and technical review. Infrastructure 9 would also adopt a 1 to 100 numerical GWG scoring system to align across CIRM programs and improve granularity and visibility for the score driving decisions. And the selection and review criteria would be developed in alignment with the guiding principles that I previewed earlier. I'll wrap up by sharing our timeline and outreach. Today, we're at the June ICOC meeting presenting the concept, and in September, we could open the program announcement with applications due in November and or with applications open in November and due in January, with a longer window to the grants working group review in May to allow for GWG selection, and then ARS meeting in June 2027. We have an outreach plan in development to ensure that the potential applicants are made aware of the funding opportunity. So in addition to targeting our existing awardee portfolio, we intend to target outreach to non-UCs, community colleges, and Cal State universities. We think this program has the potential to be more accessible to these institutions relative to R&D awards because the program requires less capital equipment and physical lab space, and the award can be accomplished with computational resources and personnel. And we also plan to reach out to professional organizations and data science and informatics networks and academic departments to spread the word. And below are some of the tools that we can use in collaboration with our communications department to get the word out. And I'm very keen to hear your feedback, and thank you for your consideration of this concept. And I'll stop here for discussion, but ultimately we'd like to request that the ICOC approve the data science and software engineering concept plan. Thank you.
Thank you, Jamie, for the presentation. This is a request to the board to approve the concept plan. We need a motion to consider and start the discussion.
Second.
Okay, we have a movement and a second. Thank you, gentlemen. So the discussion is open first to members of the board. Judy?
This may not be, my question may not be directly related to the program, but just curious about, have we stratified the data we have now in the stage of discovery, pre-dep and clinical, and see how the applicant, what kind of source of data? You have this whole summary about overall, right? But for us, probably particularly intrigued about, you like see that involvement about enrich certain data, and that's how we want to encourage the applicants. So that's my question number one. Question number two is then, when we look at the applicants, have we done this something like the high score versus the low score? What particular data source they have been using? And this may actually be a useful tool to guide our applicants about what we are looking for.
I'm not sure I understand your second question, if you could rephrase it.
Yeah, so we have review and approve so many projects by now, right? So there are some success cases, some I wouldn't call failure case, but like then more or less didn't make it to get funded. They, in their submission, they all have supporting data. I mean, typical scientific evidence. Do we see any algorithm about the success case tends to be having those amount of data will drive for that? Because I think that's not necessary for our approval. I trust our review group has been doing a nice job, but more about to give the applicants some guidance about what we are looking for.
Yeah, that's a great question. I actually, so I haven't done that analysis about whether applications that propose to do, larger data generation or more sophisticated computational analyses, whether they do tend to perform better at review versus those that do not. Is that the heart of your question?
Yeah, that's the second part of the question. Yeah, I think that would be interesting. Now we have so many, our sample size is big enough.
Yes, I am not sure about that. In regards to your first question, whether we've done the stratification of discovery, preclinical and clinical data. So we have been tracking our discovery stage awards since 2023, and we just implemented the data sharing and management plans for our preclinical and clinical awards in January. So we don't have data to compare to for those programs of what they're generating and where they're sharing it, and whether there's an abundance of that data out in the world that can be used to for these types of programs. And so that's why we're focusing the scope on discovery right now, because we have the data for the discovery programs. We know that they have been sharing, and it's been a requirement of our programs for several years now. So
and my thought is then while we are establishing this infrastructure will be a good idea. We see where it's going. So then make this infrastructure much more applicable all the way down to the whole life cycle of product development.
And that, if I may,
yes.
Dr. Chow, this is the goal, and I'm not I'm just complementing the great response from Janie. So the goal right now, we had initially very early on with pre-calls with the science subcommittee. The goal was very ambitious to tackle discovery, preclinical, clinical. We also realized we don't have enough categorized data because the data sharing management plans for preclinical and clinical have been implemented in the last couple of years. There is not enough understanding and enough power on that data. And the focus is on the discovery, especially on the networks that I mentioned earlier on with the Remind these four. And now that we are going to have enough power with like 2 rounds of calls of this $87 million funding from or investment from the state of California into these networks, how can we exponentially leverage? And the focus is discovery of novel targets, biomarkers. And as we learn, and there's also the incoming power of AI and everything, as we learn all of this, our intention is to move into potentially preclinical and clinical, and there is a budget item that is in later years that could implement, take care of this. Hopefully that answers.
Thank you. So does that mean we have an AI platform already?
No. What I meant with my comment was that there are new technologies, AI included, that are coming up. And we are just learning. And as we are evolving, there might be an opportunity for CIRM to leverage this, but I'm not saying we have that at all. No, that's not what I was.
Thank you.
Are there any other questions? Yes, Pat.
That's great. We heard this morning about a rebalancing, whatever the right word is, the budget that changes things a little bit. I'm wondering, so you showed us the timeline, and I can't remember. We went through this before. That's phase one. Is there a consideration that this is
Yeah.
going to
Yeah.
continue?
Yes, we have budget projections for the lifetime of CIRM. However, annually, the board would determine whether that is something that you want to fund, and there's opportunity for evolution of this project
And I
as
think
well.
what Dr. Levitt is asking is also that gap that is created by one gap year of the DIS4, whether it's supplemented by this, and the response is yes, that's how we have designed it. So now it will start by the time that these are out is when the gap of DIS4 will happen. And we will be able to, these teams are the ones that we hope that they will come to us. And through the network that we have yearly meetings, the hope is that there will be questions there that will generate like the type of solutions that we hope to fund with this.
So then the other question I have is, because this is an area of, technology that's changing by the hour, right?
Absolutely.
And so the reason why Python is because it was actually built to be Python. It was built to be a foundation upon which thousands of computational folks or those code could actually use it as a tool, as a starting point. Is this going to be an area of emphasis when you describe the kinds of projects that you want to see, right? They're going to want to know, because you can also design very specific pipelines for specific projects that may not have that kind of flexibility and therefore may be dated in a relatively short period of time. So what are your thoughts about that?
That's a great question. So the review criteria that the grants working group will use to evaluate the applications will consider how broadly applicable these tools will be to the scientific community, and also what their plans for maintenance or sustainability are in terms of how user adoption as it changes, whether there will be a contributor community that has appetite for building on these tools and developing them further. And also with them being 2-year awards, they can also come back for reinvestment as well, so that they can continue to be supported if it turns out that they are a successful, needed tool in the regenerative medicine community.
Hi, so thank you so much, excuse me, for this. And I think this is so important. So I'm really excited to see you all tackling this. I have some experience with software development, not open source and not in science. So just a couple questions, and I think a lot of this discussion has been really helpful, that people will be able to come back for more money if they need to maintain, because you always need to maintain.
Absolutely.
Right. All right, that's great. The GWG will be reviewing these applications. Will there be some like technology reviewers added to help us?
Yes, so
Yeah.
it is a gap in our current review panel that we will have to outreach and find suitable reviewers to build a panel that will be able to accurately, rigorously review these. And it will be a mix of regenerative medicine expertise as well as computational expertise and open source, people with the open source background, development background, and software engineering expertise.
Okay, that sounds great.
And one other note on that is that we have this need for these reviewers, the computational experts in our other programs as well. And so they will be able to serve on ideally multiple review panels for our discovery programs as well.
Cool. Yes. I'm just curious, does scientific, oh, did you want to say
No,
something?
I just wanted to add something about the maintenance. The goal is not sustainability. And we need to be careful with that, because one of the things that we need to to promote with institutions is once there is the funding from CIRM to develop these tools, is finding ways, not maybe in 2 years, but maybe in 3, to figure out ways for sustainability in the long term, because as we said, we are finite, right? So I just want to clarify this point.
Okay, yeah, that's important.
point.
How much cleaning does scientific data require?
Cleaning?
Yeah, I ran into that a lot with communications data coming from different,
Yes, I think it depends on the data type, but I think all data types require substantial amount of cleaning and harmonizing and wrangling so that you can actually integrate these disparate data types. And so that is something that would be an allowable activity is getting data into the shape that it needs to be to interface with other data types.
And that's something also then that's ongoing that will, need to be done as every project generates all this tons of data. Oh, there was one other question I had. And so, is the CIRM team going to be kind of monitoring progress and creating milestones? And so do you all have the technology expertise that you need?
Yes.
Yes.
So we do have some of that expertise in-house, and we also can rely on an expert network that we can develop to have advising, but they will be milestone awards and have progress reports that they report on semi-annual intervals and be reviewed by science officers with appropriate expertise.
Thank you. Dr. Carruthers.
Yeah, exciting, very exciting. Could one of the goals be, I mean, I know when data is generated by many of the different grantees, and the purpose is to try to get into some, I assume, some standard format for other, because it's public, it should be public accessible. Is the goal, or could the goal be that there's, I'm not saying a central repository, but a central front door that could be through CIRM or CIRM's successor or whatever, that if I'm a 25-year-old researcher, wanted to see some former gene therapy data, how would I find that? Is because the first one I would think is probably CIRM versus was it this person in this place in this thing. So I don't know if that's one of the goals, or could that be one of the goals, that at least there's a front door to access the data once all this is done.
Yes, wonderful question. So we actually have a platform that is new in the last year called CIRM Data Explorer that is for tracking where CIRM-funded data has been stored. It is, as you say, more like a front door than an actual repository. People can browse the data sets to find where, if they're in GEO, if they're in dbGaP, where they have been deposited and their accession number, so that a researcher can go and ask a question, is there diabetes related research in this area using this data type, and where can I find that data?
So Janie, I just want to take a step back, because this is something that we plan to bring to the board to explain it. And it's been actually, Janie, so the data infrastructure part of our programs is something fairly new that we created, and Janie's leading this. And we hired a science officer with data expertise that works with Janie directly, and then they are very coordinated with the discovery team. But we also developed this data explorer that also took the legacy data from the genomics and everything, and other data that we've worked with Rancho Bioscience to bring it in there. We haven't done a good job to communicate that, because you just asked the question, right? So that's something that's in our radar, and that we want to polish that and also communicate it better. But I just want to also give a kudos to Janie that took this and has been working very hard to make it possible.
Thank you.
Okay, let's see. I don't see any question. Anyone on Zoom? No. Okay. Is there any public comment on this item? It doesn't seem to be in the room, anything, and nothing online. Any further comments from board members? If not, we can go to a vote on this request to approve the concept plan for Infrastructure 9.
Okay, thank you. And again, it'll be a voice vote in the room, and I'll poll the members on the Zoom. All those in the room in favor say aye.
Aye.
Those opposed say nay. Any abstentions? And on the phone, Leandra Clark Harvey,
Aye.
Monica Carson,
Aye.
Carolyn Meltzer,
Aye.
Shauna Stark,
Aye.
and Kevin Zhu.
Aye.
Great. Thank you very much, Chair.
Thank you.
Thank you. Next, I'm going to invite Dr. Sembrano back.
Actually, sorry, little discussion while that other item is going on for process for voting, making sure we can preserve quorum. We're actually going to, if it is good with the group, to proceed to the voting items on the budget and the contract, and then we'll come back
Smart.
to the informational item.
Great. So should we go to 18, the research budget?
Please.
That would be, and there she comes. Jen Lewis, Vice President for Operations.
Thank you, Dr. Balsiani and members of the board and CIRM team and public. My name is Jennifer Lewis, and I'm here today to present the 26-27 research budget to you. And so today I'm going to go over the awarded funds overview. So to give you a snapshot of where we are in our research spending, you heard from my colleague Rosa this morning, but I'll give a more in-depth and related to Prop 14, the actuals from the 25-26 research budget and the 26-27 proposed research budget. So this first slide shows the total funds available as of April 30th, 2026. The 7.7 billion covers both propositions and any return of funds that CIRM has had over its lifespan. That includes loans and interest payments, which I'll talk about a little bit more. As you can see, we've encumbered just over 50% of that portfolio, and we have roughly just a little over 3 billion remaining to allocate over the next several years. This next slide, I want to pause for a moment, and this is a new slide that we added in response to Board Member Daron and her in previous meetings asking for more clarity about the return of funds that come back to CIRM and those different areas. So I'd like to walk you through this slide and share with you a little bit more. This is as of the last quarter. To date, CIRM has awarded over $5 billion. So that awarded meaning this board has approved all time over $5 billion. Thank you, Maria. Bigger number. And what that means is there's several ways that we get funds back and to the state to redeploy. The first is we award grants from our research budget, and during the lifetime of our award management, we recover funds. And what does that mean? We recover funds from during the time that we're contracting as we're negotiating milestones and looking at allowable and unallowable costs. We have unobligated funds at the end of an award period, or funds that perhaps were earmarked for specific things like equipment that perhaps aren't used, and then we redeploy. We also have times where awards are terminated or declined, and that's where we recover funds back to redeploy to give out more grants. So to date, at all times, CIRM has recovered $572 million. In addition, over CIRM's lifespan, we've had two different loan policies, and with that has resulted in over 77 million in loan return repayments and interest from those loans back, which we deploy back to the research budget and can allocate for more grants. Additionally, as you can imagine, as we fund research, we do receive royalties from the research we funded. To date, we've received 17.6 million, and this now goes to the patient support program that is managed by Sham and team, the patient access team, to support patients in lodging transportation needs. And lastly, we have general use, which I'll say is more to say that we get interest earned. So any of the bond proceeds that we receive and we're holding in our accounts, we earn interest on, as well as we have received donations. So interest earned to date is 69.3 million, and we have received donations over the lifespan of CIRM. Generally, these are of general use. There has been cases that they have been earmarked for particular things, which is 12 million. So moving on, the next slide is showing Prop 14 research budget allocation. As you know, there are certain earmarks in the proposition and how much we can allocate to certain areas. Of the 3.43 million for research therapy development and therapy delivery, we have committed just over 1 billion to date. For diseases of the brain and central nervous system, we have 1.38 billion, and to date we have allocated 655 million. And lastly, for access and affordability, there's 96 million in Prop 14, and we have allocated just over 2 million to date. And then lastly, in the research therapy and development bucket, we also are able to build and equip and operate shared resource labs and community care centers of excellence. And so to date, this board has approved 21 million towards the 26 million for shared resource labs and 27 million towards the 78 million for community care centers of excellence. And then this is just, since Prop 14 was initiated in January of 2021, this is giving a lifespan showing how much has been committed by the board and uncommitted. As you can see, what I'd like to focus on is in this past year in 25-26, what you'll see is our uncommitted balance after the approvals that were made today is roughly 14%. Historically, since 2021, we've been at an average of 25%. And so I attribute that to this team's well planning and managing of timing of our application reviews and deployment that has allowed us to ensure that we can allocate all the funds appropriately. So here is a review of the 25-26 approved research budget and actuals. This first column shows the budget that you approved last June, which was a total of 505.7 million. That was the largest budget we have approved to date. As of this meeting, the board has committed 373 million towards each of the pillars. And with the approvals of the PDEV awards today and the clinical trial awards today, we will end the year at $434 million. There is a variance, as you can see, of 14% of 71 million. And I want to go in a little bit deeper of what the drivers are of those main categories of clinical and infrastructure. So for clinical, that 30% variance is due to, at the beginning of the fiscal year, we had planned to have 4 clinical reviews. As this board knows, during this fiscal year, we implemented new changes to the review process, and those changes started a little bit later than anticipated. So there was one cycle that did not happen during this fiscal year. As you'll see in the proposed budget for next year, the plans for 4 reviews will occur. So that 40 million balance is due to a cycle of reviews that didn't happen and approvals that didn't come to the application review subcommittee. For infrastructure, I'm going to jump down to the first largest is 47%. The variance is due to 2 factors. So as you saw from Dr. Patel early today, the community care centers of excellence are off and running. Those were approved in this fiscal year. However, the board has opened an additional application for Northern California as we saw a gap in the applications that were approved. So there's 9 million in this budget that was an additional community care centers of excellence remaining balance that you will see allocated into next fiscal year. In addition, there was 15 million planned to deploy a support system for the community care centers of excellence. This was focused on possibly targeting community-based organizations. Due to the strategy development that happened in the Access and Affordability Working Group and the plan that was approved in December, and now with permanent leadership in the patient access team, those plans are still going to happen. However, that 15 million, we never brought a concept for that because as we were still deploying the community centers of excellence. So you will see that in future years, not in next year's budget. So now I'll review the 26-27 proposed research budget that was brought to the Science Subcommittee last month. The major drivers of this research budget are application review subcommittee approvals of grant applications for our regular development pipeline of preclinical development, the inaugural round of the RAPID program, and reviews and approvals of the DISC-4 and DISC-5 programs, the early stage research. There will be new concepts, which you just heard from Janie Byron for the data science and software engineering concept approval, and those awards will be approved during this year. And later this year, Dr. Rosa Canet-Vilas will be bringing a CLIN 2 amendment that will look at how to incorporate late-stage BLA related activities. So that is also considered into this budget. Lastly, there's a significant investment in education. The Bridges Compass Educ 8 program that was approved by the board earlier this year, the new concept, you will see funds allocated for approvals in this fiscal year. A renewal of the Spark program, the Educ 3 program. We've allocated funds for a renewal of the CIRM Scholars Program that Dr. Thomas mentioned earlier this year for the postdoc, pre-doc, and clinical fellows program, as well as an allocation for conference grants. And I'll get into the details on the next slide. And lastly is there are funds allocated for a Northern California Community Care of Excellence approval this year. So this slide shows the discovery, preclinical, and clinical budgets. The total request for the discovery budget is $136.5 million. This includes $84 million for 6 DISC-4 awards and $52.5 million for 21 DISC-5 awards. For the preclinical budget, we are requesting $146 million. That is $91 million for the PDEV program, which will yield roughly 7 to 12 awards, depending on which stage that those awards come in at. And the inaugural round of $55 million for the RAPID program, which could be roughly 2 to 3 awards. And lastly, we are requesting $160 million for the clinical 2 program, which will be roughly 11 to 20 awards, again, depending on which phase and stage that those programs come in at. And then as mentioned, we have a significant investment in education this year of $200.5 million. And I wanted to acknowledge Dr. Blumenthal, your question earlier during the president's report. One of the reasons that this investment looks so significant here is because these are 5-year awards. So the Educ 8 and the Educ 3 program are 5-year awards, and these would be a one-time RFA for another 5 years, as you saw earlier this year. And those will last roughly through 32, 33, probably get us to 31, 32. So that investment will sustain our projections at this time. We have $90 million for an extension or a renewal for the Educ 4 program. Currently, we have 18 awards, and that concept will come in September to the board for your review and approval. And then allocating $2.5 million for conference grants. So these would be our contracting with one provider to execute roughly 7 to 10 conferences through the fiscal year to support our grantee work. And then lastly, we are requesting $19 million. That is for 1 award in the Community Care Centers of Excellence Delivery, Northern California RFA for $9 million. And then as Janie just presented, $10 million for data science and software engineering, which would be roughly 15 to 20 awards. And so with that, the Science Subcommittee endorsed approval of this 26-27 research budget and requests adoption by the ICOC of a total budget of $662 million. And Dr. Imbasciani, I will hand it over to you.
Thank you, Jen. That was very succinct together. This went through the committee and got a good recommendation. So if we have a motion, we can discuss. And it's been Anne-Marie moves and Hala has a second. Thank you. Discussion on our research budget. Open to the floor. Is
looking at you,
Fabulous.
Just as thank you. Just as a historical note, is this the highest budget?
This would be the highest budget that CIRM has ever approved. So this is a landmark, I think, moment for this board.
Good. Is there any member of the public that might want to speak to the budget? If you do not, then without delay, Scott, let's vote on it.
And once again, this will be by voice vote in the room and roll call for those on the Zoom. All those in favor in the room say aye.
Aye.
Those opposed say nay. Any abstentions? And on the phone, Leandra Clark Harvey,
Aye.
Monica Carson,
Aye.
Rich Lahara. Sorry, Rich, not sure if you're muted. Carolyn Meltzer,
Aye.
Shauna Stark,
Aye.
and Kevin Zhu.
Aye.
Great, thank you. The budget is adopted.
The budget, good. We're set for another year. Let's move on with the numbers to our administrative budget that's gone through our finance committee. Michelle Lewis, are you ready to make presentation? Thank you.
I'm a little bit shorter. Good afternoon, members of the board, CIRM team, and members of the public. My name is Michelle Lewis, and I am the Director of Finance, and today we're going to talk about the administrative budget. A little bit less than the research budget. As always, we have our mission in mind when we're crafting our budgets. Today, we're going to do a brief overview of Proposition 14. We're going to review the results of the 25-26 fiscal year, and then discuss the proposed 26-27 budget. So this graph shows our $5.5 billion proposition, and these are the categories where the funds are allotted. As you just heard, there's $4.9 billion allotted for grants, and the other three categories I'll discuss on the next slide. So these are the major funding categories of the administrative budget, and these figures are as of April 30th, 2026. Our first category is capitalized interest and costs of issuance, which are costs associated with the issuance of bonds to fund our activities. First, the proposition states that all interest on any bonds issued through the 5th full calendar year from the start of the bond will be paid from bond proceeds. This is called capitalized interest. Second, there are fees that are paid to professionals associated with the sale of bonds, and those are called costs of issuance. Both of these reduce the amount of bond proceeds that we are allowed to spend. The proposition doesn't stipulate a set amount the bond authority can be reduced. CIRM had estimated that these would be $100 million, and that's what was budgeted. We are now past the 5-year mark, and CIRM's authority has been reduced by $85 million for those two purposes. Our next category is direct grant administration. There are three components to that: grant compliance, and $18.5 million of the $100 million has been spent as of April. Access and affordability, $5.5 million of the $55 million has been spent. And for grant administration, 35.7 of the $165 million has been spent. Our final category is general administration, and 45.4 of the $192.5 million has been spent. This slide shows our historical admin budget performance. So this is the budget to actuals for the admin budget. We're historically running between about 17%, which is what we're at this year. Our outlier would be that first year, which was 40%, and that's due to coming back from wind down and staff vacancies. Now we'll go over the results of the 25-26 fiscal year. So just for some budget context, recruitment continued due to new and increased business activities. We recruited 7 vacant positions, which gave us a head count of 68, and we have 4 positions currently in recruitment. We completed the first year of awarding grants under the new strategic allocation framework, with the team focused on strengthening internal business operations and cross-functional alignment. These are the results, projected results of the 25-26 fiscal year. In that first column, you'll see that the 25-26 approved budget was $34.8 million. We are estimating that we will finish at $28.8 million. And that variance is $5.9 million for 17%. That meets our average, and it was 4% improvement over last year. Some major drivers of that budget variance were that travel and training, due to our increased business operations, that it didn't allow for the time for planned travel or training. For our reviews, the program expert reviewers for award management were budgeted as a contingency. The rollout of the reviewers launched later than anticipated. However, the PDEV team recently launched their program, and they're expecting to spend approximately $6,000 in this fiscal year and an increase in next fiscal year. So we're going to talk about the 26-27 proposed budget. I have 2 views for you this year. Last year, we only had 1 view. This first view is a budget to budget comparison. So we have our proposed budget for the 26-27 fiscal year at $36.2 million, compared to the approved 25-26 budget, which was $34.8 million. and that is an increase of 1.4 million for a 4% increase, as opposed to last year, which was a 10% increase. This next slide is our budget to actual. So this is our proposed budget versus our estimated to finish. So our proposed budget again is 36.2 million. We estimate to finish at 28.8 million, which leaves a variance of 7.4 million or 20%. Some major budget drivers for this new budget are memberships and training. So we anticipate that training expenditures are going to continue at a lower rate. Therefore, we've adjusted the budget to more closely reflect the recent actuals. So we did do a reduction in the training budget. Just to note, the travel budget stayed the same. We had a very minimal increase in external services, and we actually decreased facilities by 5% due to the completion of some special projects. We are having an increase in software for IT software to support growth in staffing and new projects. And then we do propose an increase in employee expenses, and there are 3 components to that. The first being a salary increase, which includes a performance-based merit of 2% for eligible staff and a 3% cost of living adjustment. Just to note that the COLA and merit applies to the chair, vice chair, and president. This makes the increase available in the budget should the board choose to award as part of their leadership evaluation process. Our second component is the updated leave buyout for employee departures or retirement. Just as a reminder, leave buyout is vacation hours. All staff receive vacation hours that are their property. We are required to cash out those hours should they decide to leave the agency. So our calculation reserves the amount equal to 1 staff member from each cost center, as we can't predict who will leave, except for the 1 confirmed retirement that we know about. This allows the cost centers to backfill their positions without impacting their budgets. And then our final component would be the request of 1 full-time position. The risk factors for the budget are, as always, are recruitment and personnel growth and increased costs of goods and services due to inflation. So those are difficult to control, but we're actively managing those costs and we're constantly evaluating them. And in May, we received Finance Subcommittee recommendation to approve, and we are asking for budget approval for $36.2 million. Chair Bassiano.
Yeah, Michelle, that was great. Thank you. Very succinct. Chair will entertain a motion to accept. George, and seconded from Judy. Thank you. Thank you. Discussion on our admin budget? Yes, Dr. Barrett.
Just want to commend the whole team for really running a very lean operation with outstanding results. So this is a very impressive percentage.
Thank you. Anyone else have any comments? Is there any public comment on this budgetary item? You don't see any, yes, Lana, or no? Okay, Scott, we can proceed to the final budget vote.
Yep, this will be again a voice vote with a roll call of those on the Zoom. All those in the room in favor say aye. Those opposed say nay. Any abstentions? And on the phone, Leandra Clark Harvey.
Aye.
Monica Carson.
Aye.
Carolyn Meltzer.
Aye.
Shauna Stark.
Aye.
And Kevin Zhu.
Aye.
Great. Thank you. The budget is adopted.
Adopted. Music.
Just a point of order.
Yes, sir.
We're going to go very quickly to a governance subcommittee item, and then we'll have a 10-minute break to follow before we resume with the remainder.
The governance subcommittee is this contract issue. Yeah.
That's correct.
Yes, okay, because the next action item is item number 20, consideration of a contract. Judy?
Thank you very much. And back to you, Michelle, to describe this contract.
Yeah. So we're requesting the governance subcommittee's approval to continue a technical services contract with nCloud Systems in the amount of $270 to support the grants management system. This system is a core operational platform, and continued engineering is necessary to maintain system stability, modernization, and ensure alignment with current technology standards. As this contract is over $250,000, governance subcommittee approval is required per the purchasing policy.
27.
I did? What's
Thank you, that would be great.
Pat and I will entertain a motion to approve this contract request.
So moved.
Thank you.
Second.
Thank you. Any discussion? In the room, on the phone? Hearing none. Scott, will you please call the governance subcommittee only?
Yes, for those members, you're all in the room. Congratulations. So this will be simple. All those governance subcommittee members in the room in favor, say aye.
Aye.
Those opposed, say nay. Abstentions? Very good. The contract is approved.
Thank you very much. Back to you.
Yeah, thank you.
Now we'd like to take a 10-minute break if possible. So that those on the phone will come back at 2:55.
Gil. Where's Gil? Is Gil in the room? Ah, okay. Okay, everyone, we're about to reconvene. When we start, we're going to be on agenda item 17, which is an update regarding the patient perspective score. And yeah, I was just talking. This isn't official yet. Thank you. Okay. Are you ready? Okay, we're going to start up again with the last two agenda items. Number 17, Dr. Sembrano is going to talk to us about the patient perspective score in the review process.
Okay, thank you very much. Good afternoon, everyone. So I want to share with you a proposal that we have to implement a patient perspective score into the grants working group reviews. This is something that we've presented to Science Subcommittee and Finance Subcommittee. And I want to start first with our mission, because I think this is kind of core to what we do and why we do it. And because of how we've structured CIRM to be inclusive of patients and patient advocates in both our activities and our process, that's what allows us to achieve this mission. And in our conversations, thinking about how it is that we can improve or enhance the role of our patient advocate and nurse members, particularly those on the grants working group, one of the ideas that actually came from Christine Miskowski in one of those conversations was to develop a patient perspective score. So we took that idea and developed that into something that we think will be very beneficial both to the grants working group in terms of what it does, but also provide a more meaningful and enhanced role for our patient advocate and nurse members. So, when it comes to the purpose of this patient perspective score, that's exactly what is intended to do, provide patient advocate and nurse members of the grants working group a mechanism that allows to formally voice the viewpoints or evaluations of applications that are under review. Before this idea, our patient advocate and nurse members often suggested a scientific score, and it wasn't one and isn't one that gets officially recorded. And so by virtue of creating a different score that is reflective of the patient perspective, it allows to formally recognize that role and that viewpoint. Of course, the focus of the review on applications that are most relevant to patient advocate and nurse members is also one of the benefits, because we can tailor the score to focus on those elements. And overall, as mentioned, enhances the role of the patient advocate and nurse members on the grants working group. And so in terms of how this would work, just very high level to begin with, it's only the patient advocate and nurse members of the grants working group that would provide this score. And so this is somewhat akin to the DEI score that some of you may recall we utilized for a few years, in that was only provided by our patient advocate and nurse members. So it follows that example, and that has also allowed us to put this together in terms of the infrastructure that we have in the grants management system more effectively and smoothly. The patient perspective score would only be used in funding opportunities where we have formal assignments that are like our clinical program or preclinical programs, so CLIN and PDEV, but not the discovery programs. The number of applications in those are just way too many to make it practical. And of course, to follow, we have a set of specific review criteria that I'll go over that are focused on patient-centered elements that we would use to base the score on. And so that's this slide here. These are the proposed patient perspective score criteria. And in thinking about what these criteria should be, I looked upon other organizations that include patients and patient advocates in their reviews and in their process. One in particular, which is PCORI, it's the Patient-Centered Outcomes Research Institute in DC. And so they are a good example, among others, including also even CPRIT in Texas. And so from those come what are essentially very common elements in terms of these criteria. And so we talked these through with our patient advocate members on the board to discuss whether these resonate, whether these seem appropriate. And I think they do. And so these include relevance, asking what is it that whether the project addresses a real and significant problem for patients, whether the project offers a patient benefit, meaning if it's successful, would the therapy make a meaningful difference to patients? Is it patient-centered? Asking whether the proposal is designed with the affected population in mind. Patient engagement, is the patient perspective included and used to inform the therapeutic objectives? And lastly, the California benefit. How beneficial would this therapy be for the people of California? And here we were also careful to note, and we want to note it just for the public, that our intent with this last one isn't to advantage necessarily therapies that are for more prevalent diseases as opposed to rare diseases, that we value both. The intent is to look at it in a more broad and holistic way in terms of the benefit to the California patients. This is the proposed scoring methodology. And here we are proposing to use a Likert-like scale from 1 to 5 that allows us to have both a simple numerical score that we can assign a meaning, and in this case, patient value. And if we think of patient value as encompassing the criteria that I just described, you can imagine having applications that would show exceptional patient value, maybe some moderate, others that might be inadequate. And so this would be the measure that we would use in scoring the applications against those criteria. And so in bringing then that patient score along with the scientific score from the grants working group, we now have 2 different elements that will describe the merit of an application. And so in that proposed process, in terms of how do we manage having these 2 scores that we bring to the application review subcommittee, we would do the following. First is we define the fundable pool of applications, meaning those that score 85 or above, because ultimately we want the scientific merit to be a core element of what we ultimately fund. And we would prioritize applications that have both a high scientific score, or firstly, a high scientific score, as well as a high patient perspective score, because that's what we're looking for ideally, having a high scientific and patient perspective score. Now, in the case where you have an application that receives a low patient perspective score, but is otherwise deemed to be scientifically meritorious, we want to trigger a discussion to help justify why we would fund something that from the patient perspective seems to, at least by the score, not offer a good patient value. And the key points from the grants working group discussion on any differences, because we expect and we want to organize the grants working group review to bring out any potential differences between a scientific score and patient perspective score, that we invite that discussion at the grants working group meeting, so then we can share with you the key points from that discussion about why any differences, if they exist, are there, or potentially resolve any differences that are brought to the table from the scientific members and the patient advocate members on that review. And here's just an example scenario of what will come, and I'm just using some imaginary PDEV applications as an example. You would now have both a scientific score and patient perspective score that would be coming your way. And in this scenario, we have 5 applications that are all higher than 85. So this is our defined group of meritorious applications with variable patient perspective scores. So the one in the middle, the third one down, has a patient perspective score of 2. What we're proposing is that that application would merit discussion to understand why there is that difference. Of course, we would have as reference the discussion points from the grants working group meeting to help with that. The other example where this would be helpful, as you know, we often have now more applications recommended for funding than we can afford to fund. And so let's pretend in this example that we can only fund 3 out of these 5. And so knowing that you have high scientific merit, you have a ranked score from the scientific members, but then you have the patient perspective score that adds another layer of consideration that can be included in what we may debate or think about in terms of what ultimately gets funded. So that third one may, in fact, not be funded in favor of funding the one below it. And in terms of implementing the patient perspective score, our intent is to do this for the CLIN and PDEV cycles that are upcoming. So following this board meeting, assuming we have agreement, we will revise the program announcement for CLIN 2 and post that. And applications coming in at the end of July would then be subject to that patient perspective score. Those will have the grants working group review in October and come to the board in December. And the revised program announcement is going to include the process by which the patient perspective score is applied, along with the criteria that we utilize, so that applicants are clear on the fact that this is being implemented and what the basis for that scoring is. For PDEV, it will follow just behind or in parallel. Those applications are due a little bit later in October, but we would revise and post that PA next month as well. And so that is the proposal. And so I will gladly take any questions.
Judy.
You might have said it's just not clear to me about what do we anticipate the applicants should include in the application to demonstrate the patient's perspective?
Yeah, no, that's a great question. And let me go back to the criteria, because I failed to mention that aspect of it. So when we were looking at these criteria in terms of determining, what we would use as a basis, these actually align very well with the existing criteria that we also have for the value proposition that the scientific members use. So whether the project addresses a real and significant problem for patients, would the therapy make a meaningful difference to patients, is part of the value proposition and elements that applicants already include in the application, and something that from the scientific perspective is also being looked at. In terms of the patient-centered and patient engagement, whether the proposal is designed with the affected population in mind and the patient engagement and other related activities is also currently part of the existing application, and that folds into the population impact criterion that we utilize for the scientific members. So again, that information is in the application already. And so, for the most part, I think this already is covered.
Thank you for answering that. So just to make sure I completely understand, so in a sense, if an applicant application comes along, and we actually see that has quite a bit of the patient perspective together with the scientific perspective would be valuable, even if the applicant doesn't include that. Somehow we're going to.
Well, we would expect that they would include it, given that we are asking these questions in the application already, and are incorporated into the scientific scores by the scientists. And so right now, the patient advocate members don't have a formal mechanism by which they can essentially voice their opinion about these very same things. And by having this patient perspective score, they would, and you would get to see it.
That's clear. Thank you.
Good. Kim.
Yeah, I'm very supportive of this concept, and I think it really is a huge strength of the CIRM program. But I'm curious with these 5 criteria, how reviewers will necessarily generate a single score, because presumably they're not scoring each of these. They're not going to give an average. They've just got to come holistically up with something from that. And are these various criteria weighted in any way? I mean, if the first 4 were fantastic and yet it was not necessarily specifically of benefit to people in California, how would somebody score that?
Yeah, no, it's a good question, but it is a holistic approach to doing this, much the same way as we do with the scientific score. And the reason for doing that is that in some cases, one of these criterion may outweigh all the others depending on how deficient it is. If we were to assign, say, an equal number of points to each, then it sort of gives you an artificial number that may not be reflective of truly what you want to do with it. So what we want to do is continue to try this, and I think also learn from it, because I think that's going to be one of the important elements in implementing anything new like this, and continue our conversations with our patient advocate and nurse members to see, is it working? Are we, is there anything that we need to change? Or do we need to restructure the scoring in any way? And sorry, the other thing I failed to mention is that even the discussions we've had so far, I think there is general agreement that anything that's a 2 or below is sort of the threshold, I think, in just our conversations of what merits a discussion at the ARS.
Okay, good. Thank you. The order of speakers will be Hala, John, Christine, and Yael.
And me?
And, sorry, I didn't see you. Okay, start with Hala.
To follow up on Judy's comments, I still feel like scientific review is very different. I mean, science, you have patient advocates trying to decipher that from a patient perspective. And I feel like if it's just embedded somehow and they have to go looking for it, is very different than asking people to respond specifically to these criteria in a single section, whether it's a half a page or whatever it is, that would allow them to actually be much clearer, and obviously allowing your research community itself to write it appropriately for the patient and the advocates to be able to review it and understand it. So I don't know that just using the format we have will give us the ability to do this very well.
Yeah, no, I appreciate the point, because we debated that and continue to debate whether we need a specific section for this. And we have had some experience with the DEI scoring, where we also had a separate section, which we then incorporated into the application across different sections to avoid applicants repeating information that was going to be looked at anyway. So I think at least where we're starting is to point our patient advocate members to the places in the application where this exists. And this is something where we've been pointing to them already in terms of the role they play currently. And, you know, but I think it is something that we want to keep in mind in terms of if that doesn't work, we may need to create a specific section for it.
Sure. I had a similar kind of question to Dr. Barrett. Was thinking it was just going to be a component score of these areas, and then that's average. Or if not, well, how many patient or patient advocates will score this? Because the scientific score is, you know, is a summation of multiple scores, so it's an average. So you know, it takes care of the outliers. So it's just going to be one person, two person, or a whole panel.
It's
then I'm sorry. Go ahead, Nancy.
Yeah, it's going to function the same way in terms of being the median. So it'll be the median of the patient advocate members serving on the grants working group, of which we have 7.
Okay. 7, okay. And then the other thing is, I guess, kind of get into Hala's query about this score is not really adding to the scientific weight, but it's more of an influence after the scientific review. Is that correct? It's not a component, because I mean, I always thought the science was driving, you know, the research. Of course, it's got to, you know, help with patients. That's the big component of CIRM. So it's really more of a score that influences the post-scientific review versus adding weight. Like a weight means it's 5 or 10% of the scientific score. What I'm thinking I'm hearing, maybe I'm wrong, is that the scientific score is 100% of the weight, and this score is an influence once that score is done. I think that.
So let me clarify. So the scientific score is what determines the funding recommendation, and so that's not changing. We will still use 1 to 100. 85 or above recommended for funding. Below that, it's not recommended for funding. So what the patient perspective score provides is an opportunity for a couple of things. At the grants working group, to have a mechanism by which patient advocates can share and say, I think this either offers great patient value or it doesn't. And that itself should engender discussion between scientific members, patient advocates, to ideally agree that this offers a good overall patient value, also value proposition, because as mentioned, a lot of these elements are the value proposition which exists in the scientific review criteria. So I think our expectation is that in most cases, there will be that agreement. We often have these discussions already in the grants working group, but there's just no, again, formal mechanism for us to record the patient perspective part in that, because only the scientific members score. And so our hope is that that conversation is, if anything, enhanced at the grants working group, and that we bring it here too. And if we get to the point where we actually still have a difference, we can have further discussion here.
So just related to that, so let's say the score was 84 and the patient perspectives were 5. Would there then ensue a discussion in which those 5s could raise it from 84 to 85? Would that be permissible?
No, so the 84 would still be a not recommended for funding, but it still offers a perspective from the patient advocates of what they felt the patient value from that application was. And so if the board would want to consider that application, you have now 2 elements you can use in your consideration, the scientific score as well as that patient perspective score.
Yes, Chris.
I'd like to reiterate a couple of points that Gil made. All of these criteria are in the application now. They're there in different sections, and the nurse members and patient advocates who have been reviewing grants for a number of years know where they are. I'm not sure I should be given attribution for developing this, but thank you, Gil. And we'll see if it works out. If it doesn't, then we can come back to me. What I see this as doing, and Gil said this, it's giving a formal voice to the nurse members and patient advocates. We are saying this out loud in the grants working group already, all right? But it gives us an opportunity to put a numeric value to it. And it's no different than what we did with the DEI score. It was a Gestalt score. And if it's there, you know it's there. If it's not there, you know it's not there. And I'll use an example. There was a pediatric grant that didn't have a pediatrician on it. Well, how is that patient-centered? It was a clinical grant that didn't have a pediatrician on it. That was absurd to me, all right, in terms of having input from providers. There have been grants that were developed with nobody talking to a patient or a community advisory group, and that was really obvious. The grants that do it really, really well, everybody agrees. The ones that are borderline, we are having discussions now. And I've been on the grants working group, I don't know, 5 years, I guess now. I've seen a marked change in the scientific reviewers in terms of the engagement in talking with all of us about the relevance, the benefit. Is the route of administration going to be something that's so arduous that no patient's going to sign up for this or a family member can't support it? So I think this is going to provide a really, really valuable contribution to our discussions. And I'm with Gil, we'll see how it goes, and where there may be discrepancies, I think we can talk about them.
It's Yael.
I just want to really echo what you just said. I think as a patient advocate, it does give me an opportunity to raise my voice and contribute to the application process. And I do think that overall, there has been continuity in it's a great application or it's not, and they haven't thought about the community in general. So I think it's in there. Some people do it better than others, but I appreciate the voice on which I can speak to.
I was also going to mention, this provides an opportunity for, at the ARS, another factor to consider in applications that we fund or don't fund. Because as our dollars dwindle and the applications continue to rise, we will be faced with more applications that were meritorious to fund than we have money for. We saw that today. So you can imagine a patient perspective score also adds to a determination the ARS can make about whether or not to fund an application.
Thanks. I just don't want to belabor the points, but I agree with all of the merits that have been brought forward by the other patient nurse advocate members. And I felt so sort of relieved in a way when Gil brought forward this idea that had come from other patient advocates, because I feel that this is actually creating significant more rigor in our role in the grant review process. And I think to the point of like where the content is, I think many of us in the room have been reviewing these grants for many years, as Chris mentioned, and we do know where to go and find it. And I do think it's important that it is embedded so that the scientific reviewers can also take this information into consideration, because they should. The one thing I just really wanted to drive home is that I think this is adding a tremendous amount of rigor to our grant review process for an organization that has chosen to have patient advocate and nurse members as a part of their grant review.
Great, good, thank you. Yes, Isabel?
I'm actually a little bit disappointed because I thought that was what we were doing when we added DEI several years ago, to add rigor, to bring in the patient voice, and to try to move science towards change. When you tell me that the science 100% score is all that matters, or that's the winning number, I keep thinking of the wonderful project that was in here today on HIV, and we being told that it was too much trouble to include women. That's the bottom line that I heard. That's not scientific rigor. And if you had a patient advocate at the table who would score that and say, women get HIV too, where are they included in this? Send it back, and when you can come back with a plan, no matter how much work it is, to include another group, an underrepresented group, I call it labor intensive, but those are the groups that are missing regularly and historically from research. And they're not at the table and they're not heard and it doesn't count against scientific rigor. And then I'm disappointed because I thought that that's where we started and we're headed when we put DEI into the mix. What I'd like to know is if the new, I'm going to say it's a new application, will now say, perhaps in a box, we have established this patient advocate or patient, what do we call it? Patient perspective score, which we will take very seriously and consider this, this, and this. And I appreciate, Christine, that in fact, or Elena, that in fact you're seeing shifts. Well, I thought we started to see them, 4 or 5 years ago when we put DEI in, that the science was recognizing, yeah, we got to listen to the patients and the patient advocates. So I'm glad we're going here again. I would like to know if we're going to see a way in which this makes a difference. Metrics, remember, we talked metrics? So I'm hoping that we're going to see this, but I would like to know that the patient advocacy and the patient's opinion and concern counts as much as scientific rigor. Otherwise, what's science without the people?
Thank you very much, Isabel. I'm going, because almost everyone in the room has spoken, I'm going to open it up. Is there any member of the public that would like to address this issue? No one in the room and no one with their hand raised. Any final comments on this? This is not an action item. So we're not voting on this. No final comments? Okay. Good. Gil, thank you very, very much. You clearly engendered the most discussion of the day. I'm going to invite our Director of Communications up, Amy Adams, again for her update on communications.
Members of the board, my esteemed colleagues on the CIRM team, and any member of the public who's still with us, it's an honor to be here today. This is your afternoon of data and metrics and budget, because I'm going to present metrics and budget of the communications program. So when I came and presented my budget to you 6 or so months ago, I said there were four steps to my strategy. The final part of that strategy is measure the impact. And I kept saying, I'll come back to you with that. Stop asking me. I need to get the program up and running before I can measure anything. So you're going to get all of that today. So first of all, I want to start with the communications and outreach mission, which is driving awareness of and support for CIRM's programs and accomplishments. And that's really important for this particular presentation because we have to know where we're trying to go so we understand what we're trying to measure. And in this case, I'm measuring more the awareness of and less support for. We'll get there, but for right now, let's just see who knows us. So we're really looking at volume. Volume is the point of this presentation. Okay, so this is the olden days, the way the communications ecosystem used to work, and the way I have always given presentations on communications metrics, the way many of you have heard many of these presentations in your own organizations, from this organization. And these olden days ended maybe 6 months ago, and we'll talk about why. So in this glorious past, the website was kind of a real focus for communications. You were really trying to drive people to the website, and the website is where you told a bigger story about yourself. And you had email newsletters and social media and other things. You're looking at the metrics of how much your social media is driving people to your website. And then you're looking at how many people are getting information from your website. So you're really looking at the website And you're hoping to see a line that goes up and to the right, in terms of the number of people going to your website. And the way people were getting to your website was search. Certainly in the last decade, people put thousands, hundreds of thousands of dollars into optimizing their websites so they were easy to search. And then off to the side here, I have both PR and YouTube. They were direct conduits to people. Those were less conduits to the website. That has changed. And I'm unable to click this and tell you why. It's not changing. It has changed. Yay! All right, now we're with the modern era. And you'll notice it looks super complicated, and that's because it is. It's a changing ecosystem. It's not going to be this messy forever, but right now things are a little confusing, and I want to talk to you about why. And as with absolutely every industry everywhere, it is AI that is disrupting things. So let's talk about what that means. That doesn't mean that CIRM is using AI to push information out, or we're not doing anything different. But what you'll notice, you see all those happy people up there at the top of the screen? On that last slide, they were being driven to the website. That's where they were going for information. And now what we're finding, people refer to this as Google Zero, where no one's coming to the website through search. I'm also hearing the term zero-click searches. So people do a search and they don't click on anything, and that's because they did a search and AI just gave them an answer. So that's the new world that I'm looking at, is how do I get my information to people using sort of their AI as a tool. So I'm not using AI. They're going to whatever Google, and they're doing a Google search, and they probably don't even know that they're using Google in AI mode. Okay, so let's talk about what that means for me, and also what that means for the metrics you're going to see today and what we're going to do going forward. Okay, so a lot of our effort now is thinking about, if people are getting information from AI, what does that mean? How do we get them the information about CIRM that we want them to have? So think back to the story that I've presented in the past. We're still trying to tell that story of this organization that is doing research in a data-driven way and providing therapies to people. It's the same story. We just want AI to deliver that story. Here's how we do that. AI is scraping the website. AI is reading all of social media. AI is reading the newspaper, gathering information, and then delivering it to people. So now my whole team is really pivoting to thinking about how do we feed AI the right information. A lot of that is going to hinge on the website, and we're going to talk more about that in this presentation. So I've circled the things I'm going to mostly talk about today. I'm going to talk about the website because until 6 months ago, it was really important to us. I'm going to talk a bit about the blog. I'm going to talk about PR. I'm going to talk about that little podium at the top there is community outreach. What do you see that's not circled? It's that gigantic AI in the middle, right? The thing I'm telling you is so important. I'm going to talk a lot about AI, but not in terms of metrics. And the reason for that is because at this point, there isn't a great tool for measuring what AI is delivering to people. There are some tools coming along, and I think by next year's budget, I will be asking for money to do that. I want to add one thing here, and that is, I'm telling all of you this, and maybe this is new news to some of you. All of your organizations are dealing with this same thing. So you're going to be hearing this from other people, and all of your communications teams are having the same, like, oh no, AI moment. So what this all means, I kind of just went through this. I think the key things here to know is that, I showed you that AI is scraping earned media, the newspaper. So we have hired a PR agency, and PR is going to become increasingly a focus for my team because it is increasingly a focus for AI. So that is something you will see in my budget that I do have. My slides just did something. We are going to be optimizing the website to be better scraped by AI. There are tools out there to help me with this. Everyone on my team, or most people on my team, we are taking classes in how to better leverage this new world. So you will also see that in my budget, that I do have a line item for a web vendor, and we will be doing some things to improve the website. I'm gonna move on. So, okay, and the metrics, I'm going to talk about the website, the blog, social media, and outreach. Okay, this is let me demystify this slide a bit. So here's what we're seeing. I mentioned that in the past with the website, we're looking for a line that goes up and to the right, right? We want more people coming to the website over time. And if you look at the website traffic with a blind eye, which I did a couple months ago, it goes up and to the right, which is awesome. And that's the kind of blue dots. And then I looked deeper, and what I found is that we had a 2,000% increase. It was 2,000-fold, sorry, increase in traffic from one particular region of China. And we used to have no traffic from that region of China. So I asked AI what was going on. So this is a worldwide global phenomenon that started about roughly September of last year, that it's essentially, from what I understand, AI scraping places. And it's bombarding all of your websites are doing this. It's harder to see who the humans are. These bots, these are all bots. They're not doing anything bad. They're just scraping our website for content, just like AI bots are supposed to do. What it means is this is probably the last time I'm going to show you a slide of our website traffic, because I don't think it's going to be meaningful anymore. The orange line is my best attempt to weed out who the actual humans are. And it goes up a little bit, but not much. You know, in the past, you really looked at that traffic to go up. I think it's going to go down. I think we're going to get less and less traffic to the website and more trying to do the same kind of monitoring through AI. So you can quote me on this. I am betting that this is the last time I show you this slide. Okay, we're gonna pivot to the blog, and this is something that I know is not apparent when you look at the website. The blog is actually a different platform. So when you go, if any of you have looked at the blog on our website, it looks like it's on our website because you click on things on our website and there's the blog. It's not. It's two completely different platforms, and we've just kind of set it up so that the blog looks like it's on the website. But it's not. So any optimizing we do to the website is not going to help our blog. And the blog is the main source of our storytelling. So this is blog traffic over the last year. Everything I'm showing you for a year ends in April, and that's because I put this presentation together in May, because I first delivered it at the beginning of June. So we only look through April. I'll just tell you the trend continues through May, but we're just going to talk about April. Okay, a couple things about blog traffic. I just finished telling you that you can no longer trust metrics around who's coming to your website. And then I told you that the blog is a separate website. So can you trust any of this? The blog right now, I think because it's separate and not terribly optimized, gets so little traffic that even the bots don't care. So we're hoping to change that. But in the meantime, I think those are actual humans on those little charts there. So let's talk about what you're seeing. This is traffic to our blog, which is our main source of storytelling over the last year. And I made them blue and yellow just to indicate blue is the first half of the year, yellow is the second half of the year, right? And what we see is second half of the year over the first half of the year, 122% increase in views. And just for those of you paying very careful attention to your math, in the memo my team put together, I think we said something like there's a 60-ish% increase year over year. We were looking at just April over April. This is looking at 6 months over 6 months. I think it depends on how you slice and dice it. It's about double in the last year, right? So that's, we feel good about that, gonna say. And then the red lines here are the dates when I presented my evolving strategy to you. And I will say that as that strategy evolved, we were making changes to the blog. So if you look at this, as we rolled out the strategy, the blog went up. And I could stand right here and say, we're doing awesome, and you should give us lots of money in our budget. The story is actually more complex and really interesting, and does point to some of our asks in the budget. And I'm going to turn it over here. Do I have Scott here? So Scott Hadley is our new Director of Content Strategy. So he, from now on, is going to be overseeing our storytelling and our social media. So I'm going to let him talk to you about some of those metrics and also what they mean in terms of what we'll be doing going forward.
Thank you so much, Amy. Appreciate it. I was hiding behind the flag. I don't know if that's a metaphor for something, but this is also my first rodeo with you, so apologies off the bat. So these numbers are really small, right? They're when you look at the big picture, but they're still interesting. And we did look at whether this was related to bots. By the way, not all bots are bad. And as a matter of fact, you kind of want your website to be scraped by bots because that means that content is being used. And it's not really for us. We're not trying to aggregate people to come to our site and buy something, right? We want our information disseminated in the best way. So we want to encourage that. The blog is interesting because that is a place where we can do more fully formed storytelling, and we can experiment a little bit, and we can do it quickly. So I'm going to look at a very small dimensions of some of the stuff we're doing in communication. So let's just look at that blog traffic. And we're wondering, why is it that we saw that increase? It's a simple question. So if we speculated that because of the way staffing had worked, our frequency of posting on the blog varied over the last year, and there were a couple months where we didn't have anything. So we wondered, is this just related to frequency? You have more shots on goal, more traffic. So we looked at that. We had more, 2 posts in September. We didn't have any posts in November, and then we had 1 in February. Clearly, that's not playing a role. But maybe a better way of thinking about this is nothing that we do in comms, not in these individual channels, is done in a vacuum. There's other things that are going on. And so you are all well aware that some things were happening towards the end of last year that related to CIRM and the beginning of this year. So if we plot some of those things, and these would be mentions in social media and mentions in print or other traditional media. So I'm just overlaying these 3 specific events or moments, I should say. So the openings of the CCCEs, the rapid announcement, and the rocket announcement. Those were covered in the media. If you look at social media mentions, I think Rapid and Rocket were the first or the highest and the 3rd highest impression rates for social media during that time. Why am I pointing this out? It seems sort of obvious. I think why we're pointing this out is that as we improve our messaging and coordinate all the different channels that we're using, we're empowering ourselves to improve how that message gets out. And that's going to take us to social media, where there's a lot of opportunities there and maybe opportunities we haven't yet tapped. Maybe you are aware, but our strongest platform right now is LinkedIn. It sort of makes sense because of who we're communicating with. We're communicating with people who want to know about our grant making, either their own or opportunities. And we are also, more professionals maybe are looking there. And so that's that big swath of blue that you see there. And I just wanted to draw your attention to 2 others. Those were the orange and is that red? I'm sorry, I'm a little colorblind. But so that's Instagram and YouTube. I think someone's going to bring up TikTok, but we can get to that later. Why am I bringing to your attention that? Because what this is showing is an engagement, which is just a measure of how often someone clicks on a post or interacts or shares with it in some way. And what you see is like an uptick in Instagram and YouTube. If I look at just video views, it's much more profound. I think the increase in YouTube was like 26%. Doesn't sound like a lot, but that's a lot. Again, these are small numbers, but why am I showing this? Is because the opportunity is there, right? We should maintain that presence on LinkedIn, but the audience on YouTube, the audience on Instagram, the audience on TikTok is a little different. And what we communicate there is different. And so what we see is an opportunity to expand who we're communicating to through those platforms. It's going to maybe change some of the form in which we communicate. And so we have a great social media manager, Christina, and she's been busy doing a lot of other things up to this point, and now she's experimenting more with short form video on those platforms and experimenting and seeing how that works. And so now I will turn it back to you.
Okay. When I've been thinking a bit about all this data that that my team has been collecting. And I feel like it's sort of a rising tide lifts all boats. We're not exactly sure which thing is exactly working, but we are seeing an overall increase in views to our various pieces of content. So it's a little frustrating that we can't pinpoint a thing that worked, but we're seeing a rising tide, which seems like a good sign. Okay, so this, I'm pivoting now to our outreach work. This is the state of California, as you can see. The blue dots are the places where our outreach team touched this year. The size of the dot reflects the number of events in those locations, not the size of the events. And what's not on here are things like you heard today that our chair and vice chair did some really interesting presentations in the past month or so. I think that's so important for us to be doing. And that's not on here. I happen to know that at least one of our education trainees did a Rotary Club presentation recently and had reached out to us. That's not on here. So there's a lot of things going on that aren't here, and that will only grow as our CCCEs are doing more and more outreach. So I think as a data visualization, this is my kindergarten starter version. But I think over time, what I'd like to represent is more holistic, not just, where my outreach people were and coordinated, but more of like where is CIRM. All that said, even if you showed every single thing everyone had done, there are some gigantic empty yellow spaces on that map, and I think it's a huge missed opportunity. So one of the things that you will see in my budget is I currently have one full-time outreach person on my team, and then there is one contractor who works in LA. That's why you see so many dots in San Francisco and LA. It's because that's where they are. In my budget, I want to hire 2 additional outreach contractors. And we want those people to be in some of those yellow swaths of the state so we can be reaching out much more broadly. So the key takeaways before I turn to the budget, we are going to be spending time this year optimizing all of our materials, especially the website for AI. And that's going to be a lot of background work, but I think is incredibly important. We are going to be moving the blog to the website. I think that will both optimize that storytelling and optimize everything for AI. And I just want to thank Marguerite Casilla has very graciously agreed to be part of the team that's going to provide feedback as we move that over. She made the dire mistake at a recent meeting of speaking up about the blog, and you speak up, you get voluntold. We are going to have a really consistent focus on messaging across channels. I think that's part of the rising tide is that we are being very consistent across channels now, and I think it's helping. We are going to do continue to do a push with PR, both because it is a direct way of reaching people in communities we don't always reach, and because it's good for AI. Video, as Scott pointed out, is a real missed opportunity, and I'm really excited about some of the work Christina Smith has been doing. And our outreach efforts need to be beefed up. So with those takeaways, I'm going to try to click through the budget. Okay. So I doubt any of you can read this on the screen, but you should all have it in the memo and in the slides in front of you. And what I've indicated is just the line items on my budget and the ones that are increasing or staying the same. And for right now, I'm just going to talk about a couple that are increasing instead of going through everything. Forsmarsh, that's the PR team. We hired them in October of last year, and so their contract ran from October to the end of June. And so next year increases, but it's just a scaling increase to reflect the fact that they're starting in July instead of October. We are doing some focus groups around California. This is with the urging of Pat, who has some great ideas on that. So we will be doing focus groups to understand what the people of California are interested in and what kind of messaging resonates with them. So that's a one-time budget item. We are adding 2 outreach contractors. That is an increase that I hope is long-term. And then there's a small increase in the budget item that we use to pay for outreach events. So if one of our outreach people goes to an event of some kind and they have to pay for a booth, that costs money. Well, and if we have 2 additional contractors, they're going to pay for additional booths or what, entry fees or whatever those things are. So that's an increase. And then we have a temporary increase in personnel. I've got a person on my team now who's on a fixed term contract, who's really thinking deeply and I think doing terrific work, helping us communicate more effectively about our programs and policies and processes with the awardees and potential awardees. That seemed like a real need. So we're filling that need with a short-term position. And with that, I say thank you. And do you have any questions? Yes.
Yeah, Judy. Yeah.
Maybe way back we talked about this. Please remind me about how do we define our KPI, the key performance index? What do we call as a success about the efforts pays to help people understand CIRM? Or can you elaborate that more?
Yeah. So I don't have my strategy in front of me, but I've got KPIs in the strategy document. And it might be easiest if I share that with you because off the top of my head, I don't remember all the KPIs. But the KPIs are things like, well, I used to have a KPI around increased visits to the website, but I'm going to modify that one. But it's year-over-year increase in engagement on social media, year-over-year increase. I look at things like are people requesting our presence at events? Because that's sort of an indicator, not just were we there, but also an indicator that people know about us and they want us to come to the event. So there's KPIs like that.
Yeah, that's what I'm thinking about now with this evolving environment, right? We really need to adapt that. If you look at the California map, no surprise, the concentration will be where people do use social media. But we actually want to reach out to the place. People don't typically look at the phone to check on those information. So I feel like we need to redefine the KPI a little bit about multiple different kind of media, maybe certain place emphasizing social media, I don't think there's anything wrong, but maybe for certain area, based on the demographic background, we just need to do that differently. Maybe even traditional way is the key.
Yeah, I think KPIs should be evaluated every year. Yeah, I agree.
Isabel?
Amy, I was actually hoping to see a budget year to year.
Oh.
And to see an increase, how it increases in staffing in the particular places so we can measure over year if in fact the increases in budget made a difference in the work we had to do. So it would be nice if you could send us one.
I could do that. Absolutely.
Okay. Pat?
So, the map that you showed where the events, personal contact looks exactly like the map of where CIRM funded investigators live.
Yep.
It looks identical,
Yep.
right?
Mm-hmm.
Smaller number of circles, but they're exactly and geographically in the same place. That was just a comment because that's where people go. And it's challenging to think about a strategy that would really broaden it, except including more domains that include CCs and other activities that are not necessarily related to where a grantee is or a grant receiver is located. So that's one thing. The other is that you're developing stories, and when we're hearing stories, it might be helpful for board members who are sometimes asked to talk about CIRM to actually have a resource that tells a common story, so that they don't have to spend time figuring out what story they should tell, or do screen captures off of the website, which is what a lot of people do, which is not fabulous. But having a resource or a toolkit that would and have it distributed rather than just, well, you can go to the website and find it, and you just click 5 times and then you can find it. And it's going to develop because the stories are developing, and those stories in particular, like most of us can tell the stories that we heard about today. We may not include all the data. We may include just the punchlines about a grant that got funded and in a disease that is X, and this is where it has transformed hope, kind of thing. So that's would be helpful. And maybe it exists, and I just don't know.
Yeah, I mean, what's going through my mind, so we have a toolkit, we have a deck with a narrative and all the, links to, well, that's what I was just thinking, like meaning like me and my team know where it is. So I think I need to work with.
Unwilling to share.
Well, you know what I mean? I'll share. No, I think that's a really good point, though. I mean, we have all of those tools, and I think I linked to them one presentation ago, maybe two presentations ago. They're linked to an agenda that you probably can't find anymore. But why don't I work with Claudette and just make sure that those resources are somewhere where you guys can find them? Because there's a lot of materials available and more coming.
Pat, can I say in Amy's defense that in fact she did have a presentation which she sent to me when I went to the Oakland Rotary, but I adapted it.
Yeah.
Both for me, but also for things I thought were critical in talking to a business group. So it is there. I think the basics are there, Amy. I just.
One of the things that's changed quite a bit is the board hearing stories now, which are really powerful. And the communications group could take those, because you see those slides, and extract what you think would be palpable and understandable information that would allow us to tell one or two of those stories, in addition to the core story about what CIRM is, right? And helping people understand what CIRM does. But as more and more of those come, they give us more and more opportunities to tell stories that resonate with folks without having to have the same scientists. They're not going to go around the state.
Yeah, I think that's a great idea, and I'll work with Claudette to get that done.
I have a comment. Just for those folks in rural areas, internet access is very limited and very costly. I think people utilize radio discussions and TV. Not sure if you have a plan for that.
Yeah. Yeah, WhatsApp. They use WhatsApp. It's hard to get into that. Yeah, no, I think you've hit on a really hard point. It's hard to reach those people. I mean, I think a couple of things we try to do. We have been trying to pitch stories in small regional publications, because I think people do read in small communities, they read their local paper. So like around the season. Oh, really? So there's that's interesting, because there are some free ones that we've tried to get into. Yeah. Okay.
I would use
Yeah.
Yep.
me, especially in the areas where the CCAs are popping
Yeah.
up. Just a blip of a clip in between a Spanish TV station or something about a cure for something, basically.
Something
Just
relevant
one
to
little
them.
clip. I mean, that's all it takes to have people start thinking about it.
Yeah. We did get one Spanish language radio pick up a story of ours, and I was really excited about that. It'd be fun to get more. But no, I think that map of California, it's exactly what we all expect, right? There's no surprise on that map, but it is pretty stark. I mean, it really makes you think we've got to figure out how to reach some of those other regions, and it's a lot easier to reach LA and San Francisco, I'll say. Yeah.
Amy, I know we're adding two additional outreach consultants, and outreach is near and dear to my heart, as you know. So I think in looking at that map, I would hope, and I can't remember, I know I saw the statement of work, but I do not remember, but I hope that the consultants would be in those areas, so that that's the work that could be done in those areas, which would help, Adriana, some of what you were referring to about how do we get into places where someone might not have internet access or be able to, get online or whatever it might be. And that would bridge a gap in by doing the events there and going to established groups where we would do the outreach.
Yeah. Yeah. So exactly. we're not specifying, we're not designating where we want the people to be. I think we're hoping to get the applications and try to map out who the best people are and where those people are, and try to get the best people in the best places rather than like predetermining where they're going to be. I will say, I hope I'm not, if Aditi is listening, I hope I'm not getting too far ahead, but I think we might send the statement of work out through the trainees as well, because we've got a bunch of students out there, who many of whom have graduated, have had an education in how to do outreach, and are like smart young people who live in remote parts of the state. So I think they could be good options. But yeah, Maria, exactly. And then the one thing when I was looking at you earlier, I was thinking, I have to remember to say this. In the statement of work for those contractors, we have a line that says something like multilingual, a preference for multilingual. We're not specifying what language, we don't know what we're going to get, but we have specifically put that in.
Good. Dr. Barrett?
Yeah, thanks very much. As always, your presentations are very clear and informative. And just to respond to what Pat said, I did go and give a presentation to the Rotary Club and felt extremely well supported by Aditi, with a very useful PowerPoint with stories and everything. As it turned out, they couldn't connect to my laptop, so it didn't work anyway. But it was a good template for me to then talk to the group, and it was a small group, so it was informal. And I think, we can all do more of that. But I have no answer to this whatsoever, but beyond the metrics that you've talked about, the social media and stuff, how are we actually measuring understanding of what we do? Because in my one anecdotal, very limited experience, 7 o'clock on a whatever morning it was, with some really delightful gentlemen in the Rotary Club, they didn't know anything about CIRM, even though I was there talking about CIRM. They didn't know anything about regenerative medicine, even though 2 of them had family members with rare diseases. They didn't know that cell and gene therapy was an option, and that people were actively doing research to try and cure rare diseases with these modalities. So we've got and that's in Sacramento. So we've got a real uphill battle, and I don't know how we measure this, whether we're moving the needle, but just reading stuff doesn't mean that people understand it.
Yeah, no, I agree completely. That's why I kind of weight things, and I've been thinking about my map a lot. Like, how do I better display what's really going on? Because I think those of us who have done sort of Rotary Club presentations, right? It seems like a really small audience, we're really achieving much, but they're super engaged, and you've got them for 20, 25 minutes. And I think we come out the door with a glimmer. So that's great, but it's small. And then there's other times where we go to these huge events and someone comes by our booth and they grab a flyer that they then throw away, and it's hundreds of people. And did we really move the needle? And I think that question, the question about KPIs was fantastic. Most of my KPIs, I put a lot of thought into like, what's not just like performative, right? You can cheat and, do better on social media. There's a few topics I can think of. So you can cheat and increase those numbers. I'm trying to think of KPIs that aren't cheating, but that actually reflect something. But I don't think my KPIs are perfect, and I welcome ideas, because it's tough.
Isabel, yes?
One of the things that might help Amy is even whether it's a fair or, a table, if someone stops by, you do give them a little survey. But also at the Rotary Club, where I equally had some technical issues, it would be nice perhaps at the very end to give them 5 questions.
Yeah.
What did you learn?
Yeah.
Can you recommend somebody we should talk to from here? Just 5 questions, but a sense of starting to get that kind of data about, oh, okay, I'm smarter now and I'm good. So,
Yeah.
and so you add it at the end of the presentation so they can do it right there.
I think that's a great idea. I will say I'm gonna speak to Scott, not behind his back, because he's right here. When I was looking to hire him, one of the things I really loved about him, made me want to hire him, is he kind of came at me with all these questions of why aren't we doing things? And one of the things that he's come up with since then is like, what are you doing to measure? How are you figuring out, like, is there a survey? Is there a thing? Is there something like is there a thing you make them do to indicate that something happened. And I loved that thinking and I wanted that thinking on my team. Yeah.
If I may just add one point to clarify why I asked KPI, it's not so much about if you or your team performing or not. I really think we want to be focused what do we want to achieve first.
Yeah.
Because otherwise we'll cover too much, right? So really is that the patient group? Is that certain age group? Is that even, I mean, I personally will be fine about if social media is where we are going and we target the generation knowing this so they can help their family who that do not use cell phone, that's also fine. And I just worry about we got too ambitious and make you not be able to achieve what CIRM really need to as a tool for outreach.
Good. Well, thank you very much. Thank you, Amy. Thank you, Scott, for your presentation. If I don't see any more comments, so I would like to, thank you. I think we're set. Are there any, this is the part of the agenda at the end here, any general comments on our review process, the ARS, from either board members or the public? Yeah, and is there someone from the public who would like to make a comment of any kind, whether it is on the agenda or not? Good. The microphone is.
And you have 3 minutes. Go ahead.
Okay. My name is Magualida Seño. Now? No? Okay. Okay. Now you guys can hear me. Okay, my name is Magualida Sarceño. I'm the mother of Ciara Sofia Sarceño and Dwight. My two kids were diagnosed last year. They both have Batten disease, the CLN6. Ciara is 6 years old. My son Dwight is 4 years old. And they are outside. I would like for you guys to meet. So basically, this disease take away from our kids their sight, fine morals, the ability to solo walk. Ciara is been losing partial. She still can see. She still can walk some days. This gene therapy is the only chance that she have to live longer. According to what we know, is they can only live it until 8, 10, 12 years old. Ciara can still join the gene therapy. And we know we live across the bridge in Fremont. I would like to ask for some, I don't know if you guys have any kids, grandkids, if they're healthy, but that's good. But I have 2 kids who are affected, and this is our only chance to get this grant, this money for her, because in 6 months, her life can change completely. In 6 months, it can be too late for her. My son is 4 years old. But this is Ciara. I would like to ask for you guys to reconsidering the grant for the gene therapy for Batten disease CLN6. And we sent letters 48 hours ago. I don't know if you guys take a look of all the moms that we struggle with our kids. Ciara cannot sleep. There is many nights that we have to wake up at 1, 2, 3, 4 a.m. and make sure she needs assistance. She wants to walk, but sometimes she's falling because her body is not responding. This is her only chance that she can have. She's not the only one. We have other 15, 17 kids who can join this gene therapy. So, like I say, if you guys have any kids, grandkids, would like for you guys to take in my place and reconsider in this grant. Thank you.
Thank you very much for your comment and for coming over from Fremont. We've reached the end of our agenda. I'd like to tell the board members that we will convene our next meeting of this board in San Diego. Do not come to San Francisco. We'll be in San Diego. Hear that, Hala, John? On Thursday, September 4th, we will meet 24th, I'm sorry, September 24th. We'll meet at the Manchester Grand Hyatt and we will start at 9 in the morning. And thank you very much for your attendance and participation. This meeting is adjourned.